Hello students, welcome to Vedantu NEET English. I am Miss Gopika, your own biology master teacher. I hope I am audible and visible to all of you. Please let me know in the chats if I am audible and visible to all of you. Yes. Wow, I love the energy in the chat box that ma'am, even if it is 3 a.m. we are ready to study. Even if it is 7 hours we are ready to study. Wow, I love it. Wow, very nice, very nice. Yes, exactly. This is the seriousness that we were looking for. And yes, I think students have very close to that, right? Awesome. Good evening, good evening. How is everyone doing? Happy New Year. We did meet on New Year. I had posted a video wishing all of you New Year. But other than that, we did not meet. So happy New Year to all of you. It's a little late New Year, but still wishing all of you happy New Year. It's a very special year for all my students as well as for us, because my students are going to write NEET 2025 this year. And we pray and hope nothing happens like 2024. Let the paper be easy. But other than that, let nothing other things happen. And let the paper not be very easy also, where the competition increases. So my students should be able to write, right? NEET English students, that is Vedantu NEET English students, should be able to write. That is the only goal. So let it be easy for you people. And wishing all of you happy New Year from our end to all of you. I hope all of you had a very safe and nice New Year. Yes. Shall we start, students, the class? So as all of you know, today we are going to do genetics, right? Genetics, which is one of the biggest units, also the highest weightage unit, right? As you can see, 60 marks. Again, this is less. It can go above 60 marks because 9 to 10 questions maximum from molecular basis of inheritance and 7 questions from principles of inheritance is the maximum that has come. Usually it is 8, 6, this range. Okay. So we'll always get more marks from this chapter. So I'm going to start with molecular basis of inheritance, and after that Baswa sir is going to start. And those who haven't liked the video, those who haven't subscribed, quickly do that. Also share it with your friends. Okay. Done? Shall we start? So yes, molecular basis of inheritance. So we are going to learn this chapter in such a way that you will never forget this chapter. But if there are any students who is learning this chapter for the first time, or revising it after their 12th now, let me tell you, it will take you some time to understand the chapter. It will also take you some time to remember the concept. It is very normal, like in day-to-day acid structure of polynucleotide change, all the polynucleotide, all the experiments and the mechanism of replication. Okay. Second part of the class will be where we will learn how replication happened, then transcription, genetic code, translation, gene regulation, under gene regulation, lac operon, HGB project and DNA fingerprinting. So it's a lot of work for me, right? Is there any lag? Is there any lag? Or is it only for one person? Please let me know. There's no lag in my phone. So just please check your internet, students, once again. Yes, the likes haven't increased. What is happening? Yes. Okay, now I can see. Yes. Is it lagging, huh? For all of you? One second. What is happening? What is happening? Yes, the likes haven't increased. What is happening? Now it's okay, no? No lag, no lag. Okay, yeah. In my phone, it is showing fine. I don't know what is the issue, okay? I think it should be fine, students. Some chuttu-muttu internet issue will happen. Okay, yes, HGB, DNA, fingerprinting. Okay, now discovery. Okay, we are going to start the chapter. From now on, no talking, only listening to me. If there are any people who are talking unnecessary, please call out their name in the chat box. I will remove them from the class, okay? Because I will not be able to see all of your chats. Yes, students. So, when we talk about molecular basis of inheritance, the main concept that we are going to talk about is nucleic acid. Someone who has all the information that is required for us. Now, all of you know that whenever a person gives birth, no, we very easily we go and talk to them and we'll be like, you resemble exactly like their mother. This child resembles exactly like their father. But how does this resemblance happen? It is all coming from one place called where inheritance happens, right? Something is getting carried away from the parent to the offspring, and that is the reason why they are looking similar, why they have some characteristics. Now, as we grow up, okay, you will start noticing that you are exactly or similar to your parents in many characters. Okay, maybe you get angry for small things, maybe you shout at or small things are not kept in proper places, you start getting irritated about it. These things are carried away, but these things we learn. Now, there is a person, or like let's say a molecule that has all the information, okay? A molecule that has all the information that is required. Just give me a minute. This mic is Yes, done. Okay, if there's any lag, please let me know. I'll just inform the team also. Okay, fine, no problem, no? Okay, yes. So students, a molecule, okay, a molecule that has all the information, that has all information. Who is that? All the information, and all this information will be protected by this molecule as well. Okay, so all the information of a cell, who is that? Is the genetic material. It is the genetic material. And when I talk about genetic material, I need to understand one more thing, that all the organisms have nucleic acid as their genetic material, right? All organisms have nucleic acid as the, like Gopika ma'am, GM, no, like that, genetic material. Okay, genetic material. Students, all these notes will be available on Vedantu NEET English Telegram as well as Biology by Gopika. So quickly go and join these two telegrams for getting all the notes. Okay, yes, all the organisms have nucleic acid as the genetic material, and I want one of you to put the target in the chat box so that they can follow the target. Now, when I talk about nucleic acid, what am I talking exactly about? When I specify nucleic acid, I am talking about two main people. Who are they? One is called the DNA and one is called the RNA. Okay, one is called the DNA and one is called the RNA. So obviously you people know the full form of it, deoxyribonucleic acid. Deoxyribonucleic acid and ribonucleic acid. Ribonucleic acid. What is the biggest difference that you people spot? Come on, tell me in the chat box. What is the biggest thing that you notice in both of this? Ma'am, here you have mentioned deoxy. That means something is lacking. Yes, exactly. One oxygen is absent here. Absence of an O group. Okay, one oxygen is absent here, because of which your DNA becomes more stable, because oxygen is highly reactive. Because of the absence of this oxygen, the DNA becomes highly stable. And if you look here, ribonucleic acid, that means here there is a presence, okay, presence of the OH. Okay, that means again oxygen group. Now tell me, all of you in the chat box, in what position this oxygen is present and absent. That is, ma'am, if you draw a structure like this, it is going to be absent in the, look here, I have drawn a sugar. In this you will notice, in the second position there is going to be a H and a H, CH2OH. That means in the second position there is going to be an issue that you're facing. That is, what is the issue here? That is, ma'am, here H is there, but in ribonucleic acid OH is going to be there. Correct? Second position. Very good. That can be asked as a question. Okay, second position can be asked as a question. Now students, when I talk about the word nucleic acid, you should also know the discovery of it. So we will learn the discovery also in this slide itself. Okay, now who discovered? So there was a person called Friedrich Mischer. He was actually working on WBC. Okay, WBC, again on owl cells they cell, it is basically WBC cell, okay, white blood cells. When he was working on white blood cells, he noticed that there is one cell or there is some molecule that has all the genetic information, or that this has something to do with genetic information. So he called it nuclein. What did he call it? Nuclein. So a person called Friedrich, okay, Friedrich Mischer found nuclein. Okay, when he was working on what cell? WBC cell. He also found protamines. Okay, protamines. Now this main thing is what? He found nuclein, but there was another person called Altman. Who? Altman. Altman was like, this nuclein, I feel, shall I write it with the same color? Yes. Altman found that this nuclein had acidic. Nuclein is acidic in nature. So he ended up calling this nuclein as nucleic acid. Discovery is clear? Yes or no? Why did he call it nucleic acid, ma'am? Because he noticed that nuclein had an acidic nature, because of which he called it nucleic acid. Is this clear? This is how the word formation happened. That is how you're telling about DNA and RNA. So two people who are mainly behind this is Friedrich Mischer and Altman. Mischer was working on WBCs and he found a molecule that was very different and which was able to have all the genetic information. So he called it nuclein. Altman noticed that this nuclein has acidity, or it is acidic in nature. So he called it as nucleic acid. And the two nucleic acids are your DNA and RNA, where all the organisms have nucleic acid. Majorly 99% of the organism have DNA as the nucleic acid. Okay, almost organisms. Okay, almost all. Okay, most of the, I'm writing ORG, most of the organism have DNA as the genetic material, but RNA, even though it is not a genetic material for all the organism, but for some viruses like your TME virus, like your COVID, all of you remember, no? And your HIV. So there can be some viruses which have RNA as a genetic material, but mainly it is going to act like what? Like a catalyst, like an adapter molecule. So it is going to do other functions to help us continue with our central dogma. Clear? Thumbs up in the chat if all of you understood so far. This is just the general idea of it, okay? All these notes will be available, so you please scribble and write your lecture notes. But I don't know why many students have not attending. Students, please share the link with your friends and ask them to join, okay? Or share it on your Instagram and ask them to join. Yes, this is clear? Yes, virus, viroids, all of this we have studied in biological classification. Later also we studied about retrovirus, okay? So some viruses definitely have RNA as a genetic material, but most of the organism have DNA as the genetic material. So this ideology is clear. Now, what is exactly this made up of? Ma'am, when you're talking about nucleic acid, can you give us the components of the nucleic acid? What are they made up of, right? That question is going to come. So students, remember, nucleic acid is mainly made up of 3 components, okay? All of you tell me the 3 components in the chat box. It is made up of 3 components. Okay, 3 components. What are these 3 components? Ma'am, first component is going to be definitely the sugar molecule, right? The sugar. Second one is going to be nitrogenous base. Okay, nitrogenous base. And what is third one going to be? A phosphate group. A phosphate group. Anyone has doubt in this, right? Very good, very good. Atluru and Bhargavi, very, very good job on answering the question, okay? Yes, tell me. Okay, very good. So, 3 components are mainly there to form this nucleic acid, that is going to be your sugar, nitrogenous bases and phosphate group. Now, when we go into detail of understanding all the 3 components, we should understand that when these 3 components are going to be together, that means a sugar molecule, okay, a sugar molecule is going to be attached with a nitrogenous base, okay, nitrogenous base. And to this nitrogenous sugar molecule, there is one phosphate which is going to be attached. Okay, there is going to be a phosphate going to be attached. So let's say these are the 3 components that is going to form your nucleic acid. What are you going to call all these 3 components together as? You're going to call it a nucleotide. What are you going to call, students? You're going to call it a nucleotide. Clear? Okay. Bonding, everything I'll teach you. And I will teach you how, why is it 3 dash, why is it, 5 dash, how phosphodiester bond, everything in detail I'll teach you with the diagrams, okay? All you have to do is trust the process and listen to me till the end, okay? And make notes on your NCERT, clear? You don't have to worry about, ma'am, is ma'am going to cover this, is ma'am going to cover this, is this extra information? Whatever is required for you, I will be giving, and also you will be solving questions, okay? Done? Yes, very good. So, let's say this is a phosphate group, this is a sugar molecule, this is your nitrogen bases, okay? This is your nitrogen base, this is your phosphate, and this is your NB. Now, students, this together, I'm going to call it a nucleotide. But there is one more terminology which is called a nucleoside. Now, who is going to form a nucleoside? When a phosphate sugar and nitrogen base is there, it's a nucleotide. But when sugar and nitrogen base becomes friends, okay, so what is going to happen? In a trio, two people will always be closer than the third person. Always, have you noticed, if you have three friends, two people will be very strong friends. One will be there, but like that closeness won't come. Same thing here also happens. Sugar and nitrogen base are big friends, okay? They're like full trio. So these two together is going to end up forming your nucleoside. Pushing who? Phosphate to one side. Pushing phosphate to one side, okay? Thank you, Ajay. Yes. So pushed phosphate to one side. So in the trio, one person is outside, okay? These two are always texting each other. These two concepts are clear. Now, many nucleotides. Let's consider this entire thing. Okay, let's consider this entire thing as one orange color dot. One nucleotide, another nucleotide, another nucleotide. Many nucleotides together is going to form a polynucleotide chain. Clear? Polynucleotide chain. Polynucleotide chain. Okay, clear? Okay, so many nucleotide together forms a polynucleotide chain. This is going to form your nucleic acid. Now, let's go into depth of this. Now, students, when I'm talking about sugar, remember that you're talking mainly about two sugars. Which are the two sugars? In the chat box. See, please understand, those who have learned the chapter, I want you to constantly keep answering in the chat box. Don't expect that let ma'am teach, I'll just listen. Those who know, please continue typing. As soon as I ask you which are the sugars, your answer should be in the chat box. Clear? Okay, so it will become like a revision. Those who are studying it for the first time, please listen. Now, students, sugar is going to be two of them. Okay, one is going to be a ribose sugar. Okay, one is going to be a ribose sugar. And one is going to be a deoxyribose sugar. Deoxyribose sugar. So very simple. Ribose sugar means one oxygen is going to be there in the second position. Deoxyribose sugar means one oxygen is not going to be there. Simple, no? Simple things. So here, as you can see here, this positioning, here one OH is going to be there. Here one OH is going to be there, okay? And same way here, I'll just draw it very thin, students, otherwise I can't show you the bonding. Okay, yes. I hope this is visible. Yes. So this is your first carbon, second carbon, okay? On the second carbon, there is going to be an OH. This is going to be the fifth carbon, CH2OH, okay? I cannot write the whole thing, so I'm just writing it this. Now, deoxyribose sugar, what is going to happen? Same thing, ma'am. You continue here and H is going to be there. Again, third carbon, CH2OH. Okay. This is the ribose sugar and the sugar. Clear? Yes. Fine. Again, there is lag. Okay. Giridhar. Yes, yes. Okay. You don't have to spam, dear. Once you tell me, I understood. Okay. Don't have to tell multiple times. I saw. Okay. Fine. Baswa sir has taught biomolecules. You can go and check it out. Okay. Is there a lag? Please tell me now so that I can check with the team. I don't think there's a lag. Okay. Fine. Yes. Fine. So, this is your sugar. Okay. With OH. This is going to be ribose. This is going to be H. Okay, deoxy sugar. Now, when I talk about nitrogenous bases, remember there are two main people that we will detailedly learn about it. And then you have your phosphate group. Phosphate group is nothing complicated. You have P, okay, or you can just write PO4, okay, with a double bond, okay, with a double bond, which will go and bind with this. So, bonding, everything I will teach you right now. So, students, please do understand one thing, that when I am talking about this second position hydrogen, I am mainly focusing on few things like stability, okay, Stability is one main thing that I'm focusing on. Because of the absence of the oxygen group, it increases the stability on the DNA molecule. Not only the stability, it also chemically and structurally better compared to RNA. That means chemically and structurally the stability is there. Clear to all of you? Yes or no? Yes. So, yes, this gives the acidic property. Very good. So, they are chemically and structurally less reactive also because of the absence of the oxygen group. Okay, less reactive. This I'm telling about DNA. Okay, fine. This is clear. Polynucleotide chain is clear. Now remember that when you're talking about polynucleotide chain, you will always remember two things. What is that? Ma'am, I always notice that one is going to be from 5' to 3' and one strand is going to be from 3' to 5'. Okay, I don't think these two are straight lines. Okay, straight lines, but we can adjust. Okay, is this 2 cross? Okay, wait students, I'll only draw. Anyway, I'll draw it cross, so I'll only draw these 2 strands, right? So one is going to run 5' to 3', one is going to run 3' to 5'. Now the reason for this we are going to understand in the bonding. So I just want you to remember that, okay, both the strands are there, they're going to run anti-parallel to each other. Okay, anti-parallel to each other. Okay, clear? Yes. So they are going to be anti-parallel to each other. Okay, anti-parallel to each other. Yes, fine. Done, done. Now, going further about the nitrogenous base. Very important concept, nitrogenous bases. I know all of you know purines and pyrimidines, but nitrogenous bases, okay, there's no lag, no? Okay, nitrogenous bases is important. Yes, we are going to come to that. So, nitrogenous bases are 2 of them, purines and pyrimidines. Purines and what? Pyrimidines. Important, very, very important. Now, I have given trick to remember this, but more than that, you have to know a little bit more about their details. Okay, little bit more about their details. What is that? That, ma'am, purines are a 9-membered ring. 9-membered ring. That means, students, name is short. Purines. Name is only 6-letter word, but then 9-membered ring. Okay, but look at this pyrimidines, long fellow, but this fellow is a 6-membered ring. Okay, 6-membered ring. Now, one more important thing that can come as a question is, other than 9-membered ring, what do you know? That is, you have to remember that in the first position, third position, 7th position and the 9th position, there is going to be nitrogen groups. Okay, there is going to be nitrogen groups. And in pyrimidines, you have to remember in the first and the third position, there is going to be nitrogen groups. These 2 things you have to remember, very, very important. Easy, no, to remember? Yes, very, very good. Some of you are answering, very nice. Yes, good. Now that you know this much, we can go into the detail of it. That is, what do we know about purines? Okay, we know that purines, when I talk about purines, purines have 2 main people coming under it. Who are they? Adenine, pure as gold, guanine. Adenine and guanine. How do I remember this, ma'am? You are going to remember it as, the trick is going to be pure as gold. Okay, pure as gold. Whenever you start, finding friends, whenever you go into, something like you want something really bad, okay, you tell your mom, no, mom, my heart is pure as gold. Remember, like that, you try to open, okay, your heart and you show, no, ma, I'm so pure. Please give me that phone. Please buy me that iPhone and give. I will not sell it. I am sure. So, pure as gold, you remember like that. Pure as gold. So, P for purines, A for adenine, G for guanine. Okay, pure as gold. Okay, ma'am, understood. Moving forward. Then we have who? Ma'am, we have pyrimidines, right? We have pyrimidines. Shall I write it here? Okay, we have pyrimidines. So, students, if you have learned biomolecules thoroughly, this chapter also is going to be easy for you because that chapter is the foundation for this chapter. Okay, yes. Now purine, students, you're going to remember cut. Okay, you're going to remember what? Cut. C-U-T. C for cytosine, C for cytosine, U for uracil, T for thymine. Thymine. Ma'am, how can here 2 come? How can here 3 come? You can ask me this question. Yes or no? So here remember that we are talking about uracil. Uracil is going to be present only in RNA. Thymine is going to be present only in DNA. But cytosine is going to be present in both RNA and DNA. That means what did you understand from this? Thymine, which is going to be there in DNA, will be replaced by uracil in RNA. Simple. AUG is there. Instead of that, what will be the U? Instead of U, T will be there in DNA. That's all. Okay. So, cytosine will be going to be present in RNA, DNA, uracil only in RNA and thymine only in DNA. Is this clear to all of you? So, cut you will remember. Now, one more very important thing that you have to remember is when we learn Chargaff rule also you will know. But before that, just understand. Okay. Now, students, remember, see when I am talking about your purines. Okay, when I am talking about purines, I am talking about a 9-membered ring, and these people are going to bind. These people are going to make bonds with the sugar molecule, right? Sugar and nitrogenous bases are there. They are going to be what? Nucleoside. Phosphate is going to be pushed apart. But please do understand that the DNA's backbone is made up of phosphate and sugar. Nitrogen bases are going to be projecting outside. That means, let's say this is the DNA molecule. Okay, this is the DNA molecule. Okay, this is the DNA molecule. Now, remember that when I'm talking about nucleoside and tide, one thing that you have to remember is the sugar and the phosphate is going to make the backbone. Okay, all this is going to be sugar and phosphate. They make the backbone. But who is going to make the projections? This base pairs are going to make the projections. Any doubt in this? This is the line of NCERT and also a salient feature of your DNA. Okay, salient feature of your DNA. Okay, those who haven't liked the video, quickly like the video as well so that the energy will be continued. Yes. So students, now most important, purines are there, pyrimidines are there. Ma'am, how are they related? Right. So remember, purines are going to bind with pyrimidines. Pyrimidines are going to bind with purines. Okay. So they are going to bind in a pattern, and the pattern is called as, okay, the pattern is called as at Gopika ma'am's class. Right now, all of you are in my class. At Gopika ma'am's class, remember like that only. A, T, G, C. At Gopika ma'am's, ma'am, you keep in mind, okay, Guru Dakshana, keep it in your mind. So at Gopika ma'am's class, so A, T, G, C, this is how the binding is going to happen. Adenine is always going to bind with the thymine with 2 hydrogen bonds. With thymine with 2 hydrogen bonds, and guanine is going to bind with cytosine with 3 hydrogen bonds. So at Gopika ma'am's class. Thumbs up in the chat. All of you put the at Gopika ma'am's class quickly in the chat box so you memorize as well. Okay, you memorize as well because you will just listen and then you will forget. So remember, at Gopika ma'am's class is the binding. That means how purines and pyrimidines are going to bind with each other. ATGC at Gopika's class. So adenine will bind with thymine with 2 hydrogen bonds. Guanine will bind with cytosine with 3 hydrogen bonds. Done, done, done. This is how the binding is going to happen. So you can imagine that sugar and phosphate is going to make the backbone. So these people are going to project inwards. So adenine will bind with thymine with 2 hydrogen bonds. Guanine and cytosine will bind with 3 hydrogen bonds. Clear? I'll move and stand so you can see it. Okay. Fine. So nitrogenous bases, I have given you a little more understanding about the 9-ring structure and about the nitrogen groups because I have seen 1 or 2 questions in the mock paper as well about the nitrogen groups. Okay, so just keep in mind. Clear? Fine. Very good. Now, moving forward, now I am going to teach you some important bondings. Very, very important bondings. Okay, ready? Yes, good. Yes, good. Okay, listen carefully. Now, As I told you, nitrogen base plus sugar is going to form a nucleoside. Now, whenever you see a nucleoside, you should be able to identify it with the suffix that is going to be there. That is, it is going to end in INE. Okay, adenosine. That means INE, if it is ending, that means it's a nucleoside. Okay, it's a nucleoside. Now, nitrogen base plus sugar plus phosphate is going to end, it's going to be called a nucleotide, and it is going to end in 8. Okay, it's going to end in 8. Now, remember students that when you study further about regulation of gene, you will come across methyl guanosine triphosphate. That means MGPP. Now, how did this PPP come, ma'am? What is this? That is, thank you, Supriya. Yes, nitrogen bases sugar and phosphate. Remember, they're going to form nucleotide. Other ways, in other words also, I can tell it. How? That is, nitrogen base plus sugar plus 1 phosphate. Okay, 1 phosphate. I can call this nucleoside monophosphate. 1 more phosphate, nucleoside biphosphate, triphosphate, right? So, here also remember, this nucleotide can also be called as nucleoside monophosphate. Thumbs up in the chat box if you understood this, because we are going to learn these terminologies in charging of tRNA, and further also we will study it, locomotion and movement. One or two times you will hear the word triphosphate. Then don't get confused. Understand that nucleoside, with the nucleoside, if I keep adding phosphate, instead of calling nucleotide, I can call it triphosphate, biphosphate, monophosphate, depending on how many phosphate groups have been added. Okay, fine. Moving forward. Now, bonding, very, very important. Bonding is very, very important. Okay, have we reached the target, guys? Yes, Madhumita, tell me in the chat box, have we reached the target? And also, let me drink some water. You also drink. Okay, Bama, we haven't come to the bonding, dear. I will tell you the bonding. Ninja has put thumbs up in chats. Look here. Yes, adenosine triphosphate we tell, no? So here what is going to happen? Okay, what is going to happen? I'll tell you. See, nucleoside means what? We pushed one person to the side. In a trio, when it is a trio, you will call it tide. Nucleotide. Trio, TT, tide. Okay. Now, I pushed phosphate, telling that, hey, I don't want your friendship, man. So, phosphate is one side. Okay, phosphate went to one side. So, we call it nucleoside. So, for nucleoside, you are going to add the suffix. Okay, you are going to add the suffix called INE. I-N-E. I'll show you some examples, don't worry. I-N-E. Okay, now, if it is a nucleotide, that means nitrogenous base, sugar and phosphate. All the 3 trios of your friendship is there. Okay, all the trios are going to be there. It will have a suffix called 8. Okay. Now, instead of calling it a nucleotide, I can call it one other way. What is that? Nucleoside monophosphate. Yeah, 1 phosphate I added. If I add 2 more phosphates, what will I call it, Bama? Tell me in the chat box. Ma'am, you will call it triphosphate. That's it. Okay, I'm not talking about the bonding, I'm just talking about the terminologies. Okay, because they can ask you questions and they can ask you if is it a nucleoside or a nucleotide. Any question can come, no, we cannot expect. Okay, yes, how much? Let me check the target. Those who haven't liked, quickly like. I'm checking your let me see your enthusiasm. Oh, conjuice people in the class. Yes, done. Chalo, okay. Okay, very nice. Now we will learn the bonding. Listen to me, 5 minutes. Listen to me, don't listen, don't text, don't talk to each other, just listen to me. What is going to happen, students? This is a pyrimidine or a purine. Just look at the number and tell me, is it a purine or a pyrimidine? All of it, quickly in the chat box. Just put PU or PY. This much or you put. Yes, guanosine. Yes, very good. Thymidine, cytosine. Yes. Tell me, is it a PU or is it a PY? Purine or is it a pyrimidine? Ma'am, obvious ma'am. Very good. It's a purine. Why? Because it is a 9-ringed structure. 9-membered ring structure, right? So now understand, students, this is the nitrogenous base and this is a sugar molecule. Now you know to identify it by looking at the number. Now, sugar and nitrogen base together is going to form a nucleoside. But how do they bond with each other? That is important. Yes, ma'am, it's important. Remember that from the 9th position of your pyramidin to the 1st position, forget about the 9th position. Just remember that to the 1st position of the sugar, if the bonding is going to happen with the help of a nitrogenous base, the bond formation is going to be N-glycosidic linkage. What did I say? If a nitrogen base is going to bind or make a bond with a sugar, it is going to form an N-glycosidic linkage. Simple. Yes, ma'am, simple. Now, sugar is there. Yes. Nitrogen base came and attached. Happy. N-glycosidic linkage is formed. Done, done, done. Now, to the sugar, phosphate has to come and attach. Yes, ma'am, phosphate has to come and attach. To the sugar's 5th carbon, phi dash end. To the phi dash end, a phosphate is going to attach, and that is going to be with the help of a phosphoester bond. That is going to be with the help of a phosphoester bond. So what and all bonds did you study? 2 bonds. N-glycosidic linkage and phosphoester bond. How is the N-glycosidic linkage? I am a nitrogen base. I bonded with a sugar. This bonding is going to happen with an N-glycosidic linkage. To which carbon? To the 1st carbon of the sugar. Okay, that is why we put 1 dash, 2 dash, 3 dash, 4 dash, 5 dash, because here also numbering is there. To differentiate nitrogen base numbering and to differentiate your sugar numbering, we put the dash. Okay, now I am a nitrogen base, I attach to the sugar. Now, the sugar is here. Sugar has to, or phosphate has to attach to the sugar. So, phosphate will come attached to the sugar with a phosphoester linkage. It's a phosphate. So, remember, phosphoester linkage. Okay, phosphoester linkage. So, 2 bonds, N-glycosidic linkage and phosphoester bond. Done, done, done. 2 bonds are done. Simple. Okay, simple. Now, remember that when we go forward, you should know how many of them are combining. Okay, this is all the diagrams that I drew in the class. So, instead of, it will take a lot of time for me to draw this again. So, that's why I have taken the, snip itself and come. So, look here students, what is going to happen? Now, I am going to teach you what is phi dash end and 3 dash end. For this, I want all of you to give me 5 minutes of your attention. Otherwise, you will not understand this. Okay, listen. Now, this is a pyrimidine. No one has a doubt. Pyrimidine and your sugar molecules have bound with N-glycosidic linkage. This sugar molecule has got attached with a phosphate with the help of a phosphoester linkage. Now, this entire thing is one nucleotide. Okay, this entire thing is one nucleotide. This one nucleotide, if it has to combine with another nucleotide, it will combine with the help of a phosphodiester bond. With what? Phosphodiester bond. Clear or not? Clear or not? Is it clear? So, listen to me carefully. One nucleotide, another nucleotide, I need to combine because my ultimate goal is to form a polynucleotide chain. Polynucleotide, many nucleotides together form my nucleic acid. With one nucleotide, what will I do? So, if I want to combine, this is one nucleotide, this is one nucleotide, and this pen is the bond. Okay? Nucleotide 1, nucleotide 2. Okay? Nucleotide 1, nucleotide 2. If they have to bond, the pen is going to be a phosphodiester linkage. Okay, phosphodiester linkage. Now, you have learned 3 bonds. Okay, now, listen carefully. How can I tell it is phi dash end or 3 dash end? For that, you have to understand. Students, in this entire setup, you will notice that the phi dash end, okay, here, you notice a free phosphate in the phi dash end. Yes or no? This is the phi dash of the sugar. On the phi dash end, you notice a free phosphate. Do you see that phosphate going and attaching with another sugar? No. Phosphate is free, sitting chumma. I bounded with the sugar, I am peaceful. So, a phi dash end will have a free phosphate. Now, how do I tell which is the 3 dash end? Look here all of you. If you look at this whole setup, if you see this whole setup, you will notice in the 3 dash end of your sugar, there is a free OH group. Clear or not? Yes? Okay, clear or not? Perfect, very good. So, on the phi dash end, you will notice a free phosphate group. On the 3 dash end, you will see a free OH group. So, if I draw this diagram only, you will understand. Otherwise, you will not understand. I can just verbally tell and go. Okay, I will be like, okay, I told. But then, to make you understand, you should remember that if you look at this place, you will notice that there is a nitrogenous base, a sugar and a phosphate. You will notice that this phosphate is not attached to anything. It is a free phosphate. That is going to be the phi dash end because it is attached to the 5' sugar. 5' word comes from where? Sugar only, because in the sugar only we have 1', 2', 3', 4', 5'. Now, if I go down all the way, here do I see any dash? No, it is just 1, 2, 3, 4, 5 in the case of your nitrogenous bases. Okay, here you will notice that the 3' OH is free. Okay, so that 3' OH will form the 3' end. Fine. Clear or not? Because students, always remember this 3' OH is going to help for us to bind with the next phosphate. So that 3' OH is necessary for a phosphodiester linkage, phosphodiester bond to be formed. So that 3 OH is very important. Same way, phosphate also is very necessary to continue for the next bond to be formed. So these two people are forming a very important role. So if this fellow is free, I will call it at the 3' end. This fellow is free, I will call it the 5' end. Done? Okay, yes, you don't have to, biomolecules, you have to know the structure of adenine, guanine, cytosine and, okay, but try to find a trick to identify the diagrams. Okay, some tricks are there. No, now when I teach you human health and diseases, I told you morphine. Okay, how you remember with a heart diagram, you remember with the M diagram, like that. Make a crack, like make some trick out of it. Otherwise, you will not be able to remember the diagram, students. So much stress will be there, you will forget the diagram. Okay, if this is clear, I'm moving forward. So one is going to be 5'. So this is going to be it with a free phosphate. Okay, free phosphate. And on the 3' end, what is going to be there? A free OH is going to be there. These two people are necessary for the next bond formation. And 3 bonds we studied. When a nitrogen base combines with a sugar, it's an N-glycosidic linkage. When a phosphate combines with a sugar, it is going to be formed with a phosphoester linkage. When 2 nucleotides are combining together, it's with a phosphodiester linkage. That's all. That's all. Okay. This chapter weightage is around 40 to 48 marks. Okay, 48 marks. Okay, now students, some examples as I can show you here. Look here, nucleoside, nucleotide, nucleoside, nucleotide. So I told you nucleoside ends in "ine". Okay, ends in "ine". So adenosine, guanosine, cytidine, uridine, thymidine. Okay, INE it's going to come. Now I'll share the PDF with you, that time this image will be clear. Okay, tide, what I told you, it will end in 8. So adenosine monophosphate, guanosine monophosphate, cytidine monophosphate. Can you understand why I taught you monophosphate? Okay, it's for you to remember this. Uridine monophosphate. So 8 is going to come. This is in ribonucleotide. That means this is, they are talking about RNA. Okay, RNA. Now here if you see, what is going to happen? Here also guanosine. So deoxyguanosine. Deoxyguanosine, deoxyadenine. Okay, adenosine. So like that. Here also deoxyadenosine monophosphate. So we will write it DAMP. You have learned no AMP, ATP when we study glycolysis, your electron transport chain. So same way here also deoxyadenosine monophosphate 8. So tide is 8. Okay, and side is ine. Is this suffix clear to all of you? Then only you will be able to identify in the question if they have given a nucleoside or a nucleotide. Okay. If that is clear, we will go to the salient features of DNA. Bonding is clear. What is a nucleotide is clear. What are the components of the nucleotide is clear. Structure is clear to all of you. Yes or no? Anyone has any doubts so far in the bonding, in any of this? Please let me know in the chat box. Okay, if nothing is there, I'm moving forward. Salient features of DNA. Whatever you studied so far, you're going to add it as the salient features. What did you study? You studied that the backbone of the DNA, okay? Backbone of DNA is made up of what? Is made up of sugar plus phosphate. Okay, sugar plus phosphate, and bases are projecting inwards. Bases are projecting inwards. Correct? First point clear? Very, very good. Second point, DNA is a double helical structure. Double helix structure, and this was proven or this was found by 4 people. Watson and Crick, you always hear their name, but you should also remember there were 2 other people which can be asked in the question. Watson and Crick, Wilkins and Rosalind Franklin. Students, all of these people, okay, all of these people with the help of X-ray diffraction. With the help of what? With the help of X-ray diffraction. With the help of X-ray diffraction, found out that the DNA is actually a double helical structure which is twisted in a right-handed fashion. Okay, right-handed fashion. So, all these people, out of which these 3 got recognition. Okay, these 3 got recognition, that is Watson, Crick and Wilkins got recognition, but Rosalind Franklin did not get recognition. So, these 4 people did X-ray refraction to confirm, okay, to confirm the structure. Important point, please remember, to confirm the structure. Next thing, as you people told that DNA, okay, I am writing it here only, it is in right-handed fashion. Okay, right-handed fashion. Okay, clear? Then, what do you know? There are purines and pyrimidines. Remember the same thing only. Adenine will bind with thymine with 2 hydrogen bonds. Okay, that is also one of the salient features. And guanine will bind with cytosine with 3 hydrogen bonds. Okay. At Gopika ma'am's class, don't forget. Yes, it is a double helical structure. Yes, right-handed. Yes, Franklin died, so that's why she could not get the recognition, and because of which others all got and she did not get. Okay, as I told you students, 5' and 3', so that is also one point, that they are anti-parallel. Okay, they have anti-parallel polarity. Anti-parallel polarity. Yes, tell me about the helix and the pitch. Yes, all of you tell me in the chat box. All of you know the answers, okay? Your answers should come faster actually. Right, in a 360 turn, okay? Remember that in one turn, there is going to be 10 base pairs, okay? One turn, they are going to have 10 base pairs. Base pairs means your adenine, guanine, they are base pairs, they are bases, nitrogen bases, they are pairs, right? Yes, very good. So, the pitch of the helix, pitch of the helix is going to be 3.4, right? 3.4. Very good. Yes, 3.4 nanometer. And students, remember, the distance between the bases can be calculated with this two only, that is 0.34, okay? 0.34, just do 3.4 divided by 10, okay? Yes. Thank you so much. Yes, thank you. Okay, fine. Fine, fine. So fast, 1 hour went, no? So fast. So fast. That's why this chapter is going to take all the time. Yeah, and approximately, students, remember, approximately your DNA is 2.2 meter long. Okay, 2.2 meter long. Approximately, it's 2.2 meter long. So, all this is what you have to remember mainly. Main thing is you have to remember that DNA is a double-stranded structure which is wound around each other. So, we call it the double helical structure, which was found by Rosalind Franklin, Watson, Crick and Wilkins. Okay. So, these people, through the help of X-ray diffraction, notice the shape. Okay. Yes, distance between them is 0.34 nanometer. Very good. That you can just do this, divide and find the calculation, convert the nanometers and do the calculation. Okay. Now, other than this, you have to remember, most importantly, these 4 values by heart. Okay, these 4 values by heart. What are the values? That is, this is the organism and the length of the DNA. In some places, you will see nucleotides, that means they are single-stranded. The places where you see base pair, that means they are paired. Okay, they paired. Okay, yes. So, bacteriophages 5174, okay, is going to have 5386 nucleotides. Okay, there are many techniques to remember this, but I do not recommend any of the techniques because you should be really good at maths to remember the techniques. Okay, so to remember the trick, I should give you another trick. So we will cancel on the trick. Please write and learn this. Okay, 5386 nucleotides. Now bacteriophage lambda is going to have 48,502 base pairs. That means this is not single stranded, this is double stranded because it's a base pair. Base pair only happens when both the strands are there. Now E. coli 4.6 into 10 to the power 6 base pairs. Then a haploid content. Haploid content of human DNA is going to be 3.3 into 10 to the power 9 base pair. If it is going to be the diploid content, it is going to be 6.6. Okay, 6.6. Clear to all of you? Please remember. Yes, very good, very good. So we have to remember this whole box. Very importantly, they can give you match the following. They can give you anything. So it is the PYQ and it could be asked this year also. Because many students will skip this thinking that, oh, who will study this 5-6 numbers. Okay, so you study and get that 4 marks. Okay, now as I told you students, already you understood the base pairing. Okay, connecting to the base pairing, there's one more topic that you have to learn, that is the Chargaff rule. This is named after Erwin Chargaff. He came up with this rule telling that, see, even though purines and pyrimidines are there and they're also going to do base pairing, there is one thing that number of purines or the ratio of purines is always going to be equal to the ratio of pyrimidines. Okay, pyrimidines. Thank you so much, Karthi. So remember that the number of pyrimidines and purines are going to be equal. Now let's say 5 boys are there, 5 girls are there. It's easy for me to make a pair out of them and ask them to dance for annual day. But imagine 7 boys are there and let's say 5 girls are there. It's in uneven numbers. Now how will I make pairs? So my concept here also is base pairing. Adenine should always bind with thymine. That means adenine and thymine should be equal in number. So Chargaff told that there is going to be a rule called as the Chargaff rule, which is going to state that the ratio of purines will always be equal to the ratio of pyrimidines. Clear to all of you? That means what am I talking about? Ma'am, you're telling that adenine and guanine should always be equal to how many cytosine and thymine is going to be there. Okay, I'm not writing uracil here. Correct. This is correct? Yes, ma'am. Pakka correct. But remember one thing, that is A and T should always be there. Okay. G and C should always be equal. Correct? G and C should always be equal. But never ever understand, never ever think that AT, that means this ratio and this ratio should be equal. Should it be? Should it be? Not necessary, no? Let's consider. Okay, let's consider this is NEET aspirants. Let's consider this is board aspirants. Okay, okay. I am telling you, NEET aspirants, we are going to do a dance. So all the NEET aspirants should be in pairs. So your numbers should be equal. That does not mean the number of NEET aspirants and the number of board aspirants should be equal. There can be in a chromosome areas which is AT rich, and there is also regions where it is GC rich. So it does not mean that the combination, the base pairs have to be equal to each other. Just that adenine has to be equal to thymine, guanine has to be equal to cytosine. Simple. Don't write mistakes and come. Many students, what they write? They write AT equal to GC. But why they are both ratios should be equal? A and T should be equal, G and C should be equal. Correct? Okay, these two ratios should be equal. Adenine, guanine should be equal to cytosine and thymine. Fine? Okay. Yes. So this is the whole concept about your A and T being equal and G and C being equal. And students, remember that these people are bound with the help of hydrogen bonds. because I did not tell you, no, which bonds? Hydrogen bond. Did we miss something? Okay, done? No doubts? Okay, perfect, perfect, perfect. Okay, moving forward, students. Let me drink some hot water. Please wait for 1 minute. Yes, done. Okay, now packaging of DNA. Very important concept. Now, I told you the DNA is 2.2 meters. Now, I have to keep it inside a nucleus which is around 10 to the power minus 6. Now, you tell me, is it possible? Is it possible? Now, I am telling you that into a small, okay, let's say, yeah, into a small, let's say to this, okay, to this knob, I'm telling you, you have to put a 2.2 meter long thread. You'll be like, ma'am, what is wrong with you? Studying, you're not working or what? Okay, so same thing here also, students. Approximately DNA is 2.2 meters. Now, this 2.2 meters of DNA has to enter into a nucleus, which is again approximately 10 to the power minus 6. So, this is not possible. Practically not possible if I have to put this entire DNA into this nucleus. Correct? But everyone are telling that genetic material is always inside the nucleus. Ma'am, then how is it possible? So, your question will come, how did this happen? Yes, for this to happen, we are going to take the help of some special proteins, okay? Special proteins for packaging called as histone proteins. What proteins? Histone proteins. What is the charge of DNA, students? What is the charge of DNA? Ma'am, DNA is obvious. It is negatively charged. Okay. DNA is negatively charged. So, protein will be positively charged. Proteins are made up of amino acids. Anyone has doubts in this? Protein is made up of amino acids. Amino acid, two rich amino acids that are going to be present is going to be lysine and arginine. This has been asked as a PYQ before also. So, just remember this question and keep. Okay. Lysine and arginine. Okay, students, just like when you're packing your home, okay, when you're shifting somewhere, you call packers and movers, no? Just like that only, we have bought histone proteins which are going to help in the packaging of my DNA. This 2.2 meters of DNA, they promise that they will make sure to pack it in such a way that it will enter into the nucleus. We were like, okay, fine. So, they told, okay, we have to bring some more people into it because it cannot be done by just one person. So, what we are going to do? Yes, these are the basic proteins. Very, very good. Lysine and arginine. Yes, correct. Negatively charged, protein is positively charged. Yes, good job. Now, students, remember, lysine, arginine is clear. Now, they are telling that histone proteins, we have to bring more people. We were like, okay, fine. They told, okay, we will first bring 4 people. Who are the 4 people that they bought? They bought H2A, okay, H2B, H3, H4. These are the 4 proteins, histone proteins that they bought. Correct? Yes or no? Yes, ma'am, correct. They told that they will bring H2A, H2B, H3 and H4. Then last they told, okay, we will bring one more person that is called as the H1 protein, which is also called as the linker protein. Also called as the linker protein. So now that they told, okay, 5 members are going to come for the work, we asked them, how are you planning to do the work? Then they told us that they have a plan. The plan is going to be simple, that H2A is going to be here. Okay, H2A. H2A is going to bring his twin also. Okay, one more person is going to be there. Okay, ma'am. Just give me 2 minutes until I draw this. Then next person is going to be H2B. Okay, H2B also will come 2 in number. Fine, always works miracle. You'll always remember the concepts better. Okay, yes. So, H4 also told, okay, I will also bring my, this is what happened. This is what happened. That is, these people all brought one more person, and this is going to be your H2. Okay, this is going to be H2A, H2A, H2B, H3 and H4. This 4 people, okay, they brought 2 more people, okay. Class is lagging, huh? Just give me 1 minute, okay. I don't know what happened. It's the internet issue. Just give me 1 second. I'll just call them. Yes, 1 minute. Hello? There's a lag. Yeah. Can you please check? No idea. It's lagging. Yeah. Now it's fine, huh? There's a lag in between. Now they're telling it's fine, but then see, two seconds back there was a lag. Not sure if internet is okay. It was there in the starting of the class also. Yes, students, they'll check the internet, okay? Fine now? Okay, I think it's a slight 2 seconds issue. Yeah. Okay, fine. Now it's clear? Yeah, they will check, okay? They will check in case there's an internet issue. 2 seconds is fine. It will not disturb a lot. Okay, done? Okay, now students, remember, these people are coming 2 pairs, pair each, okay, they have come as pairs. So they are going to end up forming a histone octamer. So this was their plan. They told that, okay, we are going to come and we are going to help in the packaging. So these people have come as histone octamer because they are 8 in number. Anyone has a doubt in that? 1, 2, 3, 4, 5, 6, 7, 8. 8 in number, histone octamer. Okay, ma'am, understood. Now, this is how packaging is going to happen, they told. Then we asked them, why did you bring H1 protein? They told H1 protein is a linker protein to make sure that after wounding around the DNA, around this histone octamer, we have to make sure that nothing moves, right? Everything has to be, as the name suggests, linker protein. Linker means what? Something, okay, it should work fine, students. Okay, linker protein means what? It has to be, it will link and keep it in place. Okay, this is clear to all of you. So histone protein is there, which is positively charged, DNA is negatively charged, so winding will be easy, and these people have come as an octamer, so this is called as an histone octamer. Okay, histone octamer. Now look at this diagram for you to understand more depth. I have drawn the entire diagram. So look here, students, this is your entire DNA. Now this DNA, consider this one pink ball as a histone octamer, because I've drawn the structure here. Imagine the whole thing is identified as one pink ball, okay? So around this DNA is wound. Linker protein is going to be the orange color one. Can you all see the orange color one? Yes. So again, so remember, histone proteins plus the histone octamer, okay? The entire histone octamer plus DNA is going to be called a nucleosome. Histone octamer plus DNA is going to be called what, students? Nucleosome. Around 200 base pairs is going to be there, okay? Yes. Now you can see histone proteins coming together. Again, it will do condensation. Can you see? One histone, one histone, one histone. All these are octamers, okay? Many octamers together, again DNA. Condensation will happen. Again, many condensation will lead to forming chromatin fibers. Chromatin fibers ultimately form your chromosomes, okay? This structure should give you a better clarity of what you are learning. So this is how you're going to see. So all of you remember, no, in prophase you will see a circle, okay? In the microscope all of you would have seen, no? In prophase you will see something like this, okay? If the teacher asks you to identify the stages, you will say that, ma'am, something like this it looks, right? What is this? This is actually your condensation is happening. Those are your chromatin fibers. They condense again. During the metaphase, you will be able to see your chromosomes clearly. Anaphase, you will see them separating. Telophase, you will see the daughter cells happening. So all this is a continuous process. So now understand that when I'm looking in the microscope, I will notice it as a bead on a string structure. On what? A bead on a string structure. So DNA plus histone octamer together is going to form your nucleosomes. Okay, is that clear? Now H1 protein is going to be the linker protein, which is going to make sure that everything is in place, and your basic amino acids are going to be lysine and arginine. Now students, in higher organisms, we have to have NHC. What are they? Non-histone chromosomal proteins for higher coiling. Got it? This higher coiling, we will take additional set of proteins which are called as NHC. Clear? Packaging is clear to all of you? Yes or no? Yes, what are you asking me? Condensation means what, dear? Now, have you noticed condensed milk? That means what? Entire milk will be thickened. Okay, it's like a thickening process. No, entire thing will be thickened. Here, condensation means what? Everything is coming in place together, finally forming the chromosomes. Okay. Now, non-histone chromosomal proteins are additional set of proteins which we will be using for higher coiling. Okay, for higher coiling. I hope it is clear. This is how packaging of DNA happens. Okay, packaging of DNA happens. Okay, fine. Done. Moving forward. Now, most important thing, central dogma. With the central dogma, there's one more topic which is called as euchromatin and heterochromatin. So, students, this is your chromosome. Okay, this is the chromosome. In this chromosome, there will be some regions, okay, there will be some regions on the chromosome which are darkly stained, some are light, okay, some are light, and again some are dark. This is the better representation of, okay, what is going to happen, okay, what is going to happen. So look here, this portion where you see a most relaxed one. What is this, students? This is your euchromatin. So on the chromatin, you will have two types, euchromatin and heterochromatin. Euchromatin is going to be loosely packed, okay, loosely packed. If they are loosely packed, obviously they are going to be lightly stained. All of you tell me one characteristics. And they are going to be transcriptionally active. So, in the chromatin, okay, in the chromatin, there is going to be a region where it will be loosely packed. Since they are loosely packed, if I throw a die, imagine there are like 5 people, okay, 5 people in the class. I throw holy colors. It's not necessary that all the 5 people get equally stained. That means they will be loosely stained, lightly stained, right? Some of them will get, because there's a lot of space in the room, they will run that side, this side. But imagine they are closely sitting. I will throw holy color, everyone will get a color now. So here it is euchromatin. Then students, when we go further, you will notice that there is a region where it is more tightly packed. This region is going to be called your heterochromatin. Heterochromatin. Heterochromatin, please understand, here they are loosely packed, so here they are tightly packed. Tightly packed, darkly stained or densely stained. Okay, and the last is going to be transcriptionally inactive. Just learn one opposite of the other one. Clear? Transcriptionally inactive. Very good, very good. Good job. Perfect. Okay, this is done. Now let's go to central dogma. So, since you understood euchromatin, heterochromatin, we are going to learn the major concept of this chapter actually, that is called the central dogma. How does the entire molecular biology, you know, is so intense, so precise, right? Everything happens in a complete precision, like complete maximum, precisely everything happens. That is because of central dogma. Central dogma is like the ruling factor of how we are studying your genetics, okay? Principles of inheritance will start after I finish this class. That I'm not sure what time. Okay, but yeah, somewhere you can expect by 9. Okay, 9 p.m. Yes. So, central dogma. Students, if you have joined or you are attended 15-20 minutes late, it's okay. You can continue from central dogma because the topics are going to be individual topics. Okay, individual topics. So, yes, here central dogma, you should remember that DNA can become RNA, especially mRNA, and mRNA can become ultimately protein that is going to code and give us the characteristics that I spoke about in the starting of the class. Now remember that DNA can form its own, DNA can form its own replica with the help of the process called as replication. With the process called as replication. Now students, when DNA becomes mRNA, it's called as transcription, which you're going to study now. And when mRNA becomes protein, it is called as translation. Clear? Transcription and translation. Clear? Okay, fine. Yes. So, DNA, students, you please realize how big this chapter is. If you sit for reading this chapter also, you will take minimum of 3 hours to read and understand the chapter. So, how can I expect us to finish it like this? Right? You don't want a detailed lecture. I will just go through the surface. Surface, who told you that your NEET questions will be surface level? It will be from the depth, right? So, don't be like, oh my god, ma'am, 12 hours, 5 hours, 10 hours. If you can't stay, you can please leave. There are a lot of students who are dedicated and they will sit here till the end, okay? Yes. So, DNA with the help of, can form its own copies with the help of replication. DNA to mRNA is called transcription. mRNA to protein is called translation. Why? Because on the mRNA, genetic code is there and that reading will happen and that will lead to the formation of your entire thing. Okay? Got it? Yes, Baswa sir will take principles of inheritance. Yes, this is your central dogma. Any doubts in this? Francis and Crick. Okay, Francis Crick gave the ideology about central dogma. Now, let's start with Griffith's experiment. Okay, we will do the experiments very faster because the experiments are very easy. Okay, we will just do it faster. Okay, yeah, reverse transcription happens from RNA to mRNA. You want me to write that? Okay, yeah, mRNA to DNA, your reverse transcription happened with the help of reverse transcriptase. Okay, reverse transcription with the help of reverse transcriptase. Okay, yes. Now, moving forward, Griffith's experiment. So, what was Griffith working on? Griffith was working on Streptococcus pneumoniae. He was working on Streptococcus pneumoniae, the bacteria that causes pneumonia. Yes or no? Okay. So, Streptococcus pneumoniae, when he was working on, he noticed that the Streptococcus pneumoniae has 2 strains. What it has? 2 strains. It has 2 strains. He noticed that one strain, okay, one strain was called the smooth strain. He called one strain the smooth strain and he called the other strain the rough strain. Okay, rough strain. Now, why did he do this? It was because of how their structure looked and how they were as a whole. Okay, so smooth strain students had a mucopolysaccharide layer in the outside, because of which we called it the smooth strain. Okay, and the rough strain on the other end had a very irregular outer looks. So because of which we called it a rough strain. So they were assuming that is Griffith was assuming that the smooth strain had the presence of a polysaccharide or a mucopolysaccharide layer outside. Okay, there was a capsule, you can use the word capsule, okay, which was lining outside, which was absent in the rough strain. Any doubts in this? Okay, capsule was absent in the rough strain. Okay, rough strain. And one more thing that he told, okay, was that smooth strain was virulent. What did he tell? He told that the smooth strain was virulent. That means has the ability to cause a disease or can be dangerous. Okay, then rough strain, he told it is non-virulent. Okay, non-virulent. He even thought this could have happened because of the presence of a capsule layer or because of the presence of this polysaccharide layer, but no confirmation on that. Okay, none of that is discussed in NCERT. Okay, so we will not go into that. So this is what happened. He took the smooth strain, rough strain, capsule was there, one was virulent, one was non-virulent. He was working on mice. Okay, he took mice and he was planning to put these different strains on the mice. So let's see what exactly happened. Okay, look at this, you will understand. So here students, first what did he do? He took the rough strain and he injected into the mice. The mice lives because we already know our strain is non-virulent. Okay, then he took the smooth strain. He injected into the mice dies. This was also, we were aware of this. Why? Ma'am, because you already told that the smooth strain was virulent. Then he thought of one thing. Let me do one thing. Let me heat kill the smooth strain. So what did he do? He heat killed the smooth strain and he injected into the mice, and nothing surprising happened, but the mice lived as he expected. Okay, because in heat, most of the things will die. No? So he was like, okay, definitely the mice is going to survive. Then he did a combination of the one that led the mice to live. Two positive results he took and he combined it. What are the positive results that he took? He took the rough strain, which was non-virulent, okay, when we injected it individually, and we took heat killed strain, which was also individually not harmful. Now you tell me students, if you were Griffith, what would you expect? If you were Griffith, what would you expect out of the result? Tell me quickly in the chat box. Yes, yes, tell. What would you expect? You would expect that, ma'am, mice should live because individually this two led to the mice to live, so we expect that the mice lives. But for all of our shock, including Griffith's shock, the mice dies. The mice dies. He was shocked because he did not expect this result, and he told that something has got transformed from the heat killed strain to the R strain, which made R strain virulent. He told that something transformed, okay, something transformed, and he called that as the transforming principle. What did he call it? The transforming principle. Clear or not? Because he didn't know what it was. He was not ready for further, going forward with it. So he called it the transforming principle, telling that something from the S strain has transformed to the R strain, making R strain virulent. Okay, making R strain virulent. So because of this transforming principle, what happened? Many other scientists also came behind Griffith's experiment, telling that we can prove what exactly is happening. So for that students, we are going to take Avery-MacLeod and McCarty's experiment. So this is one setup, this is one setup, and this is one more setup. So Avery-MacLeod and McCarty's experiment is not very clearly given in NCERT, but you should understand what exactly happened. Okay, so they came behind telling that we will try to find what is this transforming principle. So basically from the combination of heat killed S strain plus R strain they took, and from this, okay, from this they wanted to test on the RNA, they wanted to test on the DNA, and they wanted to test on the protein to understand who exactly got transformed and who made R strain virulent. For that, they took 3 enzymes. Okay, so students, let's say this is your RNA in orange color. Okay, this is your protein in blue color, and this is your DNA in green color, because DNA is the green flag. Okay, so yeah, so look here, 3 setup they took. First, what did they do? Here they took everything, we will use orange color so you don't have any confusion. Here they took RNAs. Okay, for this no diagram, nothing is given in NCERT students, so we have to draw a diagram to understand. First they took RNAs. Now what is this enzyme? RNAs, DNAs, protease, they are going to degrade, right? They are going to degrade the RNA, the protein, the DNA. So they took RNAs. So in this scenario, what happens, students? Obviously RNA was not the transforming principle. Even though RNA was removed from the mice still ended up dying. Or let's say, in other words, transformation happened, right? Transformation happened. Okay, transformation happened. Why? Because DNA is still there, right? DNA is still there. Okay, next scenario, we are going to take blue color protein. What did they take? They took protease, which will digest the protein. They took the protease and they were like, okay, fine, it should be protein. But then here also the mice ended up dying. So here also transformation happened, because see, I am talking about transformation means something from the heat killed S strain went to the R strain and killing the mice. So here also transformation happened. That means it is not protein as well. Okay, any doubts in this? It's not protein as well. The last thing that was left was to check the DNAs. Okay, was to check the DNAs. DNAs is going to degrade the DNA. Pakka transformation here will not happen. Why? Because DNA is the one who was transforming, that we know. These people don't know. So here no transformation happened. That means here the mice lived because DNA got degraded by the DNAs. Okay, DNAs. Is this clear to all of you? Is this clear to all of you? So from this we can easily prove that DNA is the genetic material, or DNA is the transforming principle. So from this they told, hence proved that DNA is the transforming principle. Transforming principle. But students, unfortunately, like all our findings in the small age, right, we used to take pot, mix mud, and we used to tell that this is khichdi, this is, you know, the best continental dish. Like that, even there's also Avery-MacLeod and McCarthy's, whatever they did, definitely the experiment was good, but the clarity was not enough. Many zoologists, many botanists, many scientists did not approve of this and did not believe that DNA is the transforming principle, but because some of them still believe that protein could be the transforming principle, and this experiment did not give a clarity for them. So what did they do? They completely, okay, removed the whole thing. Okay, DNAs will degrade the DNA here. If the DNA is not there, only transformation, the mice is going to be alive. No. So here no transformation means what? From the heat killed S strain, nothing has come to the R strain, so the mice is alive. So here I can write that, okay, mice lives. So this is clear. Here all the mice is going to die. Here the mice will die. Here the mice will die because DNA is not there. So yeah, okay, that is the confusion here. So that's why I told you, like how Mithil has a doubt, many other zoologists and botanists also did not have a clarity on Avery-MacLeod and McCarthy's experiment. So they were like, no, let's move forward and let's find some other experiment, which is Hershey and Chase experiment. Okay, Hershey and Chase experiment. So students, the lag is happening. I think there is an internet issue in between. It's a microsecond lag. No, let's move forward and let's find some other and Chase experiment. Oh, there's a lag. There's a lag. Issue in between. Now also is there. Now is it better? I think it's just for like few seconds, no? Since I don't know what we can do about it, okay? It's like 4-5 seconds there's a lag. But is it disturbing too much? I will try to, like later in the video, I will try to remove those portions, okay? Because if it's going to disturb you. Okay, yes. So, Hershey and Chase experiment is very important because these people's experiment was completely approved by the entire thing. Okay, Baswa sir is like, okay, completely approved by everyone. That is, students, here they took a bacteriophage. Bacteriophage is a virus that infects a bacteria, right? Bacteriophage is a virus that infects bacteria. So they took bacteriophage and they grew it in two different medium. One medium which had S35 and one which had P32. Now what is the difference between S35 and P32? Ma'am, S35 is only there in protein. P32 is only there in DNA. Right? Okay? Yes. Only food you people think, okay? Food. Hershey and Chase means think about Hershey milkshake. Okay, so here they did so much hard work to give you this one experiment in your NCRT. So look here, bacteriophage radioactively S35 labeled protein capsule. So we understood one thing very specifically, that is S35, okay, is very specific only in protein. So if protein is the genetic material, we will be able to radioactively label with S35. If DNA is the genetic material, we have P32. Okay, we have P32 who is going to label. So this was a very good move because no confusion will happen. Okay, no confusion will happen. Correct? Now, moving forward, here what happens, students? The first ever process that happened was infection. So, bacteriophage already is ready. We took an E. coli and they were ready to infect. So, as all of you know, virus, whenever it is infecting something, it will release the genetic material inside the host, right? It is going to release the genetic material inside the host. So, same thing happened here. It released the genetic material inside the host, and after that, we did a process called as blending, where with the help of a mixer blender, we just removed the protein code. We do not want whatever is unnecessary, right? So, we will be like, go home. Okay, so we told like that, and we used a mixer blender and we removed the protein capsules, protein code. Now, after we did centrifugation, centrifugation is where we use centrifuge tube at a certain RPM, we rotate it at higher speed so that whatever is lightweight will come on the top, which is called the supernatant. What is it called? Okay, supernatant, which is going to be the lightweight thing, and whatever is heavier is going to be called the pellet, okay, which is going to be attached at the tip of the centrifuge tube, okay, tip of the centrifuge tube. So is this clear? Now, students, when they noticed the results after the centrifugation, they noticed that there was no radioactivity, that means no S35 detected in the cell. If you have to get transformed, you have to get transformed inside the cell. What will you do outside the cell? So they told protein is not the genetic material, but they noticed all the radioactivity was on the supernatant. That means the lightweight particle was the protein. All the radioactivity was in the supernatant. On the other side, when we checked P32, we noticed that radioactivity was detected in the cell. Obviously, because DNA is the genetic material and DNA entered into the cell, but protein cannot do that, right? Protein capsules will not do that. So here, radioactivity was detected in the cell and no radioactivity was detected in the supernatant. With this, they came to a conclusion that DNA is the transforming principle and DNA is the genetic material. Thumbs up in the chat box if it's clear. Thumbs up in the chat box if it's clear. Yes, infection, blending, centrifugation, you have to remember. Okay, the steps you have to remember, and you have to remember the result what we got. For P32, S35, they can ask you individually the result. How many of you are new to the class, yeah? Raise the hands. How many of you are first time attending my class? Raise your hands. Why your thumbs are not working to put thumbs up? People are putting chat, thumbs up. Okay, many of you are attending first time. So glad, thank you. Who was present in the pillet? Here you noticed S35. Okay, you noticed S35, that means protein coat was present, and in the pillet you noticed P32, that is phosphorus 32, which is only found in the DNA. So Hershey and Chase experiment was completely approved, and Hershey and Chase experiment proved that DNA is the genetic material. Okay, DNA is the genetic material. Okay, they proved it with the help of radioactive P32 and S35. Okay. Okay. I think when I'm turning there's a lagger, but how does it really work? Okay. Okay, voice is breaking sometimes, no? That is the problem. That is the problem. Have we reached the target? How many Madhumita in the chat box, yeah? It looks like one Madhumita full fan following has started. Okay? Okay, done. Fine. Yes. Those who haven't liked the video, quickly like the video. We have 250 target. Come on. Okay? Come on, 250 target. Oh, voice is breaking too much. Okay. What is wrong, yaar? Students, let me see if I can just restart the whole thing once again. Just be in the live, okay? Just give me 1 minute, okay? Just give me 1 minute. I've just asked them, okay? Everything is fine and clearer. That was voice is breaking, students. I just checked the live. Okay, voice is breaking in the between. Sahana, we have already started NCERT line by line. We finished also. Whichever chapters you people asked for, I have done it. Okay, every chapter is not possible, students. Whichever chapter possible, done. Okay, properties of genetic material. By the time the team sees the message, we will go. Okay, yes, come on. Now, how can I tell that, okay, DNA, you are the genetic material? For that, there should be a criteria that we are following, right? There should be something that we are following. For that, please understand that to be a genetic material, we have to go through a entire criteria, okay? Entire criteria. The first criteria is that the genetic material should be chemically and structurally stable. Chemically and structurally stable. Second thing, they should have mutation, okay, but the mutation should be at a slower rate. Okay, mutation should be at a slower rate. Third thing, They should be following the Mendelian characters. Yes or no? Now no lag, no? Yes. Slow mutation, stable. Okay. They should be able to replicate itself and expressed by Mendelian characters. Okay. Now students, let's take a competition between RNA and DNA. How many of you are in team RNA? How many of you are in team DNA? Right now, not the series wise I'm telling. Okay. Team RNA and DNA. See, let's talk about DNA and let's talk about RNA. So when we talk about chemically and structurally stable, whom do you think I would put a tick mark on? I will definitely put it on DNA and I will not put it on RNA because of two major reasons, students. DNA is more stable because of the absence, okay, absence of the O group in the second carbon, okay, in the second carbon. And the second thing is thymine, the presence of thymine instead of uracil. Presence of thymine instead of uracil. 5 seconds voice went, again it came back.
Now it is fine.
Yeah, no, it's fine.
That's because of internet.
Yeah, it's slow.
Working.
Okay, students, there's an internet issue today, okay? New Year, they are like, okay, don't take classes. Okay, so yeah. Yes, DNA and RNA, I'm going to put a tick in DNA and I'm not going to put a tick in RNA because of the presence of an extra O group in the RNA and thymine replaced by uracil. Okay, thymine replaced by... Okay, come on, tell me. So students, properties of genetic material means what? Now if you're standing for election or you are planning to vote for someone, you should be an Indian citizen, you should be above the age of 18. Simple things, no? Same thing. Like that, to be the genetic material, there are some properties or criteria that the genetic material has to go through. Students, correctly when he went, it started lagging. What? Now again I have to call him back. Just for 2-3 minutes, there is an internet issue here. I think office full internet issue is there. Today is a lagging day, exactly. But the lagging is sad, when he went, it came. Students, correctly when he went, it started lagging. Okay? Yes. Now, be calm, it will work. Okay, be calm, it will happen. Don't worry. Now, replicate itself. Both of them are going to replicate itself. Okay, both of them are going to replicate itself. So, we will put tick mark for both of them. Both of them are going to express by Mendelian characters. So, here as you can see, these two places, it's an into mark. Okay, into mark, because of which, okay, it is a big problem. Okay, it's a big problem.
No one's saying
It's lagging. I can't restart.
it is an auto stop. It is now. You can stop streaming and you can start.
Will it fix it?
Will get a problem. Sometimes lag, sometimes not lag. So it will.
Hello students, can you hear me? We live it. Yeah.
Hello students, can you hear me?
Yes. Yes. I'm back. Okay. Students, I think it's mostly a issue with YouTube. Okay, so don't worry. Few seconds only, 5 to 6 seconds, that's it. The place where the voice is breaking, I'll repeat the topic. Okay? Yeah? Because it's a big chapter, no? We cannot stop it in the middle, nor we can reschedule the class. So it is important for us to continue. Okay, ready? Next topic. Yes, next topic. Yes, so put some fire emoji so that our energy comes back. Okay, those who ran away will come back. Yes, so we were going to do replications. Okay, so we have completed up to the property. Okay, these were the properties for a genetic material. That is, first is chemically and structurally stable, and what makes DNA more stable is the absence of that O in the second position, and also presence of thymine. Okay, presence of thymine instead of uracil. These are the two reasons mentioned in your NCERT. Mutation should be at a slower rate. As I told you, you want to sleep as a human, wake up as a penguin? No, ma'am, I want to sleep as a human, wake up as a human. So the mutation should be at a slower rate, which DNA approves, but RNA does mutation but at a faster rate. Okay, faster rate. Now replicates itself, that is, students, replication happens, both of them replicates itself, express in the Mendelian characters, both of them express. Okay, fine. Yeah. Okay, fine. Okay, now before we go to RNA world, let's start with replication. Ready, all of you? Yes, very nice. Okay, fine. Okay, now, yes, Rafa, we have a board channel which is called as Vedantu, KCET and boards. So you can go follow me there. I teach for boards there. Okay, now students, replication, very important concept. Very important concept. Now, replication means what? Making its own replica, making the own copies. Okay, making the own copies, and it is one of the properties of the genetic material that if you can replicate yourself, that means you're good to go, right? If you're able to replicate yourself, you're good to go. So now students, replication, when we talk about, understand that your DNA replicates in a semi-conservative manner. Okay, semi-conservative manner. Now, what is semi-conservative manner? I've always given this example to all of you that usually what happens, parents are there. This is the parent DNA. Okay, and parent DNA, let's separate them and they will be used as a template. Okay, they will be used as a template to form the daughter DNAs. This is the daughter DNA. This is the daughter DNA. Now, most of the time what happens, students? Most of the time what happens? These people, okay, these people will go back to each other, right? These people will go back to each other, but the entire enzyme machinery for replication we will learn. Okay, we will learn. Clear? Now students, remember, this is the parent DNA. We are using the parent DNA as a template, and after forming that template means what? I am giving you a Xerox copy. Let's say I am giving Ninja one Xerox copy. He is going to a Xerox person and he's copying that and bringing it back. So the original copy I gave is going to act as a template, and the copy that he got is going to be the daughter DNA. So this is going to be the daughter DNA. This is going to be the template. Okay, this is going to be the template. Now understand one thing. Okay, understand one thing, that these two, usually the parent DNA will go back to each other, which is called the conservative one. But in scenarios like our DNA, what happens? Parent and the daughter DNAs are going to be together. Okay, parent and the newly formed template is going to be together, which ends up forming something called as hybrid. Which ends up forming something called as the hybrid DNA. Clear or not? Hybrid. Okay. Now, to prove that, we have Meselson and Stahl experiment who proved that, okay, Meselson and Stahl experiment who proved that, yes, we can actually find how our DNA is replicating. It is in a semi-conservative manner, and the experiment they set up, was, this was the experiment. Okay, this was the experiment. So I'll explain you the experiment. So first, what did they take, students? They took N15. Okay, they took N15 DNA, which is a higher. Okay, this one has the denser one, no, N15 NH4Cl. Okay, so I will go back here and write what are the things that they took. Just give me 1 minute. Okay, fine. Okay, here it is showing. Okay, yes. So students, Meselson and Stahl, what did he do? They took N15H4Cl. All these notes will be available on Biology by Gopika and Vedant to NEET English Telegram. So go and join the Telegram. Okay. Yes. NH4Cl. They took first culture medium, and students, they took E. coli to grow because E. coli replicates in 18 minutes. Okay. 18 minutes to 20 minutes it replicates. So we need something that will replicate faster. So according to your NCERT, it is given 20 minutes. Duplication, replication separately it is given. So here it is 20 minutes. So they took E. coli. First they cultivated in N15 medium. First they cultivated in what medium? N15 medium. Okay ma'am, understood. After that they removed it from the N15 medium and they cultivated this. Okay, they cultivated this in N14 medium. N14H4Cl. So they are not radioactive. They are not radioactive. They are just the denser one and the other one. Okay, they are just the denser one and the other one. After that, they removed it from the N15 medium and they cultivated this. Okay, yes. So N14, NH414, Cl medium. Now you tell me what would have happened. Notice that this is going to act like the parent. Okay, this is going to act like the parent strand and we are going to get two strands like this, right? Both are going to be N15, N15. Agreed all of you? Then after that, when I take it, okay, when I take it and I do it on N14, what am I going to get? Okay, what am I going to get? Students, don't tell chumma lag. Okay, chumma don't tell lag. I feel bad when their lag is happening. Okay, but these things happen in classes. Okay, remember, technology is not in our hand all the time. This is going to lead to the formation of N15, N14. Okay, N15, N14. Correct? Yes or no? Yes? Yes, N15, N14. Now students, remember this one thing. 20 minutes, N15, N15, N14, N15. Correct? This led to the formation of a hybrid. Now they did centrifugation. Okay, they did centrifugation to get this result so that they get the band formation. Okay, and for the centrifugation they used cesium chloride. They used what? Cesium chloride to do this density gradient. Clear? Now keeping this in mind, you see this experiment, you will understand. So first, as I told you, they cultivated E. coli in N15. Why did we take E. coli? Because their replication time is 20 minutes. Okay, 20 minutes. Okay. Now N15, students, I already told you I cannot do anything about the lagging. Okay, I cannot do anything about the lagging now. It's just for 2-3 seconds. Okay, so please bear with it. In 2-3 seconds, the lag will be fine. Okay, there's a slight internet issue. This can happen. It's a big office. Everyone will be using internet. It can happen. Okay, slight internet issue is there. I cannot do anything. I already stopped streamed and started again. More than this, I can't do anything. Okay. Yes, again, otherwise every 5 minutes I have to stop and start. So that I don't want to do. Just 5 seconds, just wait. Okay, now students, N15, higher, denser one. Okay, after that they took it. 20 minutes replication happened, led to the generation 1. Okay, that led to the formation of 2 hybrid strands. Okay, then after 40 minutes, as you can see, 4 strands are formed in generation 2. Okay, 4 strands are formed in generation 2. You're getting 2 lighter strands and you're getting 2 hybrid strands. Now, what you need to understand is they will give you numericals based on this. They will give you numerical based on this. Okay, how the numericals are going to be? Let me just erase this for all of you and I will show you exactly what happens. Okay, now students, first generation what happened? First generation what happened? Ma'am, both the strands that we got were the denser strand, that means N15, 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 N15. This is generation 1. After 20 minutes, what did you get? Ma'am, I got 2 hybrid strands. I got 2 hybrid strands. Correct? Yes or no? This will help you in the problem solving. Okay, so listen to me carefully. I'll tell you how to solve the problems. Now, percentage wise, you people tell me. That is, in first 20 minutes, I am going to get 50% hybrid. What am I going to get? 50%. Okay, I got 100% hybrid. Okay, 100% hybrid? Yes or no? From first generation to second generation, I got 100% hybrid. From here, when I further move forward, okay, in another 20 minutes, okay, I am going to get, just separate this, If you don't know how to do, just separate the colors. See, 2 strands of yellow was there, 2 strands of blue is there. So I separated them as 4 strands. Now to all of them, you add the blue strand. To all of them, add the blue strand because you are keeping it more in N14. Now you tell me what you got. Now look at the colors and tell me what you got. The percentage is going to be 50% hybrid. This is the numerical that they'll ask you. Okay, 50% hybrid and 50% light. Correct? Correct? Yes, ma'am, correct. Generation you got. After 20 minutes. After 20 minutes. How am I going to get? Again, separate all the strands, students. Separate, see. 1, 2, 3, 4, 5, 6. 6 blue strands are there. Now that many yellow strands also I have to draw. Okay. 1. Okay, these 2 I separated 2. Okay. This one blue. Yes, ma'am. These 2. Okay, these 2 and this. Okay, 1 second. I'll draw it more concise. Otherwise, you might get confused. Okay, I'm just separating the strand. Nothing else I'm doing. So, these 2 strands I'm separating. 1, 2. Okay. This 2 strands I'm separating. 1 blue is there. So, I'm drawing 1 blue. This 2 strands I'm separating. 1, 2. Okay. This 2 strands I'm separating. 1 blue. Now, yellow strands, how many are there? Ma'am, there was 1 yellow strand here. Okay, I'll draw it down. Okay, there was 1 yellow strand here and there was 1 yellow strand at the bottom. Now, to all of this, what you are going to do? Okay, to all of this, you add the blue. All this, you add the blue because it's N14. Okay, done, 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 done. You can see as the generation is passing by, hybrid is becoming less and light strands are becoming more. So, students, you look here 1, 2, 3, 4, 5, 6. Out of 8, 6 of them are light. So, that means 75% light and 25% hybrid. Clear? Now, if I go further also, I'm not going to draw that. Further 20 minutes later, I will get somewhere around 87.5% light and I will get around 12.5% hybrid. Understood? This is how you will do a numerical. In case you don't know anything, you don't know anything about this, okay, please separate the strands, draw it in a piece of paper and just do the calculation. Clear? Yes. So, this is how you are going to get. So, this is the entire Meselson and Stahl experiment, how they proved that DNA is going to be replicating in a semi-conservative manner. They took an E. coli, they cultivated it in N15 medium, then in N14 medium and they did the centrifugation with the help of cesium chloride. Okay? Yes. Therefore, DNA replicates semi-conservatively. That means 1 parent strand, 1 daughter strand, 1 template, 1 daughter strand. That is very important for you to remember. Any doubts in this? Any doubts in the experiment also? Now, students, just by learning replication, it's not enough. No, we need to know the machinery behind the replication. That means, like some person asked me, ma'am, what are the enzymes involved in replication? That is also important. Yes, for enzymes involved in replication, let's draw a diagram to understand what is it. So, as all of you know, there is going to be replication fork. Okay, replication fork mentioned in your NCERT, no? Now, you are going to see all the enzymes, okay, all the enzymes involved in this whole process. Next 5 minutes, don't talk to each other, but listen to me. Very important concept. Enzymes are very, very important. Now, look here, students. Okay, excellento. Wow, 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 wow. Okay, so first problem that I have is that these 2 people are like this. First problem that I face is these strands are wound around each other. First problem that I have is I need to make sure these people are unwinding. I need to make sure that these people are unwinding. So, for that, I am going to take the help. Okay, I am going to take the help of helicase I'm going to take the help of helicase, which is going to do the job of unwinding. Fine, all of you? Helicase told that I am an unwinding enzyme. I will actually do the unzipping and the unwinding. Okay. So, unwinding is done by none other than helicase. Helicase enzyme is going to do. So, the first enzyme I am introducing all of you is your helicase enzyme. Okay, fine. So, he is like, okay, these people are bound, no problem. I will keep unwinding them. That is my job. Okay. Then we told, okay, we want to start replication because helicase is anyway unwinding. So, they told you have to find a spot which is called the ORI. What is it called? ORI region. I'm just drawing it like this, students, but the positioning is not right. Okay, which is called the ORI, origin of replication. Origin of replication. That means what? This is the place where the replication is going to start. Origin of replication. Got it? So they were like, okay, you can start with the replication. I was like, okay, fine. Then I noticed that since these people are always together, whose students' DNA is always together, they have a tendency of coming back together. So, to avoid this coming back together, I told that, okay, I am going to use an enzyme which is called SSBP, okay, which is going to be called SSBP, okay, I am drawing it like this. I am using an enzyme called SSBP, which is called your single stranded, okay. Notes link, yes, sure, I'll keep that. SS binding protein. Single stranded binding protein. So, these two strands do not come back together because they were together so long, they will obviously try to come back together. So, SSBP will do the job of making sure these two people don't come back together. Okay, that problem was solved. Then when I went further, students, I realized that these two strands have a tendency of supercoiling. Because they are too long, 2.2 meter long DNA, they have a tendency of supercoiling. So, to avoid supercoiling, I told, okay, let me bring one more enzyme, which is called the SSBP enzyme. Okay, what did I bring? Topoisomerase. Okay, SSBP is done. Topoisomerase. I bought the topoisomerase. I'm just drawing it right now in random places, okay, because I don't want to bring it this way, okay. Yes, Josh, Kim, I will keep the notes link in the description, dear. Let me finish writing the notes, okay. So, I will keep the topoisomerase, okay, to prevent what, students? Supercoiling. Is this clear to all of you? Can I get some thumbs up? Why is the energy low? Why is the energy low? I am the one teaching. You people should have high energy. My God. Yes. I'm teaching the full chapter, dear. There's nothing called revision. Okay. Fine. Clear? Okay. Now, so SSBP is going to make sure that the two strands do not come back together. Right. Both the strands are separated. I'm writing here. Okay. Separated. That is the job. Okay, fine. Now, this much job is done. One is going to avoid supercoiling. One is going to do the unwinding. One is going to make sure that both the strands are not going to come back together. We also found the ORI side, that is the origin of replication side. Okay, done, fine, problem sorted. Now I need someone, because I'm doing replication, I need to form my new replicas, right? I need to form the dotted DNA. So for that, I need to make sure that there is one person who is going to form the new template or going to form the new dotted DNA. For that, I finally bought a hero to the set, that is going to be your DNA polymerase. What is correct? Yes, polarity is correct. Check the polarity and tell me, is the polarity correct or is it wrong? Okay. Yes, fine. Okay, very good. Now, DNA polymerase is there, students. DNA polymerase is like, okay, I will form the new template. You people don't worry. I'm going to be the one who is going to form the new template. Okay, I will form the new strand. Okay, new strand formation will be done by your DNA polymerase. But DNA polymerase is blind. DNA polymerase does not know where to start, when to start, what to do. So, to help DNA polymerase, we bought one more person, which is a primer or a primase. Okay, so primer or a primase will be added initially. Okay, it will be added initially so that DNA polymerase can work in peace. DNA polymerase is like, okay, fine, primer is there, I will continue my work. But DNA polymerase has two conditions. DNA polymerase has two conditions. What is the condition DNA polymerase has? That I will work, okay, I will work on a strand who has, that means in your NCERT it is written DNA polymerase will do polymerization from 5' to 3'. That means it will work on a strand, okay, it will work on a strand which is from 3' to 5', or shall I write NCERT only? Will do polymerization okay, polymerization from 5' to 3'. That means, students, please don't get confused. Polymerization means it will form a new template. It will form a new strand from 5' to 3'. That means it will do, okay, it will work on a strand which is from 3' to 5', right? I'm telling that I will form a strand which is from 5' to 3'. That means I need to work on a strand which is from 3' to 5'. I hope this is clear to all of you. Then the second condition this fellow has is that the strand should be a single strand. Okay, the strand should be a single strand. We were like, okay, fine, both of your agreement is approved. You will go forward with the work. Then, okay, RNA polymerase started the work and it started forming the new template strand. And finally, the template strand got formed, students, and it will continue. It will form like this. Oh, okay, 1 second. Yeah, it formed like this. Okay, done. So the newly formed strand is going to be from what? From 5' to 3'. Understood? 5' to 3'. Since here we did not face any problem. This strand is going to be called the continuous strand. Okay, continuous strand or the leading strand. Continuous or the leading strand. Please remember these two names. Okay, now one problem is there. What is the condition DNA polymerase has? That I will work on a strand, or I will form a strand which is from 5' to 3'. That means I will work on a strand which is 3' to 5'. Now we are like, oh my god, we have only one strand who is 3' to 5'. Now what to do? Then primer told, anyway, DNA polymerase does not know where to start. I will do one thing. I will start doing it in the reverse pattern. So primer kept adding from the backside. Okay, primer kept adding itself from the backside. So this would lead the DNA polymerase to get confused. Okay, primer is doing the job, that means it should be right. So DNA polymerase will start working in the reverse direction. It will start working in the reverse direction. So this strand will be what? 3' and 5'. Understood? Clear? Clear or not? Thumbs up in the chat if you understood. Okay, here what is happening, students? The primer added itself in the reverse pattern so that DNA polymerase does not make any problem. Okay, so this strand is also called as discontinuous, lagging, and these fragments are called, okay, these fragments are called as Okazaki fragments. Okazaki fragments after the person who found it. Okay, Okazaki fragments. Now students, after all the work got over, DNA polymerase is like, I don't want primer, yeah, remove all the primers. Okay, remove all the primers. So we were like, okay, fine, we should listen to the DNA polymerase, okay, because it's the hero. So we removed the primer. Okay, we slowly started removing the primer, and in the places where we removed the primer, your DNA polymerase will add the new nucleotides, but still it's not continuous. So there is going to be a space, right? There is going to be a small a person who is going to do the gluing. So for that, we bought another person to do the gluing, okay, another person to do the gluing, and that is called as ligase. Clear to all of you? All the enzymes are clear to do the gluing. All the enzymes are clear? Any doubts? Any doubts? Tell me in the chat box. No doubts, right? Perfectly clear. These are all the enzymes. Okay, this one diagram is enough for you to learn. Okay, let's do a quick recap of it. So what exactly is happening? Replication should happen. If replication has to happen, we need to do the unzipping of the entire DNA so that the new template can be formed. As I told you, Ninja is taking my original copy and going. So that original copy is this white color strand. Now I need to make another template of it, right? I need to make a daughter DNA. For that, we need some enzymes who are going to work. So here the first enzyme that is going to work is going to be helicase enzyme, which is going to do the unwinding. Okay, which is going to do the unwinding of your DNA. Okay, so that's why this is called as an unwinding enzyme. So helicase will do the unwinding. Yes ma'am, helicase will do the unwinding. Now the strands will be separated, and this portion where the mouth is opening like this, no, this is called as the replication fork. This is called as the replication fork. Okay, replication fork, because of the structure and the shape. Now students, remember that to start the entire process, we need topoisomerase, which is going to prevent the supercoiling. We need SSBP, which is going to make sure that the two strands do not come back together, because they have a tendency to come back together. SSBP full form, they can ask you, single stranded binding protein. Okay, then as we go further, the main person who is going to be involved in the entire replication is going to be DNA polymerase. DNA polymerase has few conditions that it will only do polymerization, or it will only form a strand which is from 5' to 3'. So we need to give him to work in 3' to 5' direction. So for that, we are also going to take the help of an ORI site, which is an origin of replication site, where replication starts. Then students, we are going to take the help of a primer or a primase, who is going to do the job of guiding our DNA polymerase to continue with the formation of the new strand. So primer will be added and the DNA polymerase will continue, and one strand we are going to get 5' to 3' is going to be a continuous strand. We can also call it as a leading strand. Now the second strand, what is the problem is, we do not have 3' to 5'. So primer will be added in the reverse pattern and the DNA polymerase will work, and this will lead to the formation of discontinuous lagging strand, and those fragments are called as Okazaki fragments. Now to keep all the Okazaki fragments together, we need to take the help of ligase. Please do understand, ligase will not form the new nucleotides. Ligase job is not to fill the gap, like only cementing will be done. Bricks have to be kept by DNA polymerase only. So DNA polymerase will add the nucleotides wherever the primer is present, and the joining will be done by ligase. So ligase will become a gluing enzyme. Is this clear? 6 enzymes you studied: helicase, topoisomerase, SSBP, DNA polymerase, primer, as well as ligase. Clear? 6 enzymes. This is your entire machinery of enzymes. So this is a diagram given in your NCERT, and you can see this diagram to understand this. See students, 5' to 3', you are getting a continuous strand. That means the direction you can see 5' to 3'. Okay, this newly formed strand is from 5' to 3', which is a continuous strand. Same thing goes here, 5' to 3', you got a new strand. Okay, continuous strand. Then numbering is very important. 3' to 5', you got a discontinuous strand. The formed strand is going to be 5' to 3'. Okay, arrow mark you can see, one is this way, one is this way. And continuous synthesis, newly synthesized strand, discontinuous strand, and template strand. Now look here, I will just write just one word for all the enzyme. Is it clear? Okay, gluing. Just one word I am writing, so it will help for your revision. DNA polymerase to help in formation of new strand. Basically polymerization. Okay, New strand formation, it will help. Just 1 minute, students. Yes. Okay. DNA polymerase. Like you said, I don't know. Okay, and the last one is your primase. Okay, it will add the primer. Okay, and lead DNA polymerase. Done? Any doubts? Yes, perfect. Okay, now look here, students. Very similar experiment involving use of radioactive thymidine. Okay, radioactive thymidine was done to detect the distribution of newly synthesized DNA in the chromosomes, was performed by Faba beans, okay, Faba beans, okay, Faba beans by Taylor and his colleague in 1958. The experiment proved that DNA in chromosomes also replicates semi-conservatively. So to prove that if DNA is going to be replicating in semi-conservative manner in the chromosomes, Taylor and his colleagues did in 1958 work on Faba beans, and they also proved this. So just remember the dates. Dates can be asked, and once this question also has come as a PYQ asking what did Taylor and colleagues work on. Okay, they had given the Faba beans. Okay, yes, ready for transcription? Okay, so we'll drink some little water. Okay, no break as of now. After some more time, I'll give you a break. Okay. Transcription is easy. It will take us only 10 minutes. Okay, 10 minutes. Okay, chalo. Let's do transcription. Ready? I need some energy in the chat box. Okay, otherwise I'll get tired. No, you people are my energy. Very good, very good. Meghna, very good. Throughout, same energy should be there. Okay, very nice. Rohit, no doubts. Okay. Okay, done. Chalo, let's start. Now, what is transcription? Ma'am, just now we studied central dogma. We know that DNA to RNA is going to be called as transcription. Now we studied how DNA can form its own replica by replication, and it is in a semi-conservative manner proved by Meselson and Stahl. And we also learned the machineries in a beautiful way, like a story, right? Now we have to learn about who? We have to learn about how transcription is going to happen. What is the process involved in transcription? Very simple process, okay? Don't even complicate. I'll teach you in an easier way possible. Now look here. Transcription. So students, this is the Telegram channel. As you can see, in case those who do not know any spelling, biology spelling, Gopika spelling, this is the spelling. Okay, transcription, as I told you, it is part of your central dogma, DNA to RNA. Right, DNA to RNA. Exactly what? mRNA. Exactly what? mRNA. Correct? This process is called as transcription. This process is called as transcription. Now, what exactly happens in transcription? See, I know few things that I need to form the mRNA. This much I know. Yes, ma'am, I know. That this is a double-stranded DNA. Okay, from this double strand, I need to form a single-stranded mRNA. From this double-stranded DNA, I need to form a single-stranded mRNA. Always remember, there can be a question to all of you. There can be a question to all of you. Why are we not using both the strands of DNA to form mRNA? Who knows the answer? Raise your hands in the chat box. I will give you the mic to answer. Okay? Yes. Tell me in the chat box. Yes. So, why both the strands of mRNA will not form, why both the strands of DNA will not form mRNA? Simple thing, students. both the strands, imagine both the strands are going to form mRNA. They are definitely going to bind with each other because they are going to be complementary. We know that DNA is complementary, right? We know DNA is complementary. One DNA, another DNA wound around each other. This is also going to form, this is also going to form. So yes, exactly. According to our central dogma, we cannot form more than one protein. So DNA, mRNA, protein, coding for one protein. We cannot change the central dogma. So because of these two reasons, that if both the strands are going to form mRNA, both formed mRNA are going to be complementary to each other. What is the point? And also we cannot form multiple proteins, right? So that because of what? Because of which we are going to follow the one-step process where one DNA, one mRNA. Okay, one strand only is going to become the template strand, the other one is going to become useless coding strand. Okay, yes, very good. Okay, so this is clear to all of you. Now remember students, in transcription mainly two things you have to remember: 3 steps and 3 units. Okay, many students get confused with this. Unit of transcription, okay, first is going to be a promoter region, 3 regions also you can tell. Okay, promoter, structural gene and terminator. Okay, terminator. So these are the 3 regions, or together I can call this the transcriptional unit. Okay, together I can call this what? The transcriptional unit. Today I really want to see which are the students who are going to stay till the end of this, because it is not easy to learn genetics together. Okay, even teach genetics together. But the students who are going to stay, let me tell you students, 2025 is going to be your year. No one can stop you from scoring 360 in biology, physics and chemistry also. Please put in maximum hard work to at least get 130 to 150. Aim for 150 only, so that you can land at least in 140, 130. Okay. Yes. Don't lose your marks in physics and chemistry as well. Try to practice as much as possible. Now, structural gene is clear. This is the transcriptional unit. Now, 3 processes happen. Remember, 3 processes happen. In the promoter region, there is going to be initiation. In the promoter region, there is going to be initiation. Yes, ma'am, initiation is going to be there. Then in the structural gene region is going to be the region where elongation happens. Okay, this is the process I am talking about. And in the terminator region, obviously termination will happen. Okay, termination will happen. Got it? Yes or no? Perfect. Students, and also biotechnology unit is going to happen on Monday. So go and like the video as well. All blessings for all of you. Okay. Yes. So this is the 3 process and this is the 3 unit. Okay. This is the 3 units. Is this clear to all of you? Now, promoter, from the term itself you understand that ma'am, they are going to promote, they are going to initiate. So during this entire initiation, we are going to use one word which is called the sigma factor. We are going to use one word which is called the sigma factor. This is all about transcription. Clear or not? Any doubts in this? Please tell me in the chat box. They will ask you the transcriptional unit 2024 question it is. Okay. If you all remember, it is a 2024 PYQ. What are the transcription unit? They had given 3-4 questions and they had asked the question. Okay, so remember this. And they can also give you initiation, sigma factor, rho factor, choose which is correct. That such kind of questions also can come. Okay, such kind of questions also can come. So I expect any question, simple, difficult, expect everything when you're studying. Don't just have a narrow mindset that, oh, I will only study this kind of questions. Okay, sometimes the easiest question can be the most trickiest. Okay, done? Okay, yes. Now, moving forward, I will teach you some images and some important concepts of the image. Now, I told you one thing, that I cannot take both the DNA. So, which strand am I choosing to take? That is, students, I am going to take the template strand which is from 3' to 5'. Okay, or let's say 5' to 3' depending on the arrow mark. So, 5' to 3' direction 1, I am going to use it as the template strand. Okay, I am going to use it as the template strand. Ma'am, what about the other strand? The other strand, I'm going to use it as the coding strand. I'm going to use it as a coding strand. Now, what is coding? That term, it's very contradictory to the name, because coding means you should have coded, but you are not coding. So, think like that, okay? Coding strand. Template strand, remember, there is going to be a promoter region and a terminator region on that. Now, this promoter region and terminator region will have an upstream, okay? That means, students, the coding strand, okay? Coding strand upstream. This is coding strand. So, imagine like this. This is coding strand. Coding strand upstream, what will be there? Yes. Okay. Okay. Look here. This is the coding strand. This is the upstream of the coding strand. On the upstream, you have a promoter. Okay. Now, look here. Okay. Here, this is the coding strand. The downstream of the coding strand. Okay. Downstream means again here, upstream, downstream, upstream, downstream. Downstream, you will have the terminator region. So, remember that upstream and downstream is in terms of the coding strand, okay. So, this will be present in the downstream, okay, downstream. Is this clear? Okay, is this clear? Coding strands, downstream you will have terminator, and on the upstream you will have promoter region. A small portion, entire DNA will not form mRNA, students. Only a small portion on the DNA will form mRNA. That region is called the structural gene region, okay. The region or the gene that has the ability to code for an mRNA, okay, to become an mRNA, to code and form an mRNA is called the structural gene region. So, promoter region initiation is going to happen with the help of sigma factor. Structural gene region is the region where elongation will happen and formation of the mRNA. mRNA has to be transported to the cytoplasm for translation. In the nucleus, this will happen. Then the transportation will happen to the cytoplasm. Then in the termination region, what is going to happen? Termination region, you will have rho factor, which is going to terminate the entire process. Now, for all of this to happen, you will have one main fellow, which is called the RNA polymerase. We had DNA polymerase to form a new DNA strand. To form an mRNA, I will obviously call a RNA polymerase, okay. Now, see, upstream and downstream depending on the coding strand. Now, this is the coding strand. So, look like this. Coding strand, upstream of the coding strand, you have a promoter region, and downstream of the coding strand, you have a terminator region. That's all. That's all is upstream and downstream. Upstream, downstream. In water also we tell, no, upstream, downstream. The top region and the lower region, that's all. But it is in terms of coding strand. That's all I'm trying to make you understand. Okay. Yes. Now clear? Okay, clear? My glass. Okay, fine. No problem in this. Okay, replication of DNA takes place where, students? Replication of DNA takes place at the S phase, okay, in eukaryotes. That I forgot to tell by mistake. Students, 1 second, I'll just get my glass. I cannot see anything otherwise, okay. Just give me a second, okay, just a microsecond. By then, you please copy this down, okay, copy this down, I'm coming. Without this, I cannot see it. Yes, I'm back. And I again touched. Okay, ready? After this, I'll give you a break. Okay, please sit patiently. Don't. Yes. So, upstream, downstream is clear. So, students, just look this also. Upstream, which region is going to be there? To the phi dash end of the coding strand. Okay, phi dash end of your coding strand is going to be there. And here it is going to be 3' end of it. Okay, that's all is what I wanted to tell you people. Lot of questions or nothing has come, but I just wanted to make you understand the NCERT line. Okay, now moving forward, most important diagram, or what is transcription exactly in this diagram you will understand. See, as I told you, RNA polymerase is going to be the one that is going to form the mRNA. Yes ma'am, who doesn't know this? Okay, who doesn't know this? We know this. Yes, definitely Anjali, don't worry, I'll give a break. Okay, now promoter, okay, promoter is going to be the one that is going to, where initiation is going to happen. So RNA polymerase plus the sigma factor will form a combination, okay, because RNA polymerase comes and then we will be like, okay, he wants to start initiation. But please do understand, RNA polymerase only has the ability to form the mRNA. It does not have any other ability of initiating, terminating, nothing. That is why we are giving factors from outside. Imagine you are only going to write NEET exam. You do not know anything else. Okay, that means what? You are only going to, you have a horse view, you are only focused on that. Now, for that, we will give you outside. Okay, so same thing here also students, RNA polymerase is not capable of initiating, terminating by itself. So, we are giving two factors, sigma factor at the promoter region so that the initiation can happen. Then, second C, initiation. Second step, elongation. What is going to happen? The RNA polymerase will reach the structural gene region and you can see the newly formed mRNA which is coming out, because we are going to transport this to the cytoplasm for translation. Okay, mRNA has come out. Yes ma'am. Now RNA polymerase does not know what to do. It will keep moving until it meets the rho factor. Now rho factor will combine with RNA polymerase and here they form a complex and they will finally terminate. Okay, they will finally terminate. Is this clear to all of you? What is, transcription? Very simple it is. Very simple. Nothing is there. Sigma factor, rho factor, structural gene, promoter, terminator. 3 units. What is the transcriptional unit? They will ask you. Promoter, structural gene and your terminator, and they will ask you initiation, what happened? Tell that RNA polymerase forms combines with sigma factor and they move forward up to the structural gene. RNA polymerase will form the new mRNA and finally go for termination. That's it. Okay, this is your transcription. Those who found transcription complicated should feel like, oh my god, this was so easy. Now students, please understand, RNA polymerase is little specific. Okay, they're not going to work with every type of formation. So here we have some specific thing which was asked as a PYQ. Okay, I do not know which year, I think 2019 if I'm not wrong. PYQ, this question has come. So how do you remember this? You're going to remember like this, RMT. Okay, RMT, you can easily tell, no, RMT. So R is the first one, M is the second one and T is the third one. Okay, imagine RMT is 3 kids. R is the first one, M is the second one and T is the third one. So what do I mean by this? Remember that polymerase 1 will code for rRNA. Okay, will help in the formation of rRNA. 2 will code for mRNA. 3 will form the tRNA. So understood? So look here also, RNA polymerase 1, okay, in all eukaryotes will form rRNA. Okay, so first child is going to be R. Second one, RNA polymerase 2 in eukaryotes will form the mRNA. That means transcribe or help in transcription for the mRNA. snRNA, this you remember, okay? snRNA and miRNA you remember. This is not required, it's not mentioned in your NCERT. So snRNA, miRNA. Okay? Okay, nice. Now here, RNA polymerase 3 is going to transcribe for tRNA, small rRNA and some snRNAs and miRNAs. This question has come, so please remember RMT 123. RMT 123, you can at least identify by looking at this. Okay, you can identify by looking at the first words. Okay, RMT 123, RMT 123. Done. This is your one PYQ question. If it comes that day, you can thank me. Okay, now cistrons. Okay, 1 minute. My throat. Okay, yes, ready? Quiz is definitely good. Okay, I have checked all the questions of the quiz. It is definitely good. Okay, yes, it is easy if you learn it. If you have not learned it, it's not easy. Yes, Okay, now cistron. What is cistron? Students, it's a small segment of the DNA that is going to form the polypeptide chain. Okay, a small segment that is going to form the polypeptide. That means protein. Simple words, protein, right? A small segment. So cistron is mentioned. What? Okay. So look here, students, it's a segment of the DNA coding for a polypeptide. Okay, a segment of DNA, coding, instead of forming I'll write the word coding. Okay, coding for polypeptide. Okay, coding for polypeptide. Aryan, you can ask your batch teachers only, no? That is the best thing. Okay, they only finish the syllabus, no? So they will know the answer for it. Yes. So a segment of DNA that is coding for a polypeptide. Clear? This is what a cistron is. Now, please keep your mind open. There is two things that you have to learn. Monocistronic and polycistronic. Monocistronic and polycistronic. Now, what does mono means? Mono means single. Let's consider this as an mRNA. Okay, one mRNA. This is your DNA. Okay, mRNA will code for one protein. I told you that is according to our central dogma. Okay, protein. Correct? Now, on the other hand, polycistronic means what? Multiple or more than one. Right? More than one. This word poly is associated with prokaryotes. Poly is associated with prokaryotes. This question can come asking you polycistronic and this. So here remember students, DNA, let's say mRNA is there. Okay, mRNA is there. They will code for multiple proteins. They will code for multiple proteins. No problem at all. Okay, no problem at all. Understood? What is monocistronic and polycistronic? Yes? Yes. Very good. More than one and single. So in eukaryotes, it's monocistronic gene. That means we will code for one polypeptide. That means we will code for one protein. Here, poly means many, more than one. Okay, more than one segment is there which will be able to code for a protein. Understood? This is called as polycistronic. So that means they have more segments which are able to code for polypeptide, but we have only one segment that will code. So understand that we are talking about one segment. Okay, one segment and there many segments or more than one segments. Many segments. Done. Now, one more very important process, very important process. Okay, this is going to be, students, what is exactly happening. So as I told you when we spoke about cistrons also, you should remember that eukaryote genes, okay, are called as split genes. What are they called? Split genes. What is split genes? Students, in the gene we have two things. Okay, we have coding and non-coding. Coding and non-coding sequence. Now, because of coding and non-coding sequence, our genes are called as split genes, and we do not want the area which is not coding, right? Because we are ultimately coding for the polypeptide. So, we do not need regions which are non-coding, which are called as introns. And the coding region is called as exons. So, to remove the introns, our eukaryotic genes go through a process called as RNA processing. What do we call it as? RNA processing. This processing does not happen in prokaryotes because their translation and transcription is coupled. Okay, it will not happen in prokaryotes. In eukaryotes, we have something called a split gene which ultimately forms your has both introns and exons. So, the process of removing introns is called as splicing. Okay, this process where I am removing the introns is called as RNA splicing, so that I do not get any non-sequencing portion in my mRNA. So once I remove all the genes, okay, all the introns, that newly formed mRNA will go through two processes which is called as capping and tailing. Capping will be done with the help of methyl guanosine triphosphate, as you can see here, MGPP, okay, MGPP, that is what I told you, triphosphate. This is a nucleoside with phosphates, okay. So capping will happen with the help of methyl guanosine triphosphate, and tailing will happen with the help of poly-A tail, okay, poly-A tail. Clear, all of you? Why this is done? Because this mRNA has to go from the nucleus, it will go from the nucleus to the cytoplasm to undergo translation. So for that we will make sure that we do capping and tailing. Now if I send you to a guest home, I will definitely put you cap, I will tell you behave like this, don't do like this, take care of yourself. If some strangers give you something, don't take. Why do I do that? Because I know you're going from one place to another place. Same way here also, students, we do capping and tailing so that there is no unnecessary add-ons. At the same time, we are also getting a 5' and a 3' end. Proper marking is there, okay. So capping will happen with methyl guanosine triphosphate. I will write it here, okay, methyl guanosine triphosphate, and tailing will happen with poly-A tail, okay, tailing will happen with poly-A tail. So this mRNA, newly formed mRNA, will be going from the nucleus to the cytoplasm for translation, and students, in between it will form the hnRNA, okay. What is the full form of hnRNA? Tell me in the chats, all of you, quickly. Any doubts in this, by the way? Any doubts in this? Please let me know. Please let me know if you have any doubts. And also tell me the full form of hnRNA. Yes, very good, Padmavati. Okay, heterogeneous very good, heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA. Heterogeneous is name, why? Because there is combination of both. Shall I give you a break now so that 8 o'clock we can resume back? Okay, I'll make you people do questions after genetic code. Okay, I've put the questions, students. Questions are there. Now we have translation, HGP, operon. 1 more hour we will need. Okay, yes. So I think I'll inform Baswa sir. Okay, now questions are here for you. Okay, so yes, we are going for a break. Okay, done? Happy? All of you will come back? If all of you won't come back, then I won't give you break. I can still take class. Okay? Will you all come back? And students, I want to tell you people, this offer was planned to end on Jan 1st, but they have extended it for 2 more days. Up to Jan 5th, this is extended, okay? So those who want to take, you still have a chance. I would recommend all of you to go for this. That is your All India test series is there. Okay, you will get around 22 tests. Okay, along with 30 OMR sheets. So you can definitely practice with the OMR sheet, okay? So you can take this test. So make sure that you make your decision and buy this test. Right now it's at 1,400. Later the price will increase. Okay, so yes, so see you all at 8:05. Okay, it's already time. I will run outside. Stay in the live itself. Don't go anywhere. Just go have some coffee, something hot, and come back. Okay, ready? Yes. Okay, done. Okay, let's start. My God, we have so much to do. Oh. Yes, okay. Now students, we have to do genetic code. Okay, let's start. So, genetic code, basically what happened during the genetic code, we had around 20 amino acids, okay. So, there was a person called George Gamow. He was a physicist and he was like, wait a minute, you are having a problem. What is the problem that we have? The problem that we have, see, many of them did not come back. Wait, I'm assuming they'll come back now. One second, students. Where did these people go? This is why I don't give breaks. All are one number lazy people. Okay, yes, fine. Okay, listen here. George Gamow was like, okay, so you people are telling me that you have AUGC as the nucleotide, but we have 20 amino acids to code, right? 20 amino acids to code. So he was like, okay, let's try permutation and combination and let's do this. That is, we will try 4 to the power 1. That means we are making a combination of individually AUGC. Okay, individually taking 1, 1, 1, 1. Now you tell me 4 to the power 1 will code for how many? It will code for only 4 amino acids. So not possible. Okay, then they were like, let's do one thing. Let's try 4 to the power 2. That is where I form AUGC or let's say CUAG, something like this. Okay, where we form permutation and combination of 2 of them. This would lead to the formation of 16. It would allow us to code for 16 amino acids, but still it is less than 20. Okay, it is less than 20. So what is going to happen? This also wasn't agreed. Then they were like, let's try the last one, that is 4 to the power 3, which will lead to the formation of your triplets like your AUG, UUU, triplets that will help us code for 64 amino acids. Even though we had found 20 amino acids, we were ready to make more. Okay, more is better than less, right? More is better. So 64 amino acids, and out of which currently what we have 61 coding for amino acids and we have 3 stop codons. Okay, we have 3 stop codons. Clear to all of you? So this was what happened. Now there are 2 other important things that you need to learn. That is about Hargobind Khurana. He synthesized homopolymers. Now what is homopolymers? Basically students, like this, A, okay, U, like this, okay, homopolymers he made. Understood? So these are homopolymers that he found. Now next thing, Marshall Nirenberg, he developed cell-free system for protein synthesis. So imagine one person is capable of forming your entire mRNA because he was also able to form templates, right? He was able to form templates, RNA templates. Without the cell, he was able to form RNA templates at the lab. And there was another person who was able to form protein at the lab. So imagine how much of a thing they are, right? Hargobind and these 2 people can entirely make your central dogma happen inside the lab. So he was able to form RNA template and he is able to form cell-free system for protein synthesis. So please remember their names, they can ask you as a question. Okay, so the genetic code is clear, no? 61 codons. Now students, how did they come across all this triplet formation? Remember that there were 3, like how you studied your Punnett square. Here you have 3 positions: first position, second position and third position. As you can see, U will lead to the formation of the first one. Same way, UUC will lead to the formation of third, UUA, UUG. This will lead to the formation of the first box. Then goes the second box. Like this, they found it. Luckily, you do not have to study all of it, but some common ones you can go through. Okay. like methionine, which is already informed for you. Valine, you can see GUU, so just remember this formation. UCAG and GU together will form the valine. Okay, and students also remember the stop codons UGA, UAA and UAG. Okay, these three are the stop codons. What is it? UAA, okay, UGA and UAG. Clear? These three will form the triplet. You understood, no? How we formed the triplet? It was by permutation and combination, where we did 4 to the power 1, 4 to the power 2, 4 to the power 3 and finally landed in 64 codons. Now out of the 64 codons, 61 codons are going to code for amino acids and 3 are going to be the stop codons. Problem sorted. Now some amino acids are coded by more than one codon. Okay, some amino acids are coded by more than one codon. This shows that the code is degenerate. I had made a short on this and all of you would have seen that. Okay, all of you would have seen that. Now the codon is read in mRNA in a continuous fashion. That means no punctuation, no commas, nothing. You will always notice that it will be written like this. You will read it like this only. AUG, UUU, CCC. You will read it like this, not with the help of any punctuations. Okay. Yes, the code is universal. That means be it from your bacteria up to your larger organism, everything is going to have the same coding. That is, here, look here. From bacteria to humans, your UUU will code for phenylalanine. That means this is going to be a universal thing. Okay, it's going to be a universal thing. Degenerate means more than one. See, now example students, as I told you, valine. UUU codes for valine. UGA codes for valine. GUC codes for valine. That means what? This means for one amino acid, multiple codons will code. That means the code is degenerate. Okay. So more than one codon. Now AUG has a dual function. What is the dual function? It acts as an initiator. Okay, it acts as an initiator. At the same time, it's also capable of coding for methionine. Clear? It acts as an initiator codon as well as methionine. It also codes for methionine. Is this clear? Okay. Yes or no? Clear? Evolution we have already taken, dear. I have already taken 2 times. Okay. So you can go and check that out. And evolution has comparatively less weightage. That's why we are not taking it here. And if I would have taken evolution, you people would have slept here only in this. I had to sleep in the studio and you had to sleep, okay, wherever you are at. Okay, so that will become too long of a lecture, but we will definitely take it. Don't worry. Now UAA, UGA and UAG are the 3 stop codons. Is it easy? Yes, ma'am, it is easy. What is the salient feature? It is triplet, degenerates, it's universal, it is not written in a continuous fashion, no punctuations. It is written in a continuous fashion, no punctuations or comma. And then AUG has dual function, codes for methionine. At the same time, it is also an initiator codon and there are 3 stop codons. Okay, clear or not? Now, very important thing, insertion and deletion. Okay, insertion and deletion. Now look here students, RAM has red cap. Okay, RAM has red cap. Frame, any frame shift is there, your reading also is proper, right? No changes in the reading. Now if you look here, RAM has BRE, DCA, P. Okay, can you see why? Because ma'am, you just now told the genetic code will be read in a continuous fashion. If it is going to be read in a continuous fashion, will they bother about if some letters are missed or not? No, it is going to read continuously, right? It is going to read continuously. So here what has happened students, insertion of this one thing has changed the entire reading frame. Okay. Now again 2 letters have been added. Again it is reading in the wrong way. RAM has BIR EDC AP because it is not going to give the break. It is not going to check, oh, it's the grammar teacher or what they are. No, they are not going to check. They are going to read it in a continuous fashion and they are going to do the adding. This is just an example. Okay. Now what has happened, when we added 3 letters, when we added 3 bases, okay, 3 bases, here what happens students, no frame shift happened. Okay, no frame shift happened. That means what? By adding 3 letters, frame shift mutation will not happen. But by adding 1 or 2, this can lead to change in the frame. Understood all of you? So that means when we are adding also, we need to add a triplet. Okay, when we add also, we need to add a triplet. Okay. Okay, very good. You love blue, so you can write it. Okay, clear or not? So when we add also, it should be in a triplet. Okay, it should be in a triplet to avoid frame shift. Okay, to avoid frame shift mutation. Now here if you see, deletion has happened, students. Deletion also, you will notice that ED, okay, DCA, and now removal of all these three has led to the RAM has red cap. So this also proves that when we are doing deletion also, deletion and insertion, okay, deletion and insertion will lead, okay, deletion and insertion will lead to mutation, but if deletion and insertion is with triplets, then it will not lead to mutation. That, I mean, what frame shift will not happen. Okay, if triplets are added, okay, no, okay, no frame shift. Clear? No frame shift. Okay, this is it. Okay, now tRNA. Okay, tRNA. All of you have the energy, stamina is there, not tired? Put some fire emojis. Yes. Okay. So here, one more thing we will add about your sickle cell anemia, which is point mutation. So what happens, students? GUG changed to GAG, that led to the coding of glutamate. Instead of glutamate, valine got coded. So that led to sickle cell anemia. Okay, sickle cell anemia. This is a point mutation, right? Yes. Okay, this is a point. Oh, have I written U here? Point mutation. Point mutation. Correct? Yes, good job, good job. Frame shift mutation. So this is a point mutation. Point mutation. Okay, done. Now tRNA. Very important. Listen to me carefully. Now look at the structure of tRNA. As I told you, RNA might not be a genetic material for all the organism, but they are doing the job of a catalyzing job. Okay, that is acting as a catalyst, acting as an adapter, acting as a moderator, so acting as a messenger. All this job is done by tRNA, and one of the most important jobs, okay, important jobs of tRNA is acting as an adapter molecule, that is your tRNA. Now remember, students, your tRNA in 2D, okay, in 2D it looks like a clover leaf model, okay, clover leaf model, and in 3D it looks like an inverted L, okay, inverted L, right? So this tRNA, why we call it as a clover leaf model? Because there is one loop, okay, one loop which is an anticodon loop, which you are seeing here. Okay, this is your anticodon loop, which will form anticodons. Okay, now example, AGU. What is code for A? U. G, C, U, A. So this is the actual codon. These are the anticodons. So in one side of the tRNA you have an anticodon loop, and on the other side of the tRNA you have an amino acid acceptor site. Okay, acceptor site. Because the genetic code that we spoke about now, where are we going to utilize it exactly? We are going to use it when we see the mRNA, because on the mRNA the bases are going to be arranged, and we should know exactly this triplet is going to code for which amino acid, and the tRNA will correctly attach that amino acid and finally form a polypeptide chain. So this is the entire circle. So that is why you have learned genetic code, right? Now look here, students, this is a tRNA structure, and you will notice that they have an anticodon loop as well as they have an amino acid acceptor site. So here AGU will code for serine, and here you have UAC, AUG will code for tyrosine. So this means that they will form anticodon and they will exactly code for that amino acids. So tRNA is in 2D is a clover leaf model and in 3D it's an inverted L-shaped structure. Okay? Yes. Clear? All of you? Yes. Bruce Lee, you do one thing, you jump from the wherever you're sitting, you just jump down and jump back. So it will become very interesting. Okay? It will become very, very interesting. Yes. Clear? Fine. Now, moving forward, what is translation? Okay, what is exactly translation? So when you learn translation, you just have to learn just few things. Okay, few things. For that, I will just draw a diagram for you. See, now this is an mRNA. Okay, this is an mRNA. Let's say 5'. Okay, okay, wait, before we put this, students. Okay, now in this mRNA, there are few things that you need to understand. That is, did you jump? Okay, if you jump and something happens to you, don't tell it on me. Just tell ma'am, I asked ma'am to make some fun. So ma'am made fun of me. Clear? Yes. Okay. Good. Now look here, students. In translation also there is an initiating factor, there is a terminating factor. Now for that to happen, remember in our genetic code itself we have stop codon to terminate the whole process. But before, okay, starting and at the ending you have something called as the UTR. Now in prokaryotes it could be your Shine-Dalgarno sequence, okay, which you don't have to know now, but there is a sequence in prokaryotes which will immediately recognize, and the smaller ribosome will come and attach to it, and then larger ribosome will come and attach. Now when you come to your eukaryotes, you have your capping and tailing done, okay, your methyl guanosine triphosphate, poly A tail, these people will act as a UTR. That means what? These regions will not translate at all. Even though they are present in mRNA, they will not form the protein. Okay. Now look here, this is your mRNA. Now here the major thing is your two ribosomal unit, a larger subunit and a smaller subunit. Okay. So this is your larger subunit and this is your smaller subunit. On the larger subunit and the smaller subunit, students, there are mainly three regions, which is the E site, P site and the A site. Okay, what is there? This is what is shown there. This is what is shown there. So remember that when your tRNA is coming, okay, one tRNA is going to come here, okay, with the anticodon loop or whatever. This tRNA is going to be accepted here, and from here it is going to be shift here, and then it will move out. Okay, then it will move out. So your ribosome plays a very important role of where your smaller subunit, okay, smaller subunit of the ribosome is going to help in the initiation to completely start. So tRNA with your ribosome entirely forms your translational unit. Now translation means what? mRNA to protein. Okay. So who is going to read the code? tRNA. tRNA will bring its anticodon. So understand in this, from this diagram, what do you understand? You understand that the tRNA is specific, right? tRNA is specific for each amino acids. Every tRNA cannot code for everything. No, can you see? He is bringing only the anticodon for this fellow. He is not bringing for everyone. So tRNA is specific. Okay? Yes? Correct? Yes, Wakanda, even I want to know if I'll complete the entire chapter. With your motivation, I'll complete. Okay. Now students, if this is clear, understand that the first ever process for translation to happen is called as charging of tRNA. Students, I can bring a PPT and complete the chapter in 3 hours or less, but I don't want to do that because I want to write, I want to draw the diagrams. That only makes it important. Okay, important. Clear? Yes. So here, look here, charging of tRNA, which can also be called as amino acylation. Okay, amino acylation of tRNA. Now what is amino acylation? Simple thing, students. Energy is going to be added. Why energy is going to be added? Because when two charged tRNAs come together, okay, let's say this is one tRNA. Okay, I'm drawing it as a box. This is tRNA, this is fully charged. Okay, this is fully charged. And then we have another tRNA. Okay. This tRNA also is fully charged. Now, when this two tRNA, which is fully charged, come together, Okay, when they come together, they will end up forming a polypeptide bond. This is given in your NCERT. They will form a polypeptide bond. So, amino acylation or charging of tRNA is very important. That means where we are going to add ATP. Okay, ATP to charge your tRNA, right? Energy is going to be utilized. Clear? Yes? Okay. Any doubts? Janani? Okay. Yes. Coming. Yes, explaining. 1 second. So, here what is happening, students? Amino acid plus ATP. So, this is the whole charging process. So, the 3 sites that I was talking about is the E site, P site and A site. These 3 sites are present where in the ribosome, right? So, E site is the exit site, P site is where the peptide, that means polypeptide chain will be formed, and A site is the acceptor site. This is where your tRNA will come and bind. Clear? So, what is going to happen? The tRNA will come with the anticodon loop, it will come and bind correctly, it will code for the amino acid, then it will be passed to the polypeptide, more chains will be added, from there it will exit and go. So, this process will keep continuing. Clear or not? So, the 3 sites which are present. Now, students, remember your ribosome unit. Okay, the smaller ribosome unit plays a very important role in initiating the whole process. Now, as I told you, in this region, if you have MGPP, okay, that is your methyl guanosine triphosphate, these people are going to act as like your untranslated region or your UTRs. Okay, same way here also your poly A tail will act as your UTRs, that is untranslated region. Okay, untranslated region. That means the regions which will not, even though, okay, even though, yes, Meghna has put the target, very good. Even though these people are present on the mRNA, they will not get translated. That is called as untranslated region. This also helps in improving the efficiency of translation. Okay, improving the efficiency of translation. Is this clear to all of you? Improving the efficiency of translation. Now, remember, shall I write it here? Yes. So, in the termination, students, there is one more factor called the release factor. All of you see your NCERT, release factor is highlighted. In the termination, there is a factor called as the release factor, which is going to help with the termination. With the release factor, your stop codons are going to be present on the mRNA. Okay, on the mRNA, stop codons are going to be present. They are also going to help. And then you have UTRs at both the ends. Okay, in the starting as well as in the ending. Any doubts? Any doubts? Is coming here, this tRNA will shift here. Okay, so look at my arrow mark. When this tRNA is coming here, this tRNA will shift here because it will be ready to exit. So, this entire thing, can you see this arrow mark? This entire thing will be given to this fellow. So, when this tRNA comes, this two will form a polypeptide chain. When this two tRNA comes, these two will form a polypeptide chain. And this is only because of the charging. Okay, this is only because of the charging. If you don't understand this, please raise your hands. I hope it is clear. So, when two tRNAs come together, when they are charged, you remember that okay, when two tRNAs are coming together, they are going to form a polypeptide chain, and this is only because of the charged tRNA. Okay, now the next topic is gene regulation, students. That is, the gene can decide when I should be active, when I should not be active. Okay, just like you people. Now, you might be sitting in class, some of you will be active, some of you will be texting someone else, some of you will keep my, class live on YouTube and then will be texting someone else. So, when your parents hear, they'll be like, okay, this ma'am is shouting from past 3 hours, my student is studying. No, only I am studying, okay? No one else is studying. Okay, so yes, not no one else, some of you. Now, students, just like that, the gene decides when it has to be switched on and when it has to be switched off. But there are separate types of genes because in our body so many processes happen. We cannot expect some genes to get switched off, right? So here what is going to happen, students? Remember, gene regulation happens when two genes are there. First is called the housekeeping genes. Okay, and one is called the luxury genes. Now, housekeeping genes means what? The people who are like a house help. They have to full-time work. They have to always be active, whatever we ask them to do, right? So, same way here also, housekeeping or constitutive genes, these will be always switched on. Like example, the genes that work for glycolysis, they will always be switched on. They do not have a choice to switch off. Now, here you have luxury genes or non-constitutive genes, which will regulate itself. That means they will decide when to get switched on, when to get switch off. Now, in eukaryotes, when can they decide to switch on and switch off? Look here, in eukaryotes, they can decide during transcription, during RNA processing, that means splicing, capping is happening, tailing is happening. When the mRNA is transferred from nucleus to cytoplasm, they can decide, or when translation is happening. During these 4 steps, the gene can decide to switch on or switch off, up to the gene. Okay. Now, but in prokaryotes, what happens? Such kind of complications will not happen. Only in one step, gene can decide to switch off and switch on, that is during transcription. As I told you, translation is going to be coupled. So that DNA to mRNA stage only they can switch off. mRNA going outside, no RNA processing is happening. So because of this coupling, there is only one step where they can switch off. Is this clear to all of you? Yes or no? Clear? Okay, one diagram and then you will understand the whole thing. Okay. Okay, this is a cell. Okay, this is the eukaryotic cell. This is the DNA. Okay, DNA to it can become mRNA. Now, after this mRNA formation happen, RNA processing can happen, right? RNA processing can happen where they will cut off the split genes. Let's say this is processing. Okay, after processing, we got a newly formed mRNA. Okay, now when this mRNA is planning to shift outside, that means to go to the cytoplasm. Okay, to the cytoplasm, and from cytoplasm when it forms a polypeptide chain. These are the 4 places when the gene can decide to switch on and switch off. That is 1 transcription, 2 RNA processing, 3 transport and 4 polypeptide. This is what happens in eukaryotes. That is during any of these 4 stages. Okay, you can't see the red, no? Okay, during any of these 4 stages, they can decide to switch on or switch off. Any doubts in this? Please ask me right now. It's a very important concept. This question has come to all for I think one of the years it has come, that gene regulation can happen in which all stages. Okay, so this is what happens in eukaryotes. Okay, eukaryotes. Now in prokaryotes, what happens, students? In prokaryotes, only during the transcription stage this can happen. 2 genes as there, housekeeping and luxury genes. Housekeeping genes have to be switched on full time. They do not have a regulation, but luxury genes have that ability. Okay, now according to your NCERT, you have to study some of the gene regulation, that is going to study about the operon model. Okay, you're going to learn about a model which is called the operon model. Okay, histidine, operon. So depending on the operon, depending on the thing, we have different types of operon models. Okay, operon models. Now the operon model that you're going to study is Lac operon model by Jacob and Monod. Okay, Jacob and Monod. This is the model that you're going to study. Okay, are you all ready for some questions? We will do 5 minutes of questions and after that I will continue. Lagging is there. We will do 5 minutes of questions. Okay. For 2-3 seconds it was there, no? Now fine, huh? When was it lagging? Okay. Now, according to your NCERT, you have to study. Okay. Yes. I think it should work fine, students. So, yeah. So, answer this question, all of you. In Hershey and Chase experiment, what finding led to the conclusion that DNA is a genetic material? Quickly answer. Listen, I'll make you people do the questions because anyway, test is assigned in the chat box. Okay. Quickly answer. No time. Okay, Baswa sir also has to start the class. Very good. Answer is going to be 3. Okay, very, very good. So you got in GM class first 4 marks you got. Okay, +4. Next question. DNA fragments are negatively charged, positively charged, neutral, and depends on the size. NEET 2017 question. NEET 2017 question. Next question also answer. Okay, good job, good job. A for Ammu ma'am. Yes, that is going to be, yes, very good. Negatively charged. So +8. How many of you got +8 in the chats and also in the comments? Okay, very good. Next question. The final proof for DNA as a genetic material comes from the experiment of, final proof, okay, of the experiment comes from whom? Yes, good job. Hershey and Chase experiment, +12. Okay, +12. Next question. Which of the following is not a component of a nucleotide? Which of the following is not a component of a nucleotide? Which of the following is not a component of a nucleotide? Yes, answer. Quickly, quickly, quickly. Baswa sir will scold me now. Oh, what happened? Here, nitrogenous base, phosphate group, ribose sugar. What is the answer? Deoxyribose sugar. Now, next one. Which of the following, okay, which of the following, yes, which of the following nitrogenous base is found only in RNA? Which of the following nitrogenous bases is formed in RNA? +16. Yes, come on, 20 questions, 20 marks. Which of the following nitrogenous bases is found only in RNA? Yes, good job, good job. Yes, that is going to be uracil +20. Okay, done? Done. Okay. Now we will start with lac operon. Okay, good job. Those who got 20, please put it in the comments. I will see your names. Okay. Now, as I told you, students, lac operon is found by Jacob and Monod. Now, what is there in this? You should remember there is R for repressor. Okay, repressor, or you can also call it as an inhibitor. And then we have structural genes. Okay, structural genes. What is structural gene? The gene that is the region that will code for an mRNA, that will form an mRNA, right? So this structural gene region is there. that is capable of forming an mRNA. Structural gene you have studied in your transcription, same that structural gene only is here. And decide to switch on or switch off. Okay. Now look at the scenario, students. The I gene will form repressor mRNA. mRNA will form repressor protein. This repressor protein will go and bind with the operator. Now stopping everything from happening. RNA polymerase which will be coming, let's say RNA polymerase reached here. Can it move further? It cannot move further because there is a big block. Okay, there is a big block. Okay, just wait for a minute. Operator. Now, repressor gene will form repressor mRNA. Others, is it lagging? This repressor protein will go and bind with the operator. Now, stopping everything from happening. RNA polymerase. Now it's clearer. I don't know what is wrong here. Students, today nazar fell, nazar. Okay, bad eyes. Okay, look here. So what is happening here? Repressor will go and bind with the operator. When does this happen? Structural gene to express itself or form the mRNA. Now, Z, okay, Z, YA, important, Z will release beta galactosidase which has the ability to break down lactose into galactose and glucose. Permease which will increase the permeability to accept or to absorb more lactose from the outside, and trans-acetylase function is unknown. Okay, function is unknown. Now students, look here, what is going to happen when lactose is going to be present? That means the gene has decided to switch on. What is going to happen? Again, same thing will happen. Repressor will form mRNA. Yes ma'am, repressor will form mRNA. Repressor will form repressor protein. Yes ma'am, repressor protein also will be formed. Now, inducer, that is lactose, will go and bind with the repressor and not let the repressor go and bind with the operator. So RNA polymerase which is going to come, it is going to happily go forward and start with transcription. This is happening in prokaryotes, so only in one step we can stop them, that is in transcription. Students, is it fine? I cannot see any of your message, or are you not messaging? I can't. Okay, no problem. Okay, so transcription is going to happen and your ZYA is also going to form your lac mRNA and beta galactosidase, permease and trans-acetylase. Please understand that the process, the lac operon model is a leaky model. That is why lactose is already entered, and here we have a permease that is going to increase the permeability for absorbing more lactose. Understood the problem? So it's a leaky model. Okay, lactose has entered somehow. That is why inducer job it is doing. Any doubts in this? Any doubts in this? Okay, so students, Z, okay, Z gene, Y gene and A gene are structural genes. Z gene, beta galactosidase, okay, and what is it going to do? It is going to break down lactose to galactose and glucose. Okay, galactose plus glucose. Okay, and then you have permease which will increase the permeability. Increase permeability, and then you have A which is going to release trans-acetylase whose function is unknown. Okay, trans-acetylase. This is what is going to happen, and these three people are the structural genes, so they are going to help in the formation of the lac mRNA. That means transcription has to happen. So this will happen only if lactose is present. That means the gene has decided to switch on. So simple, students, lactose is not there, gene is switch off. Transcription will not happen. RNA polymerase will get blocked near the operator. Okay. If lactose is present, repressor will not go and bind with the operator because the inducer, that is the lactose, will go and bind. Okay. Lactose will go and bind with the inducer and it will not let the repressor to go and bind with the operator. So repressor protein's main job is to inhibit the process. It is acting as an inhibitor. Okay, inducer is going to be lactose who is going to stop the repressor from going and binding with the operator, because if it goes and binds with the operator, RNA polymerase will not be able to go further. Clear? Yes? Wakanda, is your doubt clear? That is what is going to happen. Okay, so during this entire process, who is going to be there? RNA polymerase is going to be there. RNA polymerase is going to be present. This is going to help in the transcription. Okay, that is your operon model for you. Now, HGP. Okay, last second topic. After this, we'll be done. I hope in another 10 minutes we should be able to finish. Now, HGP students, there's no a lot about HGP, but whatever is there, we will learn. Okay, HGP, first of all, full form is what? Human Genome Project. Okay, Human Genome Project. Now, during this entire process was done with a lot of fun, and it was the mega project. Why is it called a mega project? Because around 9 billion was invested for this entire process to happen. And why it was done? To make sure that entire, okay, human gene, that is your haploid content, okay, entire haploid content, okay, haploid content can be understood, and every single thing can be studied so that we can make the entire process more easier. Clear to all of you? Very good. Yes. 9 billion. Very good. It was a 13 years project. Okay, it was a 13 years project, and because of these two reasons, it is called as the mega project. Okay, 13 years project. Now, students, understand, when we talk about your HGP, okay, when we talk about your HGP, it was actually understood that we have around 3.3 into 10 to the power 9 base pairs. So, if I want to make it into a place, okay, properly understand, then I need 3,300 books, okay, an entire library with each book having 1000 pages. And each page is having 1000 words. Sorry, 1000 letters. Will anyone be able to carry this book? Will anyone be able to, walk around and see about the book? No. So they were like, we should make all this into a computerized system, and that is when they came up with bioinformatics, also helping them with this process, okay, where we mix, biology plus computers to get a better way to understand this whole, haploid content of our DNA. Clear to all of you? Any doubts in this? Okay, many people came into this. France, Germany, India also was a part, UK was a part, China was, so many people came together for this entire HGP project. Many renowned institute people came into this, okay. Then, with this, we also came across Sanger sequencing. That means taking a small portion and sequencing it, okay. That also came into this existence, students. Now, there's a lot to learn, but whatever is in PYQ point of view, I will teach you. So, first of all, goals of HGP, which is very, very important. Now, keeping everything in mind, they try to make things more better for a future process. Now, look here, identify all approximately 20 to 25,000 genes in a human DNA, okay. Determine the sequence of 3 billion chemical base pairs, okay, that is our haploid content, to make up the human DNA. Store this information in a database, because book was practically not possible. Imagine Karthi had 3,300 books with 1000 pages in 1000 letters. Not possible, right? So, keep it in a database. Now, improve tools for data analysis. Also improve the tools, okay, for data analysis. Next thing, transfer related information, which is going to help in the future to other sectors such as industries, okay, pharma, other industries where this information could be helpful. Address the ELSA. ELSA is very important. They can ask you full form. Ethical, legal, social issues. Full form. Ethical, legal, social issues. Okay. There is no lagging, no? Yeah. Ethical, legal and social issues we could deal with. So, address that also. This was the entire goal of HGP. Now, for that, students, they mainly went up to two methodologies. Two main methodologies. Again, not a lot to learn. Okay, just learn the words. Okay, two methodologies. One was a blind approach. Okay, one was a blind approach. A blind approach means does not matter if these genes are going to sequence or not, or code or not. A completely blind approach where we take the whole genome. Okay, whole genome that means both the exons and the introns. Okay, exons and introns. Okay, this was one attempt, okay, where we take a blind approach, thinking nothing, just take the whole genome. Okay, just take the whole genome. Another approach was an expressed sequence tags. Okay, I will share these notes, don't worry, after the class I will share the notes. Okay, and I'll tell them to add a link also in the description. Express sequence tag, which is called as ESTs. Students, this was more specific. Okay, this was more specific. So please write this to approach and keep. This is very, very important. That is, the only DNA that is going to be expressed was taken under this category. Okay. Okay. Oh, okay, great. Students, there's an announcement. That's just, okay, sir will be going live and it is very important. Okay, don't miss the live. Yes. What is the announcement? Okay. Yes, so as the live happens, you can just go join there. Okay. We'll wait for 2-3 minutes. Okay, in 1 minute the live will happen. Okay, just join the live, students, in an important announcement, and then come back to this class. I'll be here only. Okay, by then I will just draw the genomes. Okay, you can go there and come back. Yes, exactly. You can see the announcement and come back. Okay. Yes. Has the live started, students? No. Not yet. Okay. I'm also waiting for the update. I want to know what is happening. Okay, so when they start, you people just go there, okay? We will finish off this first. Okay, so students, as I told you, two approach was done. One was blind approach where they took the whole genome, exons and introns were taken, and then there was an expressed sequence tags where they took only the expressed, which was expressing itself. Okay, only those they took. This was the two things. In bases, but size vary with the largest known gene. This is important. Largest known human gene called dystrophin, which is 2.4 million bases. Okay, 2.4 million bases. Is it stuck? Oh God. Today lag day it is. Okay. Yes, now it's fine. I don't know what is wrong with the internet here, students. Is it fine now? Please tell me. Okay, we have just one more topic, that's why. Yes, okay. The total number of gene is estimated at 30,000, much lower than the previous estimated. So previously they estimated around 80,000 to 1,40,000, but later when they understood everything, they realized, okay, when they understood everything, Janani, just check your internet, dear. So when they understood everything, they realized there are only estimated 30,000. Okay. Now important, the functions are unknown for over 50% of the discovered genes. Now less than 2% of the genome codes for protein. So you understood? That means there are less cistrons itself. That means the segments of the DNA which is going to code for the polypeptide chain, that is only less than 2%. Okay. Now repeated sequence make up a very large portion of the human genome, which I'll talk about. Repetitive sequences are stretches of DNA sequence. Important. Repetitive DNA sequences are stretches of DNA sequence that are repeated many times, sometimes in hundreds to thousands. Okay. Sometimes in hundreds to, one second, yeah, sometimes to hundreds to 1,000 times. They are thought to have no direct coding. So students, here very important thing what you need to remember is, in all of us, okay, me, you, everyone, all of us have 99.9% DNA the same. Can you believe? And this DNA is called the bulk DNA. Bulk DNA. Okay. Now 0.1% is different. And you will be like, ma'am, seriously, that 0.1% is making a difference? Yes, it is making a difference. Okay. And that difference is because of repetitive sequences. Okay. What is it? Repetitive sequences, which we also will call as the satellite DNA when we learn about the DNA fingerprinting. So these are the repetitive sequences. These repetitive sequences can repeat in hundreds to thousands. Okay, they can be from hundreds to thousands. So let's take example CCT. Okay, this is one sequence. This sequence maybe in me, it might repeat 50 times. Okay, 50 times. But maybe, let's say in Meghana, it repeats only, okay, Meghana, it repeats only 5 times. That makes both of us different. That makes both of us different. Clear to all of you? This 0.1% is what we use in DNA fingerprinting, which is also simply called as satellite DNA, which has two portions, mini satellite and micro satellite region. Okay, clear to all of you? So here, please understand that difference in one nucleotide can lead to SNPs, which is called as single nucleotide polymorphism. Okay, just one nucleotide, if it changes, that will be called as single nucleotide polymorphism, which makes me and you different, which makes my hair texture, your hair texture different. Like that, everything can change because of this small changes. Okay, because of this small changes. Now, scientists have identified about chromosome 1 has most genes, that is 2968, and Y has the fewest, 231. Please remember this number. Scientists have identified about 1.4 million locations where single base DNA difference, SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in human. This information promises to revolutionize the process of finding chromosomal location for diseases associated sequence. Now, sickle cell anemia. How do I know sickle cell anemia? It is because of the change from GAG to GUG, because of that single nucleotide, that single base that changed led to the sickle cell anemia. So for a point mutation, I need to make sure that I know what is SNPs. I need to know what is SNPs. Understood? So this is your HGP project. Okay. Now HGP and DNA fingerprinting, you have some continuations here. So I will just go through it in a very quick form. Okay. My tongue is twisting, students. Yes. So as I told you, in your entire DNA, 99.9% is not what we are bothered of. But we are bothered of the 0.1% which we will be using in DNA fingerprinting. Okay, DNA fingerprinting. Now, what is this 1% called as? This 1% is called as your repetitive sequence. Okay, 1 second, students. Hello. Okay, 1 second. Yes. Students, has the live started? No. Have it started? Yeah, started.
Conduct the exam multiple times, to conduct it online, which again seems to be pretty difficult because there are too many students who are going to write the examination. And then also there was a discussion regarding the hybrid mode, and where you have to mark it on the OMR sheet so that the paper does not get leaked. Also, conduct it online.
Okay, live has started, students. Announcement has started. Okay. Yeah. Until sir finishes, it's a break, okay? portion is there. But so sir, otherwise have to stay till 2 o'clock. Okay. Yes. So repetitive sequence, as I told you. So as I told you, 0.1% is going to be the one who is going to play a very important role in your DNA fingerprinting. Okay. In your DNA fingerprinting. Now remember, this repetitive sequence can also be called as your satellite DNA. Satellite DNA. Now, this satellite DNA, we always talk about two main things. Okay, remember, two main things are going to play a role in your DNA fingerprinting. One is called your STRs. Okay, that is short tandem repeats. Okay, short tandem repeats, or your VNR. Okay, VNTR, that is variable tandem repeats. Okay, variable tandem repeats. Now students, how do these people play a role? It's very simple. Short tandem repeats, let's say ATGCCC, 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 5 times it would have repeated. 5 times it would have repeated, okay? So what is going to happen? That means they are small sequence, right? All of them came back. Why can't I see your numbers? Okay, why can't I see your numbers? Yes, 1 second. Okay, now students, look here. STR is short tandem repeats, VNTR is variable tandem repeats. Now this short tandem repeats, I can also call it as the microsatellite region, and this region I can call it as the mini satellite, correct? Now microsatellite region and mini satellite region is important for you to remember. Why? I'll tell you. Because microsatellite region, as I told you, AGTTCC will repeat only 1 to 6 times. Okay, 1 to 6 times. So remember this, 1 to 6 base pair repetition will happen. Smaller repetitions, okay, 5 to 20 times. 5 to 20 times this will repeat. Okay, and this is going to be an AT rich region. Okay, AT rich region. And then when you talk about mini satellite, students, mini satellite region, it is going to, see here it is only 1 to 6. Here it will go somewhere around up to 100. Okay, around from, let's say we can keep approximately. Yes, very good. Variable tandem repeats, how much it can go? Around 10 to 60. Okay, again this is all approximate value. Okay, 10 to 60 base pairs, and they will be repeated up to 100 times. Okay, up to 100 times repetition can happen. Up to 100 times, and this is a GC rich region. Okay, GC rich region. So mini satellite and micro satellite regions are going to help us identify DNA fingerprinting. But out of STRs and VNTRs, most important one is VNTR. Why? Because imagine 60 base pair repetition has repeated 100 times. Isn't it more easier? Isn't it more valuable? Isn't it more easier to spot these people? Correct? More than something that has repeated only 6 base pairs have repeated 5 times. More than that, VNTR, because the maximum repetition is happening and the longer sequence is getting repeated. Correct? So VNTR we are going to use in your DNA fingerprinting. Okay, clear or not? Any doubts in this? Okay, any doubts in this? Now, what are we going to do, students? The steps for DNA fingerprinting. Let's say, okay, this is Chin2. Okay, let's say this is Min2. This is Min2. From Min2, we are going to take cell. We are going to isolate the DNA. Okay, DNA isolation. Okay, then we will run it on a gel electrophoresis. Before that, you will cleave the DNA. Okay, after cleaving the DNA, you will add it into a vector. It could be a YAC or a BAC, that is bacterial artificial chromosome and yeast artificial chromosome. Okay, with the help of yeast and bacteria. Okay, bacteria, these vectors. Okay, we are going to use these vectors. And after that, what are we going to do, students? After we get, okay, after we have inserted into the vector, now we took this from Min2. Okay, we took this from Min2. Okay, basically I'm roasting myself. Yes. So yes or no, it's like, okay, this is the best version of myself. Now students, from Min2 we took cell. From that DNA isolation we cleaved the DNA, yak and back with the help with this yeast and bacteria. Now we should remember that from Min2 there's definitely going to be maternal strands which are repeated, paternal strands which are repeated. This is going to help us identify from the crime scene what has happened, right? Okay, what has happened. Okay, you want to take it as Champa, no problem. Okay, now students, remember there is going to be overlapping sequences. Okay, there is going to be overlapping sequences. This overlapping sequences cannot be removed manually. Okay, not manually, it cannot be understood or differentiated. So instead of that, we will be using computers to get this overlapping, okay, arranged, overlapping arranged. This is what is happening. This is what one paragraph is given in your NCERT. Now, more than this paragraph, okay, more than this paragraph, what is important is to understand how do we exactly do DNA fingerprinting. That is, look here, this is individual A, this is individual B, and this is the blood sample that we got from the crime scene. Now, if you look at this, students, you will see paternal, maternal, right? Paternal, from father's side I'll get something, from mother's side I'll get something. That will not be similar to someone else. Okay, that will not be similar to someone else. So, what is going to happen here? Paternal, maternal, you have chromosome 7, chromosome 2, chromosome 16. Now, looking at this pattern, you will identify who is the person. How? By running it on a gel electrophoresis, right? Here only I can run it on a gel electrophoresis. I can use here a gel electrophoresis. Okay, after using a gel electrophoresis, I will transfer it to a blotting. Okay, southern blotting I will do. Okay, southern blotting. Now, how do I identify, ma'am? How do I identify? See, look here, students. You will notice that the C, the blood sample C and individual B has lot of similarities. Why? The repeating units are at the same place, and always remember that in your gel electrophoresis, the smaller units will run faster, no, smaller fragments will run faster. So, as you can see, maternal and paternal, you cannot damage, no, maybe other things might be similar, but that 0.1%, that repetitive DNA is very, very important. Now, for you to make it more easier, now look here, students, let's say this is the repeating sequence that I got from the crime scene. Okay, from the crime scene, I understood that this is the DNA sample, and in the DNA sample, this is the, you know, what are the repeating units. Now, using a VNTR probe, what is a VNTR probe? Variable tandem repeats, right? For that, I am using a probe. I'm using what? A probe. This is radioactively labeled. This probe is radioactively labeled. Okay, radioactively labeled. Now, what is going to happen? Let's say this unit is repeated 25 times. Okay, in the person who has committed crime. Okay, in the person who has committed crime. Let's say, take an example, okay, 25 times I have not drawn. Let's say 1, 2, 3, 4, 5, 6, 7. Okay, 7 times it has repeated. Okay, 7 times it has repeated. You tell me who is the criminal? I have used this probe and they have radioactively labeled and given me. Now, you tell me, is Pandu the criminal or Champa the criminal? Pandu or Champa of Schreiser is the criminal. Who among this is criminal? Pandu or Champa? Look at this. Probe means what? A radioactively labeled. Now, this is the sequence that is there. Opposite. Anti-codon we tell, no, same way. Complementary to that is going to be a probe. And that probe, okay, that probe is going to be radioactively labeled. Okay. Yes, Jaswant is the criminal. Good job. Okay. Yes, Jaswant is the criminal, correct. Okay, that is out of context. Now tell me, in Pandu and Champa, who is the criminal? Who has committed the crime? Quickly in the chat box. Just look at it. See, 7 times this has repeated. So I have made, this has repeated and I have made a probe which is radioactively labeled, which is complementary. This probe will go and attach to every sequence which is this. So let's say All these sequence are that. So 1, 2, 3, 4, 5, 6, 7. 7 sequence continuously repetition has happened. So who will become the thing? Pandu will become. Pandu is the criminal here, right? Because in Champa also probe went and attached, but there are only 2 and they are separated. They are not continuous. So here this becomes the criminal. Understood? Yes, Pandu becomes the criminal. How? Because the number of times this is repeated and this complementary strand went and attached to this and they radioactively labeled it. Understood? Clear? So same thing happens here, students. Here also same thing happens. Understand? These and these are the same. How do we know? By using a probe which is complementary to the sequence. Complementary. So repetitive sequence or your satellite DNA is the main important person to understand DNA fingerprinting. That 0.1% will not be same for any of you. Now how many other combinations come? That's what in your NCERT it is written. 1 out of 100. Okay, 0.1%. Okay, 0.01% people are getting, let's say 1% in the entire population is getting affected. That means this will become polymorphism. Polymorphism. Right, 1, 2, 5, 7% is affected, it will become polymorphism. If it is only 3 people or 4 people, it will become mutation. Right, it will become more mutation. Here it will become polymorphism even if 1% of the population is affected. So that is what one line they have given there. Change, no? That comes from genetics. That's what your whole chapter is all about. So students, that's it for today. And we have done our day 3, right? And all of you are in the VIP badge. Okay, all of you in the VIP batch. Those who are feeling bad that, oh my god, ma'am, I did not solve enough questions. Let me tell you, when I'm giving you break, okay, lagging. When I'm giving you break for Baswa sir's class, please solve this. See, principles of inheritance, molecular basis of inheritance. It is there in the description box itself. Okay, click on the description box. Directly, they will tell you to start the test. You can load. Non-student role not allowed. So I will not be allowed. You can attempt. Okay, you can attempt. Clear? Polymorphism is in the entire population. More than 1%. 1, 2, 3, 4, 5% of people are affected by something. And after that, write a test. Okay. And please let me know how the session was, because it was a very long session. So much hard work was put into making this PPT into this concised. Okay. And let me tell you, students, up to Jan 5th, all the subscriptions have been extended. So you can take All India Test Series plus the batch. If you are very smart, you will go for this. Otherwise, you can just go for All India Test Series at 13999. Okay, this will be getting 22 tests along with 30 OMR sheet delivered at your home. And let me tell you real examples of my students who last time went and did OMR sheet wrong. Because of that, they did not crack NEET and now they are a dropper. And many of them have taken alternatives. So they did not know how to solve OMR. And first time they opened OMR, all the bubbles went wrong. So because of that, I would request all of you, when you're practicing your All India Test Series, please do it in an OMR sheet. It's very important. Even when we used to do for Olympiad and all, OMR sheets used to be very difficult to, understand itself. Name only we used to do wrong. So please do that, and detailed solutions for every questions you are going to get. Okay, all the links are in the description. You can go and check out. Before that, most importantly, oh, I'm so sorry. What happened? Yeah, most importantly, this one, solve the test. Okay, I hope all of you understood. Last we had to rush up a little. Okay, but I hope everyone understood everything. Now students, sir will be joining now, and sir will tell you about the break. Okay, Baswa sir will tell you about the break. Clear? Yes, don't get scared, just practice. Everything is going to happen well. Okay, 1 second, I'll just inform sir, and sir will be in the class. Okay, 1 second. Yes, I have told sir will join. Okay. Thank you so much. Please don't fail to drop a comment, student. It will take you 2-3 minutes to drop a comment. Okay. And I hope all of you enjoy the class and you understood every concept. Any concept you have a doubt, you can just search with my name and you can continue. Students, let's say, I think break, it's 9:31. Let's say 9:45. Because more than that, it will be too late, students. 9:45. Please go now itself. Run. Okay. This live will happen. Okay. Yeah, I will share the notes, Sukanya.
Sound check, students. Quickly let me know in the chat right now if I'm audible and visible to each and every single one of you. Quickly let me know in the chat if I'm audible and visible. Yes. Quickly let me know in the chat right now if I'm audible and visible to each and every single one of you. Quickly let me know, students. And also, welcome back to the channel. Every single one of you, welcome back to the channel. This is Basraj, your biology master teacher. Welcome, welcome, welcome back. Now, students, quickly tell me in the chat how confident you are in the chapter molecular base of inheritance. Rate your preparation out of 5 right now. Everyone, how is your preparation out of 5 in the molecular base of inheritance? Because Gopika ma'am did an amazing job. I was there throughout the lecture. I was there to the lecture. So she has done a fabulous, amazing job. So quickly, let me know in the chat right now. How was it? All clear? Amazing? Yes, all of you, quickly let me know in the chat. Amazingly good? Yes, amazingly good. Now, students, the only thing which might be lacking right now in your preparation, that is most probably question practice. All of you agree with me? All of you agree with me, right? 1 second, students, give me a minute. 1 second. So there's something wrong with the camera. Give me a minute, students. Yes. So only thing which is going to be missing right now is the entire question practice. If the question practice is good, you are good to go. Okay. Now, with that same energy and the same enthusiasm and the same dedication, can we start right now the principles of inheritance and variation? Can we start? All of you in the chat quickly. A quick fire emoji in the chat to let me know, yes, you are ready for the next big hurdle in your preparation. Because let me tell you right now, reproduction chapter is over. Human health and disease, the entire unit is over. And if you finish this unit, the entire molecular basis unit, you will be covering almost 80% of your class 12th biology in just 3 lectures. And this is the most highest weightage out there. And let me tell you, right, if you master this, if you're able to solve every single question from this, you are good to go. Okay. Now the first question to all of you. Okay, the first question to all of you students, tell me, what do you call a branch of biology where we study about animals? What do you call the branch of biology, right? Sir, you are the right person to clarify my doubt. Difference between ma'am and miss. Don't tell difference is the spelling or the posting. Okay, wait. Miss is generally referred to person who is unmarried, and ma'am is referred to as the higher position. Ma'am and sir are the respected positions, and miss is the term used for people who are not married, the females who are not married. It's a old technicality. Okay. Now tell me, students, what do you call a branch of biology where we talk about the entire animal? Zoology. You'll tell me zoology. Now, what do you call the branch of biology where we study about plants? All of you, what do we call that? The branch of biology which deals with plants, we call it botany. What do you call the branch of biology that deals with microorganisms? Microbiology. Then what do you call the branch of biology which deals with inheritance and variation? The branch of biology that deals with inheritance and variation, that is called as genetics. That is called as genetics, and that is this chapter. So this chapter, the principles of inheritance, it belongs to an entire branch of biology called as genetics. Entire chapter belongs to the branch of biology called as genetics. Students, let me tell you, genetics is the most important branch. If you want to understand the proper functioning of cell biology, microbiology, or it could be molecular biology, cancer biology, this is the foundation. This right here is the foundation of any advanced biology. So students, let me tell you, are you ready right now? Are you ready? Because let me ask you a very simple question, student. Let me ask you a very simple question. Has any one of you ever heard in your life that your smile is like your father, or your eyes is like your mother, or your nose is like your father? Have you ever heard this term from relatives or friends or anyone in your life? You might have heard. You're like, yes, Mice are like my mother size. Okay, you might have heard about it. Now that is called as like begets like. What do I mean by that? There is some character, not trait, some character is passed down from parents to us. Parents to us, that is called as inheritance. That is called as inheritance, as transfer of a very specific character. It could be like, nice hair, nice face, nice nose, height, weight, anything. Okay. Now students, how many of you have siblings in your house, like brother or sister? You might have brother or sister, right? Now, both your brother and sister, we have the same parent. Right? We have the same parent. And when we have the same parent, right, even though we have the same parent, do we look like each other completely? Are we completely identical? Are we completely identical to our siblings? No. There are changes are there. Now, I told you, there are certain characters which we obtain from parents. Nose, hair, eye color. Okay. But are we the exact copy of them, students? Tell me. Are we the exact copy of our parents? No. There are small changes. Yes or no? These changes are called as, what do you call them? These changes are called as variations. They are called as variations. So that is the chapter name, inheritance and variation. So inheritance is what, students? Tell me in the chat quickly, all of you. Inheritance is a process by which, it is a process by which characters, right, characters are passed from parents to the offspring. That is called as inheritance. That is called as inheritance. Right? You might have observed in nature. Right? In nature, you might have observed an elephant will give birth to an elephant. A tiger will give birth to a small cub. A cat will give birth to a kitten, or a human will give birth to a human. Does it ever happen in nature? Does it ever happen in nature that one cat is giving birth to a giraffe? Hippopotamus is giving birth to a dog? It doesn't happen. It doesn't happen at all in nature. Like begets like. It's called as like begets like. From parent to offspring, some information is transferred. Okay. Some information is transferred, but in that information, there are certain changes. There are certain changes in that variation. I can call this change, or I can call it as differences. I can call it as differences, students. Differences, right? Differences by which there is a change in the offsprings. That is called as variation. Now, all of you have some idea right now what is inheritance and what is variation. Inheritance is transfer of characters. When the transfer is happening, there are small changes, and the changes is called as variation. Now, a question might arise in everyone's mind. What is the question? Why the change? Can anyone tell me in the chat? Try using the brain cells, remaining brain cells, and tell me why are these variations occurring, or more importantly, when are these variations occurring? The hint is on the board. The hint is on the board. Can anyone tell me where does this variation actually occur? Like, in what stage of human evolution or human body or any animal of matter of fact, when does this variation occur? The answer is right here. The answer is right here. All of this variation happens in sexually reproducing animals. In sexual reproduction. In sexual reproduction, to be precise, there is a process by which gametes are produced. A process by which gametes are produced. I am talking about gametogenesis and oogenesis. In gametogenesis and oogenesis, there is a step by which a diploid cell can give rise to a haploid cell. Can anyone tell me that? What is that? This diploid to haploid. The process which I am talking about is none other than meiosis. So what exactly is happening in meiosis? In meiosis 1, to be specified, in specific meiosis 1, there is a phase called as, can anyone tell me in meiosis 1 at which stage the crossing over takes place? Anyone in my any of my student? Where does the crossing over takes place? A stage in prophase 1. A stage in prophase 1 called as Pachytene stage. A stage in meiosis 1, prophase 1 called as Pachytene stage, where crossing over happens. Because of that crossing over, that leads to entire variations. Entire variations. Beautiful concept, right? Did we just connect one chapter to the another chapter? Now all of you realize the importance of studying cell cycle now. So because in cell cycle we learned that in prophase 1 at Pachytene stage there is crossing over, recombination nodules, there is recombinase enzyme, all that is connected here. So because of that variation, gamete formation, sexual reproduction and variation. Beautiful concept. How many of you understood? Raise your hands. If you understood this small concept, it will take you a long way in life. Let me tell you, because these small concepts connect together and then give you a bigger picture. How many of you understood? Green hearts in the chat. Green hearts in the chat if you understood the small concept. Okay. So there is haploid sperm, female, an egg cell, they fuse together, diploid zygote. There is variation here. Variations are there. And when once the variation takes place, entire human is born. Entire human is born. Okay. Now students, I'll ask you one more thing. How many of you know this beautiful creature? How many of you know the name of this? Anyone? Anyone knows which type of cow this is? Anyone? Very amazing cow. This is very famous. Celebrity among celebrity. Celebrity cow. It's a very celebrity cow, students. Anyone knows who is this person? This person is from Punjab. This person right here is from Punjab. Anyone knows? Anyone? Not Kasturi. It's called as Sahiwal. Okay. It is called as Sahiwal cow. Okay. Now, what is so special about this? Now, students, I'll give you. Okay. Before I make you understand this, can I give you a small example which I really love? Like, I love this example because it makes more sense to me because I'm a botanist. Okay. So tell me, how many of you love mangoes? Like, I love mango. Mango season is coming, April, May, right? Now there are two types of mangoes, for example. Right? There are two types of mangoes, for example, two types of mangoes. Understand very simple manner first of all. Okay, two types of mangoes. One mango is extremely sweet but very tiny. I repeat again. One mango is extremely sweet but very small mango. One mango is very large but it is not sweet at all. Not sweet at all. So what do? You take the small sweet mango and large not so sweet mango. You take these two mangoes and you cross them. That is called as selective hybridization. I repeat again, heavy terms. I'm telling you heavy terms. Very heavy term. Selective hybridization. What you're doing right now here is you're taking one character here which you like, one character here which you like, and you're making a combination out of it. So the byproduct or the offspring is going to be large mango and also very sweet mango. The chances of that is very slim again because there can be a mango. Imagine you get a mango which is very small and not sweet also. There is a chance you might get a very small mango which is not sweet also. But it is a luck. It is a trial and error method. But all of you get the concept. All of you get the concept, right? That is called as selective hybridization. Same thing is done with the help of Sahiwal cow. Okay. So not just us, our ancestors also very smart people. They completely use the method of hybridization, right? They use hybridization. They knew all this variation happens because of sexual reproduction. They used it to do selective hybridization and to produce Sahiwal cows in Punjab. That is artificial selection. Artificial selection. Point clear, students? How beautiful the example is, right? Mango example is always like the most easy way to remember things. Okay. Now, students, before we go to the entire genetics, monohybrid, dihybrid, polygenetic, what is pleiotropy, diseases and everything, can I teach you some genetic terminologies? If you learn these terminologies, you will understand the chapter in a much more better manner. Okay, all of you agree? So let's start with simple examples, simple terminologies, which all of you will understand and we'll go ahead. Okay, now let me tell you, If you have no idea whatsoever about this chapter, like zero knowledge, let me tell you, after my class, you will for sure obtain a lot of information, lot of knowledge, and you can solve a lot of questions. That is my guarantee. That is my deal from my side. Okay, zero knowledge, sit in my class, doesn't matter. Okay, now students, let's start with certain genetic terminologies. Okay, now if I ask you one simple question, all of you know what is a gene? All of you know what is a gene? You might have heard about something called as gene, not Jean Grey. Who is gene? Right? What is this gene? Or where is exactly this location of a gene? You might say, sir, gene is present inside a body, inside somewhere in the brain, somewhere in the cheek cell, human cell. Where is it? Like, where do you get the gene? Let me tell you. Where do you exactly find a gene and what is a gene? Understand? Very beautiful. For example, this is a chromosome. All of you know chromosome? Yes. Simple chromosome, all of you know. All of you know chromosome. Now students, chromosome, this is called as a centromere, two chromatids. Now students, if I unwind a chromosome, what do I get? If I unwind a chromosome, I get a long thread. The long thread is called as chromatin. Long thread like structure called as chromatin. I get a chromatin. Okay. Now students, if I focus on the chromatin, you will see that DNA is coiled around certain type of histone protein. It is coiled around histone protein. DNA is coiled around histone protein. And I will call this entire structure as a nucleosome. You might have heard, students, today's lecture only. You might have heard. Nucleosome. Now students, if I take out a single piece of DNA from it, now from here, if I focus even further, let's say I get a single piece of DNA. This is a single piece of DNA right here, single piece of DNA. On this single piece of DNA, there is a small segment here, smallest segment of DNA. I repeat again students, listen to me. Gene is the smallest segment of DNA. So please remember this entire flowchart, because if you want to understand chromosomal theory of inheritance, this is the basics here. So if you want to know where is the gene or where is the DNA present, genes are present on the DNA. DNA is part of chromosome. So can I technically tell genes are present on the chromosome? All of you agree with me? Yes or no? Tell me. So can I tell right now, on this chromosome, here I have a gene which codes for, let's say, eye color. This codes for eye color. I have one more gene right here which codes for hair color. One more gene present right here which codes for height. All of you agree with me? Yes. Now can I tell this each gene is coding for a character? Each gene is coding for a character. All of you agree? Now we come to one more question, right? Now one more question arises. I told you this is a one second. This is gene here. This right here is chromosome. Okay, chromosome. Now this entire one gene, this is a gene present on the chromosome is coding for a character. It is coding for a character. Now the question arises, what is a character now? The question arises, what is a character? Understand. Okay. Understand. What exactly is a character now? It's very simple. Character, it is a feature of an individual. It is a feature of an individual. Feature of an individual. Let's take a handsome plant. Yes, plants are also very handsome and cute and nice. Let's take flower color. Flower color can be a feature. Okay, let's take humans. Eye color. Let's take plant height. All of these become the what? They are called as a character. Now students, let me tell you, this particular flower color can be, let's say, red, it can be white, or it can be pink. Yes or no? Okay. Tell me, what is your favorite eye color? Tell me in the chat quickly. Favorite eye color? The eye color can be, let's say, like a Sharingan, which is red color, or let's call it as brown color, which is very common. Not so common actually. Hazel is very rare. Or it can be green. All the blue eyes crying in the corner. Okay. Now plant height. Plant height can be tall or dwarf. Yes or no? So students, understand. This is called as a character. Flower color, eye color, plant height. All of these becomes character. All of these, when I am mentioning a variety or a variant of a character, all of these red, white, pink, brown, red, green, tall, dwarf, all of these become trait. All of these become trait now. Now that is a beautiful concept of a character and a trait. I hope all of you understood. Okay. Question, all of you understood? Okay. All of you understood? Beautiful. Now the next question is, all of you understood what is a gene? All of you understood what is the gene right here? What is the gene right here? All of you understood. What is the gene? The next question arises, what is an allele? A question arises, what is an allele? Like, what is an allele? Okay, what is exactly an allele now? Let me tell you very specifically, students, because if you by heart the allele concept, it is very easy to by heart. But let me teach you with the help of a diagram today. Ready? Okay, see this. Now, for example, here is a chromosome. All of you know all the chromosomes are present in pair. All of you are aware? All the chromosomes are present in pair. Yes. Let's say this is a pair of chromosomes. Pair of chromosomes present right here. Okay, present right here. It's a homologous chromosome, one from mother, one from father, which you inherit in your body. Okay. Now, for example, on this location, I have a gene here. On this chromosome, I have a gene here. Now, this gene right here has an alternative form on the other chromosome also. That becomes an allele. See, allele cannot be right here itself. No, not present here. Allele is present right here for this. Let's say I have a gene here. Allele is present somewhere here. The beautiful part is both the alleles, right, which is called as a alternative form of a gene, they will share the same loci. And same character. If this is coding, if this right here is coding for, let's say, flower color, this also will code for flower color. I am telling, it codes for a specific character, not trait. Trait can change. This can code for tallness, this can code for dwarfness, not trait. I am telling you, it codes for same character, flower color. Let's say I have one gene here, the alternative allele is here. This is coding for eye color. Okay, this also will code for eye color. It can be different trait, but character is going to remain the same. All of you understood what is the character? This right here is the character. Where did it go? Feature of an individual. These are called as character. So it can code for same character. It can code for same character, right? But it does not code for same trait. Understood all of you? Understood? Basics. See here. Both is coding for height. That is a character. Seed shape. See, present on the same loci of homologous chromosome. Pod color here and here. So this is called as an alleles. Allele is an alternative form of a gene which are present on the same loci or homologous chromosomes, and they code for same character. Students, let me tell you all these small concepts, right? We usually generally mug up all this. We generally mug up in biology rather than understanding it. That is where mistakes arises. Okay, so I hope all of you are able to follow now. All of you are able to understand what is the meaning of allele. Or if all of you understood, fire emoji in the chat. Let me know if you understood this technicality, because we can mug up all this. Very simple to mug up. If you want to mug up, you can just mug up this line. This is the only mug up thing. But if you want to understand, this is what you need to understand. This line here. They will have same loci. Same loci on the location of homologous chromosome and same character also. Same character. Yes. So apart from that, students, there is something called as a phenotype and a genotype. All of you are aware. Phenotype and genotype. Yes or no? Phenotype is what, students? Phenotype is basically the morphological characters. Morphological observable characteristics, like how I look from the outside. This is my morphology. That is called as a phenotype. Then what is a genotype? Genotype is basically the genetic makeup. It is the genetic makeup. Not the other makeup, but what our bodies are, which genes our body is made up of. That is called as a genotype. Okay. Now, next small concept which I have to teach you is homozygous and heterozygous. What is the meaning of homozygous versus heterozygous? All of you understand? Very simply understand, simple. Like very simple it is. Right now I told you what is an allele. Allele is an alternative form of a gene. Okay. Let's go back to this diagram. Okay, let's go back to this diagram right here. Okay. Alternative form of a gene. Now here, students, if the two alleles, this and this, not chromosome, if the two alleles are identical, when I say identical, I mean, students, listen to me very carefully. Listen to me very carefully, all of you. When I say identical, I mean capital T here and capital T here. Then I will call them homozygous condition. Both are capital T, capital T. That is called as homozygous condition. Now, what if the two alleles are different? For example, here we have small t and here I have a capital T. Capital T, small t. Then I will call it heterozygous condition. Okay. So, homozygous means what? The two alleles are same to same. All of you remember that Cadbury ad, Ramesh and Suresh. Right. Homozygous are basically twins which look alike. Heterozygous is which look different from each other. The two alleles look different. The two alleles look alike. Remember that 5-star ad? Eat 5-star and do nothing. The two Ramesh and Suresh, they don't look like each other. So they are a heterozygous condition. If the two twins look alike, that is called as homozygous condition. Done. Homozygous condition. Now, students, one more small concept. Let's take, I have a heterozygous condition here. Heterozygous condition. Now, in this heterozygous condition, one allele, listen to me very carefully. Out of these two alleles, one allele is dominant allele. Now, what do I mean by dominant alleles? Okay. The dominant allele is an allele which does not care if anyone is around. It will express itself. I'm like, I will keep shouting. Backbenchers, no. They don't care if anyone is there in the classroom. They don't care nothing. They keep shouting. Blah, blah, blah. Keep shouting. That is dominant allele. This is dominant. This will express, doesn't matter if there is recessive, doesn't matter if there is no one, this will keep on expressing. Now, what is a recessive allele? What is a recessive allele, students? Recessive. Recessive allele is an allele which will express, it can express only when the dominant allele is missing. So only when students, only when dominant allele is missing, only that time recessive allele expressed. Like for front benchers, do you ever hear making them noise in the class? No. They will only talk when backbenchers are absent. When they are not making sound, only then their voice will be audible in the class. Nice, they'll talk, no? Nice, small, they'll talk. So that recessive allele, only that time the expression will come out. Otherwise, they're very silent. Chup chap, write notes, go home, aram se. Okay, tell me students, which bencher you are in? If you are in my class, backbencher only you are. Okay. Okay. Students, with that, terminologies is also over. All of you are understanding? All of you following with me? All of you following? See, this is how genetics is. If you miss out any of the small detail in the starting, the chapter becomes like a burden later on. But it is a very easy chapter, what to do. Okay. Now, let's go to the next concept called as your Graham-Mendel concept. All of you ready? Now students, tell me, what names already board? Students, all of you namaskaravan. All of you quickly. All of you know the rule in my class. If any scientist is coming, one namaskar you should give. Namaskaram. Why? Because if this person was not here, two things would happen. If this person did not exist, this chapter would be deleted from your syllabus. This chapter would have been deleted from your syllabus, and also there would be no advancements or very little advancements in genetics. If very little advancement means what? There would be no proper research, there would be no proper diagnostics, there would be no proper medicines in the world. So if you wanted to be a medical doctor also, very little chances. So namaskara all of you quickly. Okay. Now students, with that we come to Graham-Mendel concept. Now students, it is not given you in NCERT, but I will tell you when he was born, when he is dying also, because small thing we can remember, contribute, right? He has done so much for us. Why can't we just remember how much we have, when he is born at least, right? So Graham-Mendel was born in students July 22nd. You don't have to remember this, but write down somewhere in your notebook. Someday you might thank him for it or something. Who knows? Okay, 1822. So July 22nd, if you share a birthday with him, amazing, good for you. Okay, right? So July 22nd, 2022, he was born. Okay, and let me tell you students, a small story of never giving up, right? See, all of you are right now NEET aspirants. The most important thing which you need in your life right now is consistency and determination to fight against all the odds. Like, yes, syllabus is there, too much of syllabus to study, but all that is aside, the main thing which you need to have the mental strength to move forward. All of you agree with that? Because sometimes what happens, you do a mock test, you get 500 marks, 400 marks, you feel demotivated. But let me tell you students, let me tell you, do not be demotivated, because let me tell you a small story. Okay, Graham-Mendel, right? Graham-Mendel was a priest. He wanted to be a priest. Okay, initially he wanted to be a mathematician. Yes, not biologist. He wanted to be a mathematician. But guess what? Even a great person, even the greatest of scientists like Graham-Mendel, right here, he failed. Yes, not only anyone. Lot of great things only come after a good comeback, right? So Graham-Mendel failed the exam to become a teacher in mathematics in general. He failed the exam. But did he, after failing, did he keep quiet? I'm like, I failed in life, I will not do anything in life, I will just go and sit in my in a church and do nothing. No. He's like, once he hit the rock bottom, once he hit the failure, he realized my skill, my interest is somewhere else. Later on, what did he do was, he tried observing all the different plants around him. Imagine students, we see so many things on daily basis, but how many of you truly say that yes, this is different? Very few people tell that. He was one of those scientists who was completely ahead of his time, and he noticed the changes in the pea plant. And let me tell you students, let me tell you students, he worked on a plant called as Pisum sativum. Pisum sativum, right? Pisum sativum is called as the pea plant. It is called as the pea plant. Now you might think he worked for like what, like let's say 1 year, 2 year, 3 years. Like 3-year drop some people take, 4 years? No. This person right here, students, this person right here, he worked from 1856 to 1863, that is 7 long years on his work. 7 long years. 7 long years he worked. 7 long years. In that 7 long years, he did very large sample size. He took lot of plants. He took a lot of plants or different varieties, different small changes, and studied them, noted them. What do you think? Yours is easy or difficult? Look at this man's perseverance. Look at the determination he had. Even after the failure, he never gave up. Right? 7 long years he worked on it. He worked on 7 pair of contrasting traits. 7 pair of contrasting traits. All of you know what is a trait, right? One character can have 2 traits. So he worked on 7 pair, means 14 different traits. 14 traits. 7 pair means 14 traits. 14 traits. And he selected. He didn't randomly select any plant. He randomly did not go and take any plant. Not 14, right? 14 pairs of contrasting traits, right? Rama, see this, right? Let me show you the diagram only. Where is the diagram? Students, tell me. 1, 2, 3, 4, 5, 6, 7. So, how many characters? Not 14 characters. He worked on 7 characters. 7 characters. How many traits? 1, 2, 3, 4, 5, 6, 7. 7 dominant, 7 recessive. So, he worked on 14 traits and 7 characters. 7 characters. So, 14 traits and 7 characters he worked on. Let me write for you, you forget. 7 character and 14 traits. Now he didn't take any random plants. He did not take any random plants, students. No. He did not take any random plants from anywhere, like one plant is growing, I'll take it. No. He selected something called as pure breed lines. He selected something called as 14 pure breed lines. Now, what is this pure breed lines? Let me tell you. Pure breed lines are homozygous condition. They are basically homozygous condition plants. Homozygous condition plants which has undergone repeated self-pollination. Repeated self-pollination. Repeated self-pollination. For example, listen to me very carefully. There is, let's say, he might have taken, let's say, so round, capital R, homozygous, small r, capital Y, small y, capital V, small v, capital F, small f. Yes or no? Capital G, small g. So pure lines are all of them homozygous. See, they can be dominant or recessive, but they have to be homozygous condition. So that is called as 14 different pure lines he took. This also can be capital A, small a, capital T, small t. That is called as a pure line, homozygous condition. Okay, homozygous condition. Now, apart from this, students, let me tell you, since he wanted to study maths, he started maths in biology. So, he used statistics. He used stats and also large sample size. Okay, large sample size. Clear, all of you? Large sample size. Okay, so all homozygous condition he took. Okay, small r, small y, right? So 14, it's given here also. Let me write for you. How many characters, students? 7 characters. 14 traits. 14 pure lines. 14 pure lines. Okay, so remember this much. And out of this, students, out of this, which one you need to remember? You need to mostly remember the dominant one. All dominant. Okay, here. All dominant you should remember. There is a small trick here. How many of you remember the trick? Not my trick, let me tell you. I picked from internet somewhere. Not mine. I copied it from somewhere. It is called as gravity. This explains all the dominant. For example, G, green is the pod color dominant. Round is the seed shape dominant. A is axial position dominant. V, that is going to be violet flower color dominant. I is inflated dominant. T is tall, very tall dominant, and Y is going to be yellow seed color, that is dominant. So all dominant traits can be explained with the help of gravity. Not mine, somewhere internet. Okay, you have to be very honest in life. Okay, so that's where I picked it up from. Okay, gravity. Okay, now you might ask, why the pea plant? That is my dog. Okay, his little cuckoo. Okay, so why did he take pea plants? Like, why did he only take pea plants? Is there any reason for taking pea plants? Yes, there is. So are there any advantages? Yes, there are advantages. The first advantage might be clear to you right now. Did we observe lot of different type of traits? Different type of traits? Yes or no? Different type of traits. So many different type of traits we observed. Yes. So this plant clearly shows a large variety of traits, and all of them can be very easily visually seen. They can be seen visually. Okay. See here. Pure varieties can be easily available. Simple hybridization. Okay. Large number of seed produced per plant. Obviously, very advantageous. Many seeds, take the seeds, easy to grow. Okay. Easiest detectable contrasting traits, characters, sorry, I told you. You can see tall, dwarf can be easily be seen. Okay. Can be crossed manually. Short lifespan, reproduces in 3 months, can be grown easily. So what do you need more in life? You have a plant. All the things are very easy and start to do. But students, let me tell you one more thing. Right now itself. Listen to me very carefully. Okay. Mendel was very lucky. Because this plant is very special. Why? This plant is very special because I will explanation and you will understand better when I teach you linkage and recombination. Because when you do linkage and recombination, you will understand the concept of different positions of genes on a chromosome better. Very difficult to understand, don't worry. I will explain why Mendel was lucky also. Mendel was lucky because because he took pea plant. Because let me tell you, Mendel did the same work on hawkweed and beans. And let me tell you, he did not get the same result. Mendel did the same hybridization experiment, monohybrid, dihybrid on hawkweed and bean plants. He did not get the same result. So Mendel was actually lucky. Why? Because all of these characters, all the 7 different characters, right? Every single character was present on 4 different chromosomes. You will understand this small concept later on better. Why? Just because they are present in different chromosomes, this 1, 4, 5, 7, different chromosomes, why was he lucky? So wait till that concept to come, and then I'll teach you this again. Remind me, students, if I forget, remind me one more time why Mendel was lucky. Okay, how does him being lucky corresponds to chromosome number? Okay, remember. Okay, remind me. Now students, what do we understand from Mendel? Mendel gave 3 different laws. Law of dominance, law of segregation, law of independent assortment. Okay, listen to me very carefully right now. These 2 laws will arrive from the monohybrid cross. Law of independence from dihybrid cross. So monohybrid cross, dihybrid cross. Simple thing which you need to know. Okay, that is law of dominance and law of segregation can be obtained from monohybrid, while law of independent assortment can be obtained from dihybrid cross. Clear, all of you? Clear? So with that, the first part of chapter is over. It is over now. Done. Students, till now, any doubt, anyone, quickly ask me. Or is the session going very smoothly now? Till now, any doubt? Till now, students, any doubt? Any doubt, anyone? If there is no doubt, all of you drink some water. Water break. Yes. Now, if there is no doubt, students, can we start right now? Something called as inheritance of one gene, also called as monohybrid cross. Can we start? Okay. Questions are there, don't worry. Questions are coming up. Questions are there because this is the easy part, right? Not many questions till here. Okay. So, if this is very clear till now, very smooth like butter till now, because next remaining part is also going to be very smooth. Okay. So, can we start something called as monohybrid cross? Can we start, students? Can we start? Before we start monohybrid cross, let me tell you what is the Mendel's work called as. This entire Mendel's work is called as hybridization work. This entire Mendel's work is called as hybridization work, and it happens in three main steps. It happens in three main steps. Does anyone remember, students? What is the first step? First step is called as selection of parents. Selection of parents. Always remember, selection of parents is also always has to be what? They have to be pure lines. Pure lines. Okay. Second step is crossing of parents. Crossing of parents to obtain F1. Third step is called as selfing of F1. Selfing of F1. These are three main steps in hybridization. Let's go by it one by one and you will understand very easily the entire chapter. Okay. Now, the first one is called as inheritance of one gene. Ready, all of you? Ready? Okay. Let's draw the parents first. Parents here. Parents. Let's take parents. For example, let's say I have here homozygous dominant tall plant. Dominant tall. Capital T, capital T. Here I have recessive dwarf, right? Small t, small t. Okay. Both of them are homozygous condition. Can you see? Both of them are homozygous in nature. Okay. Both are homozygous in nature. Homozygous. Selection of parents done. Selection of parents done. Okay. Next is the gamete formation. Gametes formation. Okay. Here what do we have? Capital T. Here what do we have? Small t. Next step is crossing over and we get F1. We get F1. Okay. What do we get? Capital T, small t. This right here is the F1 progeny. F1 is also called as first filial generation. It is called as first filial generation. Okay. Now students, this is tall, this is dwarf. The F1 is going to be what, students, tell me? F1 is also going to be tall. And this right here is heterozygous dominant. Heterozygous dominant. See, heterozygous dominant. Next step is called as selfing of F1. It is called as selfing of F1. So what do? We take capital T small t into capital T small t. Yes or no? Now to understand the next step, we need to do something called as Punnett square. Can we start Punnett square? Let's do Punnett square here. Punnett square. So what do we have here? Capital T, small t. Yes or no? Capital T, small t. Capital T, small t. What do we get? Capital T, capital T, capital T, small t, and small t, small t. Tell me, what is the phenotype here? This one looks tall. Okay, this one looks tall. This one also looks tall because it is dominant allele is expressing. Okay, tall. This is dwarf. This is dwarf. That is right here. This right here is called as the F2 generation. Second filial generation. Second filial generation. Now, what they can ask you, maximum they can ask you is, what is the phenotypic ratio and what is the genotypic ratio? What is the phenotypic ratio and what is genotypic ratio? Genotypic ratio. What is the phenotypic ratio, students? Tell me. Tall, 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 3 tall and 1 dwarf, 3:1 ratio, 3:1 ratio. What is genotypic ratio here? Genotypic ratio is going to be homozygous dominant, 1. 2 are heterozygous dominant, capital T small t, heterozygous dominant are 2. And 1 is homozygous recessive, just like the parent. So genotypic ratio is going to be 1:2:1, and that is all is monohybrid cross. Done? Done? Yes? Rama will come. What is back cross? What is law of segregation? We'll come to it. Okay. So till now, any doubt till here? If there is no doubt, let's go to the next concept. It's very simple till here. Okay. Now, what we did right now is something called as Punnett square. This right here is called as a Punnett square. It is given by C. Punnett. Ronald C. Punnett. He's a nice, handsome person. Namaskara, all of you. Quickly give it off. Namaskara, one quick namaskara. Okay. This right here is given your NCRT. Tall dwarf, selfing, 3:1 ratio, 1:2:1. That is always the monohybrid. One small question here. Quickly solve this question, all of you. Quickly solve the question, all of you. Quickly solve the question, all of you. All of you. Quickly solve the question. Mendel studied 7 pair of contrasting traits in pea plant and proposed a law of inheritance. Yes, he did. 7 characters examined by Mendel in his experiment on pea plants were seed shape, color, flower color, pod shape and flower color, flower position and stem height. True. Both are correct. Statement 1 and statement 2 both are correct. Both are correct. So you can get such easy questions. Okay. You can get such easy questions here. I hope it is visible. It is not visible. Don't worry. I will give you the entire PDF. You can solve it later on also. Telegram channel, it will be available. Okay, entire thing. Don't worry. Okay. I hope this is visible to all of you. A cross between tall plant was made, resulting in offsprings of tall and dwarf plants into 3:1 ratio. The genotype of both parents are. The question is very simple, but you might ask, sir, is there any difficulty here? The difficulty which students might arise here is if you don't read the question properly. If you read the question properly, here it says a cross between tall plants. That is the first clue. So can I remove this because here we have small t, small t, small t, small t. I removed both of them. Now here we have both homozygous tall. If it is homozygous tall, we can never get 3:1 ratio. If you want 3:1 ratio, you need to have one recessive character. So here is the answer. Capital T, small t, capital T, small t. This basically right here is selfing of F1. Yes or no? That basically right there is selfing of F1. Yes, it is selfing of F1. Okay. Okay. Students, this is not proper. I think it's not visible very well. How much of it is focus? Okay. A heterozygous pea plant with violet flowers was crossed with homozygous pea plant with white flower. Okay. Heterozygous pea plant with violet flower. Okay. Heterozygous pea plant. Capital V, small v. Heterozygous pea plant was crossed with homozygous pea plant white. Small v, small v. Okay. Violet is dominant. Which of the following represents the expected combination among the 40 progenies formed? Very simple. Let's do it one more time, students. This right here is the one parent. So what are the gametes do we get here? Capital V, small v. What do we get here? A single gamete, small v. Do a simple cross. Capital V, small v and small v. What do we get? Capital V, small v, small v, small v. So can I tell out of the offsprings between heterozygous dominant and homozygous recessive, we get 50% is violet, 50% is white. Yes or no? 50% is white, 50% is violet, 50 to 50 ratio. So can I tell if there are 40 progenies, 20 are going to be violet and 20 are going to be white in that. Okay, 50-50 ratio, 50-50 ratio. Okay. Next question. The production of gametes by parents, the formation of zygote, the F1 and F2 plants can be understood using Punnett square. This is NEET 2021 question. Okay. This right here is NEET 2023 question. 23. This is NEET 2024 question. So you can get such questions. They will never ask you easy questions here. All the questions which they ask you is all the questions which make you think a little. Okay. So I hope all of you are able to solve the questions. If you are able to solve, let's go to the next question that is called as backcross. Ready, all of you? Ready? Can we start? What is backcross? Now understand, all of you, understand. Very simple. If you understand backcross, you can understand even test cross also very easily. Okay, very simple, very easy to solve. Okay, understand, all of you. Backcross can be of two types. Backcross can be of two types. Okay. Backcross can be of two types. In the first case, I can take F1 and cross it with dominant parents. And the second one is I can take F1 and cross it with recessive parents. Okay. When I take the F1 and cross it with dominant parents, such a cross is called as outcross. It is called as outcross. But when I take F1 and cross it with recessive parents, that becomes a test cross. Outcross is not given in your syllabus. What you need to focus right now here is only on the test cross. Ready, all of you? Ready? Very simple, very easy. Okay. Let me tell you why we are doing it first of all. Why do we do the test cross? Okay. And what exactly happens? Okay. Listen to me. It is a cross between right, dominant phenotype with recessive parents. And this dominant phenotype right here is F1. So we take F1 dominant phenotype and we cross it with recessive parents. Now the question arises, why? Why are we doing it? Okay, why are we even doing it to the entire thing? Understand. We are doing this entire thing to find the, right, we are doing it to find or know the genotype of F1. This F1 is here, right? We know it is a dominant phenotype. We know this one is a dominant phenotype. Okay, I'll give you a simple example. If you go back to monohybrid, right here, when I look at this tall plant, this is a tall, can I tell both of them are dominant phenotype? All of you agree with me? This tall is a dominant phenotype. This tall is also dominant phenotype. But this dominant phenotype can have two type of genotype. Either it can be capital T small t or it can be capital T capital T. All of you agree now? So what do we do in test cross? We do in test crosses, we find this genotype. We find this genotype in test cross. We look at the dominant phenotype, yes, it is dominant phenotype. But it can be capital T small t or it can be capital T. Both possibilities are there. Both possibilities are there. It is there. So that is what is found in the test cross, and I will teach you how do we do it exactly. I will teach you how do we do it exactly right now. Listen to me. Here is a diagram given your NCERT. Here is a diagram given your NCERT. Understand first of all what is given to you. Okay, understand. Here they have given you a dominant phenotype. All of you can tell me. All of you tell me which is the dominant phenotype here. All of you tell me which one is the dominant phenotype here. That is F1. Here. Dominant phenotype is given right here. F1. F1. F1 is given here, dominant phenotype. But do we know the genotype? To find the genotype, you have to find, yes, genotype unknown. Genotype unknown is genotype. Now, what can be the genotype here? Two possibilities. The two possibilities is that the unknown genotype can be capital W, or it can be capital W, small w. All of you agree with me? These two are the possible genotype, because capital W also it should be capital V. I don't know why they are given capital W. Technically it is supposed to be V. Ignore that technicality, ignore that mistake first of all. This capital W can be violet. Capital W, small w can also be violet. Right? Dominant, capital W is dominant. Just like how capital T is tall, capital T small t is also tall. So both of them codes for dominant phenotype. So what we do right now is we take this F1 and we cross it with recessive parents. Cross it with recessive parents. Now, the question is, how do we do it? How do we find? How do we find the genotype, unknown genotype? Very simple. We find the unknown. Listen to me very carefully, all of you. This line, if you understand right, this concept will become clear to all of you. Listen to me very carefully. To find the unknown genotype, I'll repeat again. To find the unknown genotype of F1, we need to look at the phenotype of F2. We need to look at the phenotype of F2 to find the genotype of F1. I'll write it somewhere. To find genotype of F1, we need to look at the phenotype of F2. How does that work? See this. If it is capital W here, small here, so every single location I will have one capital W everywhere. Yes. So one capital W is present everywhere. It means everything is going to be violet. It is going to be violet. Second, if the phenotype in F2, if one of them is small w, when this small w and this small w they meet together, we get violet, we get white. So when you look at the phenotype of F2 and if you find a white flower there, we will get to know that in this genotype one recessive allele is present. If you look at this phenotype here, everything is violet. It means that there is no recessive here and everything is dominant. So by looking at the phenotype of F2, we can find the genotype of F1. This is the golden lines here. To find the genotype of F1, this one dominant phenotype, we need to look at the phenotype of F2. So based on what do we get here, we can tell what exactly is going to be here. That is all. That is all, students. That is all. How many of you understood this beautiful concept? Tell me. How many of you understood this beautiful concept of test cross? Anyone? Any doubt, anyone? Quickly tell me in the chat. Very easy, right? It is very easy. That's all. So if NCERT makes this simple lines like this, it would have been much more easier. I don't know why they don't give such easy lines. So it would have been much more easier to you. Okay. Now that is test cross. Now now we come to laws. We come to few laws right now. Right? Something called as law of dominance and law of segregation from monohybrid cross. Now what is law of dominance? Now let me tell you, these laws were given by Mendel. So here he is not calling alleles or genes. Here he is referring a word called as factor. Okay, he is referring a word called as factor here. See this. Characters are controlled by discrete unit called as factors. Factors. He is not calling alleles. There is no allele, there is no gene concept here. Factors. Later on we call it gene in the future. Okay. Factors occur in pair. One dominant, one recessive. Yes or no? In dissimilar pair of factors. What do you mean by dissimilar pair? Dissimilar pair means what, students? Capital T, small t. Dissimilar pair of factors. One member of pair dominates the other recessive. Okay. So one is dominant, one is recessive here. Okay. Now, the law of dominance is used to explain the expression of only one of the parent characters in the monohybrid cross in the F1. That is, in F1 we had something called as capital T small t. Only one of the parental is expressing here. The only tallness is expressing here. Why? Why only tallness is expressing here? The reason why only tallness is expressing at F1 is because of law of dominance. This capital T is dominant. That is why it is expressing. Law of dominance. Very simple. Okay. Expression both in F and expression of both in F2. It also explains the proportion of 3:1 ratio obtained at the F2 generation. Right. 3 tall, 1 dwarf. That is basically law of dominance. Very simple. Very simple. Now, what is law of segregation? Very simple. Very simple. I will explain with the help of a small example, something called as lemon juice. Now tell me, students, all of you know how to make lemon juice? Because I know, right? All you need to do is take some water. To that water we need to add some sugar, some salt, some lemon juice, basically lemons, this thing, and completely mix it, and panka is ready. We call it panka here. Very simple. Now students, once we make that entire juice, right, lemon is added in it, right, lemon juice is added, sugar is added, salt is added, water is added. If you want to add a little bit of chaat masala also, okay. Once we add all of them and we completely mix them or we completely blend them, is it possible to remove salt from it? It's only salt, only sugar, only lemon juice, or only water from it? Is it possible to remove after the entire blending? Not possible. Not possible. Now that is called as blending. That is called as blending, where everything is mixed here. Everyone believed that until this point, that is what is happening in our body also. There are some alleles, capital T small t. Whenever we have capital T small t are both are mixing and dominant is expressing. That was the entire phenomena understood by many people till now, until Mendel's. But Mendel came and told them, what is this nonsense? What is nonsense? These two alleles, capital T small t, these two alleles actually do not blend at all. That is, students, alleles do not show blending. Law of segregation or law of periodic gamete says that alleles do not blend. They do not blend at all, right? They do not show blend, or they should not show blending. They do not show blending. Alleles do not mix at all. They may be together, but they are not mixing completely. The two different alleles, they are not mixing completely. That is what was observed by Mendel's. Okay. But if they are not mixing, what exactly is happening, students? What exactly is happening? Okay. If they are not mixing, they will separate into gametes. For an example, let me tell you one simple way to understand this concept. Okay, let's go back to monohybrid cross from where we get the answers. Okay, now tell me students, if there was blending here, would we again obtain this dwarf plant in F2? Is it possible to obtain dwarf plant in F2 if there was blending here? Not possible. Not possible at all. So that shows that monohybrid cross shows that this small t does not go under blending and it is obtained in the F2 generation one more time. So alleles do not blend. That is called as law of segregation. They remain segregated. They do not blend. They remain segregated. Now what is this law of purity of gametes? Understand. Purity of gametes. When two alleles are in a hybrid, each one will segregate into a separate gamete. That is, if you see here also, this particular gamete has tallness, this particular gamete has dwarfness. Even in the case of capital T small t, students, even in the case of capital T small t, when before they undergo F2 cross, they segregate. They segregate into gametes. One gamete is going to be capital T, one gamete is going to be small t. Capital T, small t. And this gamete right here is pure for tallness. This gamete right here is pure for dwarfness. So each gamete is pure for a trait. One gamete is pure for tallness, one gamete is pure for dwarfness. That's all, nothing else. That is called as law of purity of gametes. Each gamete is pure for one trait. Pure for tallness, pure for dwarfness. How many of you understood the beautiful concept? Tell me. How many of you understood, students? Law of purity of gametes or law of segregation. That is, alleles do not show blending. Do not show blending. That is, they separate into gamete formation. And each allele is pure for a gamete. Right? Each allele is here. Each gamete is pure for a trait. Okay. Very easy, right? Very easy. This is law of segregation of gametes. Okay. Understood? Here, can you see each allele has one gamete? Okay. Each gamete has one allele here. Each gamete has one allele. And let me tell you, students, this right here is universal. It is universal. There is no exception here. No exception at all. There is no exception at all. But law of dominance, there are exceptions. There are exceptions in your law of dominance. For example, there is something called as incomplete dominance. Also codominance. So there are exceptions here. But in the case of law of segregation of purity gametes, no exceptions at all. Okay. One small question here to all of you. Okay. Two or more alternative form of a gene is called as what? Allele. So A is going to be 3. There's only one option, student. There's only one option here now. Option number 2. A is going to be 3, only one option. This right here is NEET 2024 question. Last year, oh my god. I forgot that we are in 2025. So, last year paper, right here on the board. Easy question, easy +4, right? All of you agree with me? Easy +4. This question also you see. Again from NEET 2024. Okay. In a plant, black seed color, capital B, small p. Both are dominant over white seed color. In order to find the genotype of black seed, with which of the following genotype will you cross it? To find the genotype of unknown dominant phenotype, you have to find the genotype of dominant phenotype, that sounds like test cross. That sounds like test cross. And in test cross, we always cross with recessive parents. Recessive parents, right? So what is the answer? Small b, small b. Small b, small b. So easy plus 4 marks. All of you agree with me? Easy plus 4 marks. All of you agree? Easy plus 4. Easy plus 4. Okay. So that was your law of segregation. Now we have to study something called as exceptions. Exceptions in your dominance. One is called as incomplete dominance. All of you know. Snapdragon Antrium species. Right. Now I will teach you the entire thing once. If you know the answer to the question which I am asking, type in the chat. Let's start. Can we start, all of you students? Now, what exactly happens in incomplete dominance? Tell me in the chat. Incomplete dominance. In incomplete dominance, students, one allele, right? Out of the two alleles, one allele is incompletely dominant. One allele is incompletely. It is going to be incompletely dominant over the other allele. Okay, other allele. Now tell me, students, how does the F1 look like? Does the F1 look like the dominant allele or recessive allele? The F1 does not resemble both parents. If one parent is white, one parent is red, the offspring is going to be pink color. It is not looking like father nor the mother. Adopted child. Okay, adopted child here. Okay. Example is snapdragon or dog flower. What do you call it? Dog flower. That is snapdragon. Such a nice name. Imagine calling a flower snapdragon or Antrium species. Or Atrium species. Okay, so that is about incomplete dominance. One small cross is there. Let's do a small cross now. Can we start a small cross? Okay, let's do a small cross. Okay. Incomplete dominance, small cross. Okay, let's take parents first. Let's take parents. How are the parents going to be, students? Tell me in the chat quickly. Parents are going to be red flower crossing with white flower. Crossing with white flower. Red flower is going to be capital T, capital R. Capital R, small r. Next step is what? After parents, gametes. What do we get here? We get here capital R. Here we get small r. Done. Next step is called as fusion, where we get F1. What is the F1, students? Tell me in the chat quickly. Capital R, small r. Since it is incomplete dominance, this capital R is incompletely dominant over the small r. So small r is also expressing itself. So this is red, this is white. The offspring is going to be pink color flower. Offspring is going to be pink flower. Pink flower. Next step is called as what, students? Selfing of F1. Selfing of F1. So we take capital R, small r, cross it with capital R, small r. Okay. Now what do we get after that? After that we get the F2. After that we get the F2 here. So let's do the F2. Let's do Punnett square. So capital R, small r, capital R, small r. What do we get here? Capital R, small r, small r. So tell me, what is the phenotype here, students? This is going to be completely red. This is going to be pink in color, pink, and this is going to be completely white. So with that we come to the phenotypic ratio. Phenotypic ratio is going to be what, students? 1, 2, 1, 1 red, 2 pink, 1 white. Okay. What is going to be the genotypic ratio? 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. Okay. So we get your phenotypic ratio as well as genotypic ratio, both are equal here. P are just like test cross, phenotypic ratio is equal to genotypic ratio. It's equal 1:2:1, 1:2:1. This right here is the entire incomplete dominance. Incomplete dominance. Done. Done. Same thing is given in NCERT, same thing is given in NCERT. Red, white, pink flower, Punnett square, 1:2:1, 1:2:1. Done. One small question on the board, students. Next time I will make the questions bigger and brighter for you. Okay, don't worry. Okay. A pink flowered snapdragon plant was crossed with, see, pink color flower is crossed with red flower snapdragon. Okay. What type of phenotype is expected or are expected in the F1? This is again NEET 2024 question. Students, do you realize? Do you realize how many questions come from this chapter? Even after the syllabus reduction, this chapter and molecular basis becomes the most important and high weighted chapters for you. And if you're able to solve these questions, you're good to go. Okay, so let's solve the question. So what do we have? A pink flower, right? A pink flower, that is capital R, small r, is crossed with red flower. Crossed with red flower. Okay, so pink flower is crossed with red flower. Okay, pink and red. Okay, let's do a Punnett square. What do we get here? Capital R, small r, red R, capital R, capital R. So what do we get? Red, pink. Both do we get? We get both. So red flower as well as the pink flower. Option number 1. Option number 1. Okay. Next question. In entrant species snapdragon, red flower was crossed with a white flower, and in the F1 generation, pink flower was obtained, obviously. When pink flower was selved, the F2 generation showed white, red, pink flower. Choose the incorrect statement from the following. Law of segregation does not apply in this experiment. Incorrect statement, students. I told you. Law of segregation is universal, does not have any exceptions. So wrong statement, first one is the answer. Experiment does not follow law of dominance, it does not follow. The pink color in F1 is due to incomplete dominance, yes. The ratio of F2 is 1:2:1, yes. So 1 is the wrong statement. Okay. So with that we come to incomplete dominance also is over. It is over. There is something called as concept of dominance, very small concept and not really that asked before, but let me tell you. This concept of dominance, right, arises from a modification. Now for example, students, there are two different alleles. One is a dominant allele and one is a recessive allele. Now why is the reason? What is the reason for being recessive? The main reason for it being recessive is because it is undergoing a mutation. And when it undergoes mutation, it can become recessive in nature. Now there is an example given here that is based on enzymes. Okay. Now after mutation, three different variants can arise. One variant is a normal or less efficient enzyme. One variant is non-functional enzyme. One variant is no enzyme at all. So when we have dominant allele and recessive allele can be undergoes modification or mutation, and this can arise. When this condition is there, it can still be dominant because it is normal. These two conditions can be recessive. So the whole point is the recessive condition can arise because of the variation or because of mutation. That is all. Okay. Now students, can we start the concept of codominance now? Can we start? the concept of codominance. Friends, are we going fast? I feel the syllabus is getting covered very fast today. Are we going fast? I don't think so, because I'm taking it very basic to basic to basic, but still the syllabus I feel is getting covered very fast. We finished the introduction, we finished Mendel's introduction, we finished monohybrid cross, incomplete dominance, and then we also finished the terminologies. Everything is finished already. Now we are left with what? Codominance, and then we go to the second part of the chapter. Okay. Now let's go to codominance now. Codominance. Okay. It's very simple. Just look at this diagram. Everything is going to get fixed in your brain. Everything is going to get fixed in your brain. Let me tell you. Okay. See this. One blue bird is there, Twitter bird. White bird is there. Completely white, completely blue. But the offspring, can you see, students? The head is white, head is blue, wings are blue, but stomach is white. So what is exactly in codominance? In the case of codominance, some character from here and from here, both are expressed in the offspring. Both are expressed. One dominant allele from here and a dominant allele from here. Both the dominant alleles are being expressed in the offspring. So both dominant alleles are expressing in the offspring. Okay, that is a codominance. That is your codominance. So what exactly is codominance, students? Both alleles are dominant here. Both alleles are dominant here. Both are dominant. Okay, both are dominant. Okay, and the F1 generation, tell me, students, does the F1 generation look like dominant or recessive? Students, tell me, does the F1 generation, does it look like a dominant type or recessive type? What is it? Okay, tell me, students, what does F1 look like? F1 generation resembles both the parents, and the example here, example for codominance is human blood grouping. It is human blood grouping. Human blood grouping is a simple example here. Human blood grouping is the blood example here. Okay, exactly. Okay, both. Very simple, human blood grouping. But students, in human blood grouping, right? In human blood grouping, there is a small concept called as multiple allelism. There's a small concept called as multiple allelism. Multiple allelism. Now, what is multiple allelism, students? What is multiple allelism? Very simple. Usually, what happens is one gene has two alternative forms. One gene has two alternative forms, but in the case of multiple allelism, one gene can have multiple alleles. One gene can have multiple alleles. Multiple alleles. That is called as multiple allelism. And even in the case of human, students, in the case of humans, there are multiple alleles. Like for example, you might have heard, right? There is something called as IA, IB, and I. There are three different alleles. Three different alleles in human. Three different alleles. Now, students, even if there are three different alleles, only two are present in our body. Only two are present in our body. Only two are present in us. Out of three different alleles, IA, IB, and I, out of three different alleles, only two are present in our body. Now, students, what is the difference then? What is the difference between IA, IB, and IO? The main difference is which type of sugar it has. For example, if you look at this diagram right here, the O blood grouping, that is as basically I gene. This is I. In O blood grouping, we have along with H antigen, there is no sugar here, no sugar, extra sugar. But if you look at A blood grouping, apart from the H antigen, there is an extra sugar here called as N-acetyl galactosamine. So in the case of A blood grouping, the extra sugar which is present here is called as N-acetyl galactosamine. In the case of B blood grouping, the extra sugar is called as galactose. It is called as galactose. Can you see here? In the case of B blood grouping, there is galactose sugar here, extra galactose sugar. In the case of AB, both sugars are present. N-acetyl galactosamine as well as galactose, both are present in the case of AB blood grouping. So because of the different type of sugar present there, because of different type of sugar present in O, no sugar at all. A, we have a different type of sugar. B, we have different type of sugar. So that makes the three different alleles so unique, so different from each other. Okay. Now students, I want a volunteer. Who wants to volunteer? Raise your hands quickly. One volunteer from the chat. I will take one volunteer. Anyone wants to volunteer quickly? One volunteer I want. Okay. And tell me your blood grouping also. Tell me your blood group also in the chat. What is your blood group? All of you quickly. One volunteer with your blood group. Tell me your blood group also in the chat. I have to take your blood group, no, on the board. What is your blood group? Okay. Okay, let's take a person has B blood group. Okay. B blood group. Now, what is the possibility? What is the possibility here? B positive, let's take B positive. But let's focus on the B positive negative, that is RH factor, but not required right now. Let's focus on what do you mean by B blood grouping. What are the possibilities? This person right here can have both dominant alleles IB or IB and small I. This small I will not express itself, so capital B will be expressing itself. Let's say one more person who has A blood grouping. IA, both are dominant expressing, or it can be IA and small I. Now what about O blood grouping? In the case of O blood grouping, only one possibility, small I, or it can be represented as by I0, IO. Okay. What about AB blood grouping? In the case of AB blood grouping, both IA and IB both are present in the case of B blood grouping. So that is called as your human blood grouping. Same thing is given in the table form here. Okay. See, A blood grouping can be IA, it can be IA, or it can be IA small I. B can be what? I have told you. What is O? II. What is AB? Capital A, capital B. Both are present. That is what is given your NCERT also. Very simple to solve, very easy to solve. Okay. Same thing is given here. Now students, one simple question. They will ask you very simple questions here. This year, this is a question from NEET 2024 question. 24 questions. See here. AB of blood grouping is an example of multiple allelism. Okay. Multiple allelism. Simple question. One question can come like this. Identify the wrong set of statements with reference to the gene I that controls AB of blood grouping. When A and B are present together, they express same type of sugar, students. Are they presenting same type of sugar? The gene I does not produce any sugar, yes. Gene I has 3 different alleles, yes. A person will have only 2 of the 3 alleles, yes. But when they are present together, one is A is producing N-acetyl glucosamine, one is producing galactose. So even they are present together, they are producing different type of sugars. Okay, this is a wrong statement. This is the wrong statement. See here, when the 2 sugars are present together, are they producing same type of sugar? No. Here we have one different type of sugar. Here we have different type of sugar. So when they are present together also, 2 different type of sugars are there. Okay. Now students, before we go to dihybrid cross, right, there is a small concept called a starch synthesis concept. Very small concept, example for pleiotropy also. Now I'll teach you what exactly is with the help of a simple example. Okay. Now tell me students, how many of you have very good memory? Raise your hands. How many of you have very good memory? I'll teach you this thing. Very simple it is. How many of you have extremely good memory? Raise your hands. I'll drink water by then. Very, very good memory. Okay. Let me tell you a small thing now. Listen to me. If someone has very good memory, they can remember everything in the world. They have very good memory. Does it mean that they will always get good marks in all the subjects? Just because they have good memory, do you think they can get out of 4 in physics and maths also, which requires you to calculate and everything? In biology also, even if you have good memory, if you don't understand the concept or if you don't learn the concept, it is useless again. Yes or no? All of you agree with me? Just because you have good memory in one segment, it automatically does not mean that you are good in everything. Yes? Yes. Now try to apply the same concept in understanding starch synthesis. In starch synthesis, we have a dominant gene. Let's take, we have a gene called as capital B. Okay. This gene is responsible for round seeds. This gene will give you round seeds. Okay. Just because this gene is giving you round seeds, does it also mean that the same gene should also be dominant in other fields also? Just like I told you, just because you have good memory, it doesn't mean that you're good in everything. Doesn't mean that you're good in studies and everything. Okay. Doesn't mean that you're good in everything. Just because this gene is responsible for dominant in round seed, does it mean that it will be dominant in every other field? No, it doesn't mean that. Okay. Same concept applies here. See this. See here. Starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B, small b. Heterozygous produces round seed, and so B gene seems to be dominant. So for shape of seed, it is dominant. Shape of seed, it is dominant for shape of seed. Okay. But starch grain produced, inside a grain, starch, amount of starch produced are intermediate size in capital B, small b. It means that for the character, shape of seed, it is dominant. But for the character, starch grain, that amount of starch present inside, it is not dominant. It is not dominant at all. Okay, it is not dominant for there. Okay, right. So if the starch grain size is considered as phenotype, then for this angle, for this character, starch grain character, the allele shows incomplete dominance. So for one it is dominant, for other it is incomplete dominance. So that is the starch synthesis. In starch synthesis, what happens is, same gene, somewhere it is dominant, but somewhere else it is recessive in nature or incomplete dominant someone. Now here one small concept you can pull out, on small concept. That is, one small concept which you can pull out here is, one gene here, which is capital B, let's say any gene, this gene is responsible for shape of seed and also amount of starch grain. So if one gene has multiple phenotypic expression, same gene has multiple phenotypic expression, what do you call that? That is called as pleiotropy. Don't worry, you will learn that later on. But remember, starch synthesis in pea pod is an example of pleiotropy also. One gene has multiple expression, pleiotropy. Okay. So what is the conclusion? Just because one version of a gene or allele is dominant for one trait doesn't mean it will always act as dominant for everything else. So when it is dominant, it doesn't mean it will be dominant for everything else. If the gene affects more than one feature or more than one character, dominance can behave differently depending upon which feature you're observing. If you're observing seed shape, dominant. But if you're observing the starch, incomplete dominance. Same gene. Okay. Same gene might be dominant for seed shape but not completely dominant for starch grain size. This means that concept of dominance depends on how the gene product works and which trait we choose to focus on. So if you focus on seed shape, it is dominant. If you focus on starch synthesis, incomplete dominance. Okay. So incomplete dominance can be seen in the case of starch synthesis. Incomplete dominance. Same gene. Okay. Beautiful concept it is. Very difficult to understand, but if you understood by reading these lines which are specially made for you, you're good to go. Okay. What is the face? Look at this face. Look at the face here. Okay. So with that, your complete monohybrid cross is over. Completely done. Starch synthesis, we understood pleiotropy also, you understood. In starch synthesis, you understood that one gene, which angle you're looking at it. If you're looking at it from the angle of seed shape, dominant. The angle of starch synthesis, incomplete dominance. Okay. Rama, understand, just because I am good in football doesn't mean that I will be good in cricket also. It means, right, just because I'm good in football doesn't mean that I will be good in cricket also. If you look at the football angle, I'm good. But if you look at the cricket angle, I'm bad. One gene, if you look at the seed shape, it is dominant. But the same gene, same me, if you look at the starch grain, it is incomplete dominant there. It is incomplete dominant. Gene product, the meaning of gene product is that seed shape is a product. Starch grain is a product. So depending upon which product you're looking at, on that basis we can tell something is dominant. Me playing football is my product. How good I'm playing. Me playing cricket is also a product. If you look at me playing football, like I'm dominant. If me playing cricket, I'm incomplete dominant there. So one gene, multiple expression. All of you understood? All of you understood, students, now? That is a very, if you read, let me tell you students, if you read the lines of NCERT, I am very sure 100% it is very difficult to understand. Let me prove it to you. Let me prove it to you. Let me just show you NCERT lines. Okay. If you read the NCERT lines, it is so difficult to understand this small concept. See this, just see this, students. Here. Try reading this. All of you try reading this paragraph here, this paragraph here. This is the paragraph. I'll read for you, just tell me if you understand it or not. Dominance is not an autonomous feature of a gene or a product of that it has information for. It depends as much on the gene product and production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. Now if you read these lines of NCERT, it is so confusing. So instead of reading these lines here, the same meaning I have given here. If a gene affects more than one feature, dominance can behave differently depending upon which feature you choose to observe. Same gene might be dominant for seed shape, but not completely dominant for starch grain size. Okay, this means that concept of dominance depends on how the gene product, starch grain or shape works and which trait we choose to focus on. So which one you're focusing on, you will feel better. Okay. Crazy. Now students, in the terms of two genes, can we start? Yes, exactly. So the dominants are chosen through characters. Yes, it depends on which character you're looking at. Dominance can also vary depending upon which character you're looking at for one gene. For one gene, same gene can be dominant for some character, incomplete dominant for other character. Okay. So all of you should drink some water now. All of you drink some water. Drink some water quickly. So can we start, students, now? Sir, gene product is nothing but there is a character, sir. Yes, the product which you are referring to is character only. Like for seed shape, seed color, all that is products. Okay. Some dog show is going on outside, some dog modeling show. Now, students, inheritance of two genes. Can we start? Can we start, students? Inheritance of two genes, basically dihybrid cross. Very simple to understand, very easy to understand. Okay. Okay. No break for this. Okay. Let's start, students. Let's start. Inheritance of two genes. Okay. First step is what? Selection of parents. First step is selection of parents. Okay. Now the key difference here, students. The key difference, okay. The key difference between monohybrid and dihybrid. The key difference is that in the case of monohybrid, we took a single character. We took a single character. But in the case of dihybrid, we take two characters. That is the main difference. Okay. Two characters here. For example, here we have parents is going to be capital R, capital R, capital Y, capital Y. That is seed shape and seed color. Here we have seed shape and seed color. That is round and yellow. Round and yellow. Round and yellow. Round and yellow. Okay. We are crossing round and yellow with small r, small y, small y. What does it tell me, students? This is wrinkled and green. Wrinkled and green here. Wrinkled and green. Okay. Selection of parents done. Selection of parents done. Next step is called as gamete formation. It's called as gamete formation. Okay. What did we get here? Capital R, capital Y. Capital Y. Okay. Here what did we get? Capital R, small y. Capital R, small y. Done. Done. Next step is what? Crossing. So we get F1. We get F1. Capital R, capital R, capital R, sorry, capital R, small r, capital Y, and small y. This is F1. Now, what is the phenotype here? Round in yellow again. Round and yellow again. Okay, round and yellow again. This is going to be R is going to be round, Y is going to be yellow. Capital, 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 capital. Okay. Next step is called as selfing of F1. Selfing of F1. What do? We take the same thing and cross it again. Capital R, small r, capital Y, small y. Crossing it with capital R, small r, capital Y, small y. Yes or no? Yes, sir. Now, students, here many students find difficult to find the gametes of this capital R. Capital R, small r, capital Y, small y. Here's a small trick for all of you. This with this and this with this. This with this and this with this. That's all. A small trick will take you a long way. Capital R, capital Y, first gamete. Capital R, small y, second gamete. Small r, capital Y, third gamete. Small y, fourth gamete. Similarly, you're supposed to cross it with capital R, capital Y, small y. So once you make the same gametes here, you need to do the Punnett square again. Here is the Punnett square. Here is the Punnett square. So what do we have? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Okay. Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Now students, I want everyone to type the answer in the chat if you know it, one by one. Can we start now? I'll make the entire thing. I'll write the entire thing one go. I will write the entire thing in one go. All you need to do is follow me. If you know the answer, type this in the chat. Okay, first one. Capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Okay, done. Capital R, capital R, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, capital small y, small y, small r, capital Y, small y, small r, small y, small y. Done, students? All of you done? Yes or no? Tell me. Done? Yes, there's no blending. There's no blending. You might think, sir, in one gamete, right? In one gamete, you have used two different traits here. But students, let me tell you, each particular allele is pure for a trait. So pure for a character. This one is for seed shape. This is for seed color. Here I am not writing capital or small r. I am not adding both the traits here. So this particular gamete here, R is pure for one trait, Y is pure for one more trait. They are not mixing here. Okay. Now what is the? Let's find the phenotypic ratio. Okay. So what do we have, students? What do we have here? Okay. Level by right. Where do I write? Next page. Okay. Now, what is the total phenotype? How many are there, students? How many are there? 1, 2, 3, 4. How many are there? 16. 16 is the phenotype totally. Okay. What is the first one? First category is called as round and yellow. Yes or no? Round and yellow. Okay. Let's see how many are there round and yellow. Okay. This is round and yellow. Round and yellow. Okay. Round yellow. Now look at this one. It's round, but it's small y. So can I call it round and green? All of you agree? Round and green. Next is round and green. Round and green. Okay. Round yellow. Round and green. Yes, round and green. Okay. Round yellow again. Round yellow. Look at this one. It's wrinkled yellow. Let's wrinkled yellow. Yes or no? Again, wrinkled yellow. Wrinkled yellow. Okay. Done. Round and yellow. Look at this one. Round and green. round and green. Look at this one now. Smaller is wrinkled yellow. Look at the last one. Green and wrinkled. Last one. Green and wrinkled. Or wrinkled green. Okay. So how many do we have, students, here? Count. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 are there? 9 are there. Round green, how many are there? Count. 1, 2, 3. 3 are there. Wrinkled green, 3 are there. Wrinkled yellow, 3 are there. Wrinkled green, only 1 is there. So 9:3:3:1 is the phenotypic ratio. Now a question will arise, students, what is genotypic ratio also? They'll never ask you, but if they ask you, you should know how to find. Now students, right now I will teach you what is law of independent assortment. Listen to me very carefully. Okay. Let's just listen to me very carefully, students. In the case of monohybrid cross, there was a cross between two different characters, right? Two different traits. Tallness, dwarfness was crossing. But what exactly is happening in the case of dihybrid cross? Dihybrid cross is technically two different monohybrid cross happening simultaneously. I'll repeat again. In the case of monohybrid cross, we had a one character, two traits. One character, two traits happening. In the case of dihybrid cross, we have one character, seed shape, one more character, seed color. Both are undergoing monohybrid crosses together. Now with that concept in mind, I'll teach you one small thing. Listen to me very carefully. Tell me, what was the phenotypic ratio in the case of monohybrid? 3:1, you will tell me. Phenotypic ratio in the case of monohybrid. And just now I told you, dihybrid is what? Two monohybrid together. So one monohybrid. Let's say this is for seed color. This is for seed shape happening together. This character is independent of this character. This character will assort differently than this character. This character does not have any influence on other character. Listen to me. This character will not have any influence on this character. Both of them will assort independently. Remember that. 3 3s are 9. 3 into 3 3s are 9. 3 into 1, 3. 1 3 3 1 3s are 3. 1 into 1 is 1. Nice 3:1 ratio. So dihybrid cross is nothing but two monohybrid cross happening together. That is a dihybrid cross. Tell me, what is the genotypic ratio in the case of monohybrid cross? This was happening in genotypic ratio in monohybrid. Let's take one more. Since right now it's dihybrid. It's dihybrid right now. Okay. One for seed color, one for seed shape. Both are not interfering with each other. Okay. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 into 2 is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. This right here is the genotypic ratio. Done. Genotypic ratio. Done for dihybrid cross. So this also tells you the independent assortment. When two different characters are present in a hybrid, assortment of one character, seed shape will assort independently of seed color. Both will assort independently. We'll get to that. See here. Mendel's law of independent assortment states that when two pairs of traits, that is seed color has round and green, Seed shape has wrinkled as well as round. So seed color and seed shape are present together. Right? Two pairs of contrasting traits are combined in the hybrid. Can you see here? Combined in the hybrid. Right? Segregation of one pair of character. That is, seed shape is independent of other pair of character. So seed shape will separate independently and seed color will segregate independently. That is called as law of independent assortment. So characters will assort independently. Okay, that is called as law of independent assortment. That's all. Very simple. Here's a small question to all of you. They can ask you such questions. Here in examination. What is the expected percentage of F2 progeny with yellow and inflated pod in a dihybrid cross? They're asking you what is the percentage of yellow inflated. Okay. Experiment involving pea plants with green colored inflated pod. So what are we crossing? We are crossing green inflated pod is crossed with yellow constricted. Students, let me tell you, this same cross, dominant recessive, this is just like round yellow as well as wrinkled green. We learned this Mendelian characteristics. Based on these Mendel characteristics, we did the experiment right now. We did the cross. But same thing can be applied to this also. Like how we obtained here, wrinkled green, wrinkled yellow, round green, round yellow. Same thing can be obtained here also. So what do we obtain here? We obtain here, students. First, let's write parent, green inflated, green constricted. Then we get yellow inflated and yellow constricted. Yellow constricted is being the wrinkled green, last one, right? Like how wrinkled green was just one. Here, this also will be just 1. This is going to be 9. This is going to be 3. So 9 is 3 is 3 in 1 ratio. Same dihybrid, right? Same dihybrid cross only, right? Different types of characters involved. That's all. Okay. So remember, students, they can ask you any of 7 different characters for Mendel's experiment. It is not always they will ask you seed shape and seed color. They can switch it up. Here they're asking you seed, they're asking you pod shape, pod color and pod shape. Do you realize? So they are substituting the different characters and asking the question. But the basic question remains the same itself. The basic framework of characters being involved is same. 9 is to 3 is to 1, 3 is to 1 ratio. Now the only thing which you need to do is use the formula. They're asked you what is the percentage of yellow inflated. Yellow inflated is going to be 3, right? 3 divided by 16. So all you need to do is 3 divided by 16 into 100 and you will get the answer. So if you do 3 divided by 16 into 100, you will get 18.75. Right? That's how you will get it. Now sometimes they can ask you what is the percentage of green inflated. If they ask you the percentage of green inflated, that time you're supposed to do 9 by 16 into 100. Simple as that. Or if they ask you what is the percentage obtained by yellow constricted, 1 divided by 16 into 100. So such questions can be arised in examination. Percentage of offsprings. Now there is one more type of question. That time they can give you the number of offsprings also. See this question. In a cross between yellow round and green wrinkled dihybrid cross, a population, see they have given you the population here. In this question, the population of 800 individuals were found in F2. So they have given you the individuals right now. Okay, what would be the number of yellow and wrinkled seeds in the generation? Yellow wrinkled, right? Yellow wrinkled is how many? 3 out of 16 will be yellow wrinkled. So what you need to do is same thing, students, same thing. 3 divided by 16 itself. Instead of doing into 100, you need to do into 800. Do you realize? In previous question, they didn't ask you, they did not give you the F2 population. So into 100 you did for percentage. Here you need to do, there's no percentage asked. They ask you number of offsprings. So all you need to do is 3 by 16 into 800. Okay. So two different genes when crossed with each other, they does not affect each other in expressing, yes, the two different characters, not only genes. One gene can have multiple expression. When two different characters also involved together, they will not interfere with each other. Seed shape and seed color are also not interfering with each other. They're not interfering. Okay. So answer is 150. Okay, answer is 150. Right. So do you realize, all of you, I believe you understand how the question can be framed in dihybrid. Right. They can ask such numericals. So be prepared for numericals in this chapter. Be prepared. Okay. Be prepared, students. Okay. Now, students, we come to one small sad story. Okay, we come to one small sad story. Okay. The sad story is that, imagine you do all the hard work for 7 long years. Like you do the 7 long years, you do the hard work, but you do not get recognition at all. No Nobel Prize, nothing for you. So Mendel died because of some lung disease, lung or kidney disease, I don't remember exactly. Even until his death, his work was not recognized anywhere. He died, students. Mendel died without knowing how important his work is, how much of recognition is given to his work. He died without knowing that. Right? That is the sad story of Mendel. And you might ask me, sir, why did not they give him Nobel Prize? Because Nobel Prize is given only to the living people. That is the main reason why, even in the discovery of double helix model, Rosalind Franklin did not get the Nobel Prize because she was dead before the Nobel Prize ceremony at all. So she was dead. So Nobel Prize is only given to the people who are alive, and also main reason is Mendel's work was not recognized. Now, after many years, 3 different scientists independently worked on Mendel's work, and then only after their rediscovery, Mendel's work came into existence. You know, there are some people in this world who are ahead of time, like Nikola Tesla, right? They think so differently that all their work doesn't make sense to us right now. All of their work doesn't make sense to us right now because they're so ahead of us. We might be thinking what to eat. They might be thinking something else in general itself, right? Their work did not make sense that time. Same goes for Mendel, right? There are main reasons here, right? Mendel's work is published in 1865, but it remained unrecognized till 1900. See, 25 is nothing, no response until 1900s. Work could not be widely publicized because there was no internet that time. Right now, if there is a discovery, you add a discovery in some journal, publish it, becomes famous. Or Instagram you add, you become famous. No Instagram back then, no Google, no internet back then, no internet, right? Factors were stated by a stable and discrete units by Mendel, and Mendel's concept of alleles needed never blend was not accepted by his contemporaries. That is, law of segregation was not accepted. Law of segregation was not accepted by any of the people. They always believed that alleles are blending. That is why we have babies which look different. Like father is there, mother is there. Baby looks like them but a little different. So alleles are blending, that's all. No one believed in factors. No, even if they believed in factors, he could not prove the physical existence of factors. Why? Lack in microscopy. We could not see the factors, we could not see the genes by then. So you cannot see something, you cannot prove it, right? That's what happened. That's what happened here. Use of mathematics to explain biological phenomena was not accepted till now. Not accepted. No physical proof of existence of factors. See, no proof. So his work was completely unrecognized. And I hope hope in heaven he might be able to see all of his work come into existence. Even after his death, even after a century, right? People are still talking about him because of his research. Right now, there is advancement in cancer biology, advancement in molecular biology, this entire replication, transcription, translation, entire gene therapy, right? DNA fingerprinting, all of that basis is core right here. Okay, core right here. Right. Now these are three different scientists whose work was later published. Rediscovery. That is Hugo de Veris, Karl Konz, and Schmuck. Right. Heinrich von Schmuck. Three different scientists who independently worked. Right. They independently worked on Mendel's, and they're like, oh, this makes sense. This makes sense. All this work made sense, and then Mendel's work came into existence. Okay. Sessions will go, don't worry. Okay. Now, students, based on Mendel's work, right? Based on Mendel's work, two different scientists proved something, and they gave the something called as chromosomal theory of inheritance. Now, tell me, students, how many of you feel that this concept is difficult to understand? Raise your hands. How many of you feel that this concept right here is difficult to understand? Raise your hands quickly, all of you, quickly. How many of you feel this concept of chromosomal theory of inheritance sounds very fancy, you know, right? Very fancy it sounds, but it is very, very easy, because the answer you already know it. Let me tell you why. Okay, let's go back to the first slide here. Let's go back here. In today's class, I told you, students, where are genes present? Genes are present where, students? Tell me. Genes are present on the chromosome. All of you agree with that? Genes are present on the chromosome. What is the other name of a gene? Gene is also called as factor. Okay. Now I'll give you a simple clue, simple explanation. Listen to me. Let's take, I'm taking Meghna. I'm putting Meghna inside a car. I'm taking Meghna, I'm putting her inside a car, and car is going from point A to point B. Let's say I have here point A to point B, the car is moving. The car is at point B here. From here to here, the car went. Okay, car went from here to here. If car is going from here to here, the Meghna who is sitting inside the car, will she also go from point A to point B? Tell me. Obviously, if the car is going, Meghna will also go in the way sitting inside the car. Now you tell me, same concept you apply here. Now understand, if chromosome is moving, if chromosome is separating, like how Meghna is sitting inside the car, the gene is sitting inside the chromosome. Genes are sitting inside the chromosome. Now tell me, if chromosomes are separating, will the genes also separate? Tell me in the chat, yes or no? If gene is present on the chromosome, if chromosomes are separating out, gene will also separate out. It will separate. That is called as chromosomal theory of inheritance. That's all. That is all is chromosomal theory of inheritance. Chromosomal theory of inheritance says that the behavior of chromosome and behavior of gene is same to same. It will obviously be same, students. Why? Because genes are present on the chromosome. It will be same. Yes or no? Tell me. It will be same. See here. That is, who observed? The two scientists called as certain Bovary. The two scientists called as Walter Sutton and Theodore Boveri in 1902. 1902, they observed that behavior of chromosome is parallel to behavior of factors of gene. Obviously, students, genes are present on chromosome. If chromosome is separating, genes are separating. Obviously, simple, right? Use chromosome movement during cell division to explain Mendel's law, right? They saw how the chromosome is moving, so gene also has to move the same way. Right? There are similarities. Mendel's conclusion, chromosomal theorem. There are similarities. What are similarities? Chromosome occur in pair. Yes. Factors occur in pair. Chromosomes segregate during gamete formation. They segregate during gamete formation. Factors segregate during gamete formation. All of you know capital T, small t. They segregate during gamete formation. Chromosome also move during gamete formation. They move during gamete formation. Male gamete, one gamete, 22 pairs. Homologous chromosomes separate during anaphase 1. They separate out. Chromosomes segregate independent of each other. Factors will segregate independent of each other. So chromosomes and genes, they're parallel to each other because they're present on each other. Gene is present on the chromosome. So here, right? Now imagine, students, here we have homozygous. One allele is here, one more gene is here. Two genes are present here. If this is separating out, this also will separate out. Okay. Now same thing is given you in NCERT also. See here, G1, S, G2. G1 and S, G2. Chromosome number is doubling here, Okay. G2, anaphase 1. Students, tell me, in anaphase 1 happens. In the case of anaphase 1, there is separation of homologous chromosomes. Oh, good morning. Okay. Sir, they are so focused on chromosome, they're linked with genes. Mendel talks about, correct? Yes. They focus on chromosomes, they got the G movement also. Now see here, students, in meiosis 1 of anaphase, what happens? There is separation of homologous chromosomes. All of you remember meiosis. This will separate, they will separate out. They're separating out here. And now meiosis 2, separation of sister chromatid. Separation of sister chromatid will happen here. Finally, we get the germ cells. Where are the germ cells? If one gene was present somewhere here, it will be present somewhere here now. So behavior of gene and behavior of chromosome is same to same. Okay. Yes, Gopika ma'am is also like, Gopika ma'am, I think you should sleep. It is already late for you. Okay. Dose of energy. Okay. Same thing is given here also. Same thing. Separation of chromosome, separation of gene. The same to same. Okay. Okay, this question I have to still explain. So students, NEET PYQ 2022. Solve the question. Chromosome theory of inheritance was proposed by Sutton and Boveri. Simple question. Okay. Now students, understand. Now all of this theory is okay. But where is the proof? Without proof, we don't agree. We need proof at any cost. And how do we get the proof, students? Now to give the proof, we have one more scientist. All of you ready to give the namaskara? All of you ready to give the namaskara for the proof now? No. The proof for this entire thing was given by Thomas Morgan, T.H. Morgan. T.H. Morgan was a scientist who gave the experimental proof of chromosomal theory of inheritance with the help of a fabulous fruit fly called as Drosophila melanogaster. So with the help of Drosophila melanogaster, he gave an example, he gave the entire experimental proof of the entire thing. Quick namaskara to T.H. Morgan. Okay. T.H. Morgan. Now, you might ask me, why fruit fly, sir? What is so special about fruit fly? 3 or 4 points, write down in your NCERT itself. First thing, okay. First one. They could be grown on a simple synthetic medium in laboratory. Don't even need that, students. Take one banana peel, throw the banana peel in one corner, half an hour you wait, fruitless will come and join the banana peel. Very simple to grow, very simple to grow. Okay. Now, second thing is what? They complete their life cycle in 2 weeks. Very simple. Single mating, third point. Single mating could produce a large number of progenies. Yes. There is also clear-cut differentiation of the sexes. Fourth point. Okay. Male and female flies are easily distinguishable. Also, it has very many types of heredity variation. That is the fifth point. That is body color, wing size, eye color. Everything can be seen very clearly. So here are the main 5 points for fruit fly. Now with that being said, now we come to an amazing concept called as linkage and variation or linkage and recombination. Now tell me honestly, how many of you feel it is very difficult to understand? Because I'm going to make it very easy for you right now. Very easy. For this, I need 3 volunteers. I need 3 volunteers now. I need 3 volunteers. Okay. One is Rohit. Okay. Okay, let's take Rohit first. Okay. Okay, this is Rohit here. Okay. Who wants to be a friend of Rohit? Okay. Let's take Balaji. Balaji is here. One more person I want. Okay. Who wants to? Friends, my eyes are only bad. I'm not able to read this thing only. Okay. Ravi. Let's take Ravi. One more person, Ravi. Okay. So here we'll put on different hair to distinguish themselves. Okay. Color hair. Super same colors. Okay. Okay, done. We have 3 people here. Okay. We have 3 people here. Ready? Ready all of you? Ready? Okay. Now students, we have, let's take Ravi. There are 3 people here to follow. Okay. Now students, understand. Rohit and Balaji, they are best friends. Okay. They are best friends. Okay. Both of them are best friends. They will hang out with each other. Okay. Never mind. Let's take Ravi now. Both are best friends and connection between them is very high. Now students, when I say connection, I mean linkage. They're very best friends, childhood friends they are. Linkage between them is very high. Linkage between them is very high. Now if linkage between them is very high, will they fight, students? Tell me. They will fight, but the fight will get resolved very fast. Like good friends, no. Like we keep telling each other some random things, but we don't fight very seriously. Right? Best friends. So linkage between them is very high. So recombination is going to be very low. Recombination is very low. Recombination is very low. Okay, very low. Now, one more example. Now, Rohit and Ravi, students, between them there is a cold war. I don't know why. Because of whom? We don't know who is the cold war here. They have a cold war going on. The nice smirk also is there. Okay, between them cold war is going on. Their linkage between them is very okay, linkage is very low. If linkage between them is very low, students, what will be the recombination? Will they fight? Dishum they'll give. So recombination between them is going to be very high. Recombination between them is going to be very high. Okay. Now let's take the same example right now and let's fit the example on a chromosome now. Can we do that? See this. Here let's say I have a chromosome here. This is one chromosome. In this one chromosome right here, I have one gene here and one gene here, and one gene on this one corner, on alone gene. This is called as gene A, gene B, and gene C. Okay. Now students, understand, very simple. These two genes are very close to each other. Like how Balaji and Rohit are very close to each other, gene A and gene B are very close to each other. So tell me students, between gene A and gene B, will the linkage be very high or low? Tell me. Tell me. Between gene A and gene B, like Balaji and Rohit, they are very close to each other. Will the linkage be high or low? Tell me. Linkage is going to be very high. Recombination is going to be very low. Very low recombination. Very low recombination. Okay. Now what about gene A and gene C? Gene A and gene C. Tell me now. Gene A and gene C are loosely bound. They're very far from each other. Just like Ravi and Rohit, they're very far from each other. Gene A and gene C are very far from each other. Now tell me in the chat, will the linkage be high or low here? Gene A and C. Here, linkage is going to be very low. Just like here. Recombination is going to be very high. It's going to be high. So the small concept you learned, that's all is linkage. That is all is linkage, students. What is linkage? Linkage is nothing but physical association. Repeat after me. Physical association of two genes on a chromosome is called as linkage. Are they tightly linked or are they loosely linked? Tightly linked is here, loosely linked is here. Physical association of two genes on a chromosome is called as linkage. What is recombination? Understand, what is recombination? Recombination is basically generation of non-parental type. Recombination, where do I write here, friends? Right here. Recombination is generation of non-parental combination. Now students, what if I tell you, you have already learned this? What if I tell you, you have already learned it? You have already learned it. Let me tell you where. I will tell you where. Let me tell you where you have learned it. If you go to dihybrid cross, here students, round yellow, wrinkled green, are they like parents? Round yellow, wrinkled green. So can I tell, in your simple dihybrid cross, these two are technically parental combination, and round green and wrinkled yellow, these two are what, students? Tell me, what are these two? These two are recombinant type because they are non-parental, right? This is parental, non-parental gene combination. So this right here is recombination, product of recombination. Beautiful, right? How we can connect one concept to one more concept. This is what is missing in this chapter. But I believe all of you have learned this. All of you have learned this so many times. Linkage, recombination. But unless you connect the two things, you will not see the bigger picture. Now students, remember, in the starting I told you, Mendel was lucky. How many of you remember that? Students, tell me. In the starting I told you Mendel was lucky. How many of you remember that? In the starting I told you Mendel was lucky. You remember? See here, these two genes are linked very close to each other. These two genes are not linked because they're different chromosomes. Different chromosomes. Here also they're not linked because they're very far from each other. Okay. This will come back. Okay. Right. When the two genes are very close to each other, parents are going to be expressed more. Recombination is very less. That is, when the two genes are very close to each other, linkage is very high, recombination is very low. Basically, they are inversely proportionate, right? I forgot right here. Basically, there are students, basically linkage is inversely proportionate to recombination. If linkage is going to be high, recombination is going to be very low. It is going to be very low. Okay. Now see this. This one you see here. This is going to be so easy for you. This is so easy for you to understand now. Here can you see YW, YW. Are they close to each other? All of you can see. Very close to each other. So if they are very close to each other, linkage is high. Linkage is high. 98.7. Very high linkage. Recombination. Linkage is very high. Recombination is very low. Recombination is very low here. If you see this one, cross B. Gene, this W and M genes. Two genes are very far from each other. This example, they're somewhat far from each other. Far from each other. So can I tell, see, linkage is little less, recombination is kind of more here. Compared to here, recombination is more. Now you might ask me, sir, they are very far from each other, no? Then still why linkage is high? Answer is very simple. Answer is very simple. Even though they are far from each other, linkage is still high because still we are talking about a same chromosome. If imagine if these two genes, when I'm talking these two genes, if they were present in different chromosomes, in such cases, recombination will be higher and higher that time. Since they are talking about same chromosome still, linkage is still high. Okay, so all of you understood this concept? All of you understood this concept? Linkage is inversely proportional to recombination. Linkage is high, recombination is low. Linkage is low, recombination is high. Now, a question arises now. Why was Mendel lucky? Students, let me tell you a small thing here, very, very important. When T.H. Morgan did the experiment, when T.H. Morgan did the same experiment which Mendel did, with the same experiment which Mendel did, he did not obtain the same result. When T.H. Morgan did the experiment, he did not get 9:3:1 ratio. He never got 9:3:3:1 ratio. He never got. You might ask me why? Because students, when Mendel was doing the experiment, in his pea plant, the characters which he took, right? The characters which he took, they were present on, let's say, this is one chromosome, second chromosome, third chromosome, fourth chromosome. All the characters which Mendel took, right? All the characters which Mendel took, they were present on different chromosomes. All the characters which he took they were present on different chromosomes. Now tell me students, when Mendel did the experiment, will he get some nice recombination? All of you think. If all the genes which Mendel took, they were present in different chromosomes, will he get some nice recombination? Genes are very far from each other. When you do the cross, nice recombination you're getting nice recombination. Nice recombination you're getting. But when T.H. Morgan did the experiment, his result was different. When T.H. Morgan did the experiment, all the chromosomes were present on, all the genes were present on same chromosome. So imagine instead of being present on different chromosomes, if all the genes are present in, let's say one is present here, one is present here, one is present here, one is present here. Tell me students, here will you get any recombination? All the genes are very close to each other. Will you get any recombination? Tell me. No recombination. Very less recombination. Here, good recombination. So Mendel was lucky. Because the different characters which he took, right, they were present on different they're present on different chromosomes. That is why he was able to obtain good variety of result. Good recombination is able to obtain. If this was the case with Mendel, right, he would not get any result. That is why when Mendel worked on hawkweed and beans, he got, this was the case. That is why he did not get any result. Beautiful concept, no? How Mendel became lucky. He became lucky because all the characters which you took right, they were present on different chromosomes, and that is why he got nice variety. Okay, right? Yes, he became lucky. He was able to obtain this. But when T.H. Morgan did the experiment, he told us that in F2 conditions, it is not always 9:3:3:1 ratio. Not possible. There can be other varieties are also possible there. Beautiful concept. See here, when two genes are very far, different chromosomes are very far from each other, you get recombinants. Okay. See, Morgan observed that two genes did not segregate independently of each other. Why? If two genes has to segregate independently, it will only happen when they are very far from each other. If they are very close to each other, what will happen? They will segregate together. For example, this is the chromosome. Here I have two genes here which are very close to each other. If this has to segregate, they will segregate together only. They will not segregate independently. Why? Because linkage is very high here and recombination is low. They will not segregate differently. Okay. So did not segregate independently of each other. The ratio deviated. The ratio of 9:3:1 was not obtained all the time. Okay. Okay. Yes, you are lucky. So if the genes present on different chromosomes, recombination is high. Yes, obviously, Rama, understand. If I have one gene here and let's say I have one more chromosome here and one gene is present somewhere here, they can easily undergo recombination because linkage between them is very low. Linkage is very low, recombination is high. Simple as that, right? Right. Morgan attributed this to be physical association of or linkage of two genes. Right. Same genes very tightly linked show very low recombination. So two genes are very close to each other, recombination is there. Some genes are loosely linked. Some genes are very loosely linked. It means they are very far from each other, recombination is high. Okay. Yes, variation can be shown. So if the factors are occur in pair in both parent genes, they're closely linked is very high, but formation of zygote, the variation can be shown. See, variation doesn't technically arise in this recombination. Variation mainly arises in the meiosis. Don't get confused. Variation mainly first arises where in the gamete formation. Later on, because of the recombination, then more variation can arise. Okay. Exactly. More recombination, more variety can be obtained. That's what happened with Mendel. Yes, here. Higher recombination. So genes controlled body color. See, this is what happens in experiment also. If you see the experiment, genes controlling body color and eye color were tightly linked and showed 1.3 recombination. Genes controlling eye color and wing size loosely linked showed 37.2% recombination. Okay. Now he had a student called as Alfred Stewart Went. He observed the frequency of this gene combination. Okay. So the student Alfred Stewart Went mapped the gene position on the chromosome. He used frequency of recombination between the gene pair. There are two gene pair. How frequently they are showing recombination. He observed that, and based on the frequency of combination, he made a new unit called as map units or centimorgan. Okay. Today we use starting point in sequencing of the whole genome called as centimorgan or centimorgan or T.H. Morgan, and his main work was used in Human Genome Project also. Alfred Stewart Went mapped the gene position on the chromosome. He told us that which gene is present where on the chromosome. For example, if this is the chromosome, on this chromosome he was able to tell one gene is here, one gene is here, one gene is here. So that is called a gene mapping. Gene mapping was done by Alfred Stuart Went, student of T.H. Morgan. Okay, one small question here. See, this is a NEET 2022 question. See, if you know the concept, you can answer this question. If you don't know, forget it. See the concept. Mendel's law of independent assortment does not hold good for genes that are closely associated on the same chromosome. All of you agree? So independent assortment does not hold good when two genes are very close to each other on a chromosome. That time law of independent assortment does not exist. Okay, closely related genes assort independently. Students, closely assorted genes, if two genes are very close to each other, do they assort independently? No. They will assort independently when? Only when they are far from each other. So this statement becomes completely wrong. All of you understand? See this. Closely located genes assort independently. No. If two genes are very close to each other, they will assort together. Okay. So what is the answer, students? A is true, R is false. So do you realize, if you want to answer such questions where only around 67% students are able to answer. Why? Because they have understood the concept and not bahaded any of it. Okay, so all of you understood the first statement. Second statement, if the two genes are very close to each other, they'll go together, they'll not go separate ways. Like for example, if you go back to the example which I gave you here, the example which I gave you here was, where is the biology? Here. If Rohit and Balaji are best friends, they will go together. Why will the students tell me? If Rohit and Balaji are together, will they go separately? No, they'll not go separately. They'll go together. Right? They'll go together. But will Rohit and Ravi will assort independently together? Students tell me. Will Rohit and Ravi, will they go together? No. They have fights between them. Why will they hang out? Tell me, why will Rohit and Ravi will hang out? They'll not hang out with it all. They'll go separately. He will go to his house. He will go to his house. Students, tell me, is genetics fun or not? Genetics is crazy fun, no? Crazy fun. In Drosophila, genes for color of the body and color of the eye are situated on which chromosome? This I have not told you. X chromosome. It is present on X chromosome. Okay, remember that. This was a question. Okay. Low frequency recombination indicates that. So, recombination is low. What is the meaning? If recombination means low, means what, students? If recombination is low, what is the meaning? If recombination is low, linkage is high. If linkage is high means two genes are close to each other. All of you agree? Two genes are very close to each other, that's all. Two genes are very close to each other. Like, do you realize it's so easy to solve such questions if you know the concept, right? All of you know the concept now. Genetics, this is why I love genetics. Okay. Now students, with that we come to something called as polygenetic inheritance. Let me tell you students, one more hour. Easily we'll go for 1 more hour. Let me tell you now only. Okay. Because I have to teach you something called as pleiotropy, pedigree. I'll teach you pedigree today. I'll make you write so many things. You will never go wrong in pedigree. Okay. Then diseases, or 45 minutes, maximum 45 minutes. If you want to really finish the chapter, really finish the unit, okay, really finish this entire unit today itself and go back home and tell that, before sleeping, you want to feel that confidence, watch the lecture till the very end. Okay, because I am here ready to teach the entire thing today in one shot. You should also be ready to use your entire brain and learn it in one shot. Okay? Yes. No break. I'll give you 30-second break because my water also is over. Okay. I will give you, let's say, 5 minutes break. Okay. I'll give you 5 minutes break. I also go get some water to drink. Okay. So let's start the class at 12:37. 12:37. 12:37. Okay, 12:37 we'll start again. Very simple. So 5 minutes break, you can go get something to drink, water, hot water, something. Okay. Sound check. I think I'm audible. Perfect. Now let's start with something called as pleiotropy. Let's start with something called as pleiotropy. Very simple, very easy to understand. Very simple to understand. What is pleiotropy? Okay. Now here we have an Moon Knight and Batman. Okay. So I'll explain you in a very simple way with a simple example, and you just follow through. Okay. Now if you go outside right now, not right now in the night time or in the daytime, you will notice that there is a wide variety in the height. All of you have seen, right? Wide variety in height. You will never see only tall and only short. You'll never see that. You will see some people are 5'5", 5'6", some are 5'7", 5'8", 5'9", 5'10", 6'1", 6'2", 6'3". So there is a gradation in the human height. Same goes for human color also. Human height and human color. There is nothing called as completely dark and completely white. No. There is always a middle ground, brown color, skin color. Like we Indians, we are brown, mostly brown, right? So there is a gradation even in the skin color. Now this gradation, right? Human height, change in human height, middle, change in, you see different in human color. All of this is because of polygenetic inheritance. All this is because of polygenetic inheritance. And students, let me tell you, there are two main things here which you need to focus on. The first thing is understand, all of you understand. Okay, polygenetic inheritance. In polygenetic inheritance, right, many genes, right, many genes control one trait. Simple definition which everyone needs to know. Many genes control one trait. For example, simple example I'll give you, listen to me. There is a dominant allele, right? There's a dominant allele for human skin color. So if you, all of you should be aware that human skin color is because of a pigment called as melanin pigment. Melanin pigment. This pigment is mainly produced because of these three genes. Okay, gene A, gene B, gene C. So skin color is because of three genes called as gene A, gene B, and gene C. The number of dominant gene present, more is the effect. For example, if there is capital A, small a, capital B, small b, capital C, small c, this is medium brown color. Now, what if there is capital A, capital A, capital B, capital B, capital C, capital C? This is completely white skin color. Okay, it's going to be completely white or fair. What do you call it? Now let's take one more skin color. Small a, small b, small b, small c. This is going to be completely dark skin color. Why? Because of no recessive allele. Okay, so this is one second. Ulta I wrote off. One second. Melanin is present, produced more here, no? Melanin is produced more here, more dominantly. This is going to be completely dark. This is going to be completely white. Okay, this is going to be completely white here because no dominant allele at all here. So no melanin pigment at all. Albinism, it can be caused here. This is completely brown or intermediate, right? Brown or intermediate. Okay, so depending upon how many dominant alleles are present, the skin color is going to be more and more darker and darker. So more dominant alleles, darker is the color. Okay, darker is the color. Okay, now there can be even something like this also. Capital A, small a, capital B, capital C, small c. So one dominant allele, two dominant, one dominant. Again, this also is going to be a intermediate. It is also going to be a intermediate color, intermediate in shape. It is going to be intermediate in shape. Okay, and students, let me tell you, such type of polygenetic inheritance, right? Polygenetic inheritance can also be environmentally influenced. Environment can also influence here. Environment influence can be there. Environment influence can be there. and there is additive effect. Additive effect. This is called as additive effect. More dominant, additive effect, darker skin color, lighter skin color. Simple as that. That is called as polygenic inheritance. Many genes control one trait. Now, what about pleiotropy? Pleiotropy. See, here we had many genes, one trait, complete reverse. Single gene, multiple expression. Complete reverse. Here we have single gene, many expression. So complete reverse happens here. In the case of pleiotropy, a single gene has multiple phenotypic expression. Multiple phenotypic expression. Multiple phenotypic expression. Multiple phenotypic expression. One simple example given your NCERT is phenylketonuria. Example given your NCERT is phenylketonuria, students. Phenylketonuria. Phenylketonuria. Now you might ask, why is this phenylketonuria even working? Like, what is the main reason of phenylketonuria? Let me explain you now only, so that I don't have to explain one more time later on in detail. Okay, listen to me. Why is it caused? In phenylketonuria, what happens? Listen to me very carefully. Now, what happens is, there is a amino acid called as phenylalanine. Phenylalanine usually gets converted to tyrosine. Now, this conversion is happening because of an enzyme called as phenylalanine hydrolase enzyme. In the case of this disease, phenylketonuria disease, the enzyme is completely absent. The enzyme is completely absent. If the enzyme is completely absent here, I repeat again, if the enzyme is completely absent, phenylalanine is not getting converted to tyrosine. Not getting converted. Instead, it takes a different pathway itself and gets converted to phenylpyruvic acid. Right? It gets converted to an acid. Now, because of this condition, if it gets accumulated in the joints, it can cause arthritis. If it gets accumulated in the brain, it can cause mental retardedness. It can also cause skin pigmentation. Yes, skin pigmentation can cause skin pigmentation, reduction in hair, mental retardedness, mental retardation. So, one gene, multiple phenotypic expression. That is called as pleiotropy. I always remember, right? Listen to me. It's like, I'll give you a simple example. Always examples are better. Okay. Pleiotropy is like a mother. One mother, many work she can do. She'll cook for me. Right? She'll take care of me. She will even sometimes clean my room also if I don't clean it myself. She will even go out to work. Like how your mother, one mother can have multiple works she can do. Pleiotropy is just like that. One gene, like your mother, multiple expressions. Like a mother can do so many things. Pleiotropy also, housekeeping gene. Right? One mother, one gene can do so many things. Multiple expression. That is called as pleiotropy. Simple as that. It cannot get more easier than that. Okay. So one gene has multiple expressions. Okay. It doesn't control 3 genes. It gives multiple effects. Okay. Basically, this gene is going to be part of metabolic pathways. This gene is going to be part of metabolic pathways, and it can alter the metabolic pathways. That is the control. Okay. Now see here, question can come like this. A phenomena where a single gene exhibits multiple phenotypic expression is called as pleiotropy. NEET 2024 question. Easy +4. Easy +4 marks. Or the question can come like this. a phenomenon of pleiotropism refers to more than 2 genes affecting a single character? No. Not more than 2 genes, a single gene is there. Presence of several genes in a single gene controlling a single crossing over? No. Right. Presence of 2 alleles each of 2 genes? No. Single gene affecting multiple phenotypic expressions? Correct. NEET 2023, last year. Or they can ask you a question like this. Select the correct set of statements. A gene is said to be pleiotropic if it affects more than one trait. Phenylketonuria is an example of pleiotropy? Yes, it is. Example of pleiotropy. Right. A condition where one gene has several alleles, it is referred to as pleiotropism? No, that is multiple allelism. That is multiple allelism, student. That is multiple allelism. Okay. A trait is said to be pleiotropic if several genes are controlling it? No, only a single gene. So A and B is the answer. A and B is the answer. Okay, A and B is the answer. See, this is called as multiple allelism. Several alleles is called as multiple allelism. Okay. So with that, we come to the next topic called as sex determination. Okay. Next topic called as sex determination. It's a very easy concept. It'll get over in 10 minutes. 10 minutes, the entire thing will get over. Now, before we go to the entire sex determination, male heterogamety, female heterogamety, you need to understand the main scientist who did the first initial contribution here. I didn't add the image, but I will add the image next class. Don't worry. Okay. The scientist's name is Henking. His name is Henking. He was a scientist whose main work was in insects. Okay. He noticed that some insects had during spermatogenesis, some insects had some nuclear material called as X. Some of the chromosomes did not have the X. He noticed that during spermatogenesis, in the case of insects, some sperms had the X chromosome, X body. He did not know what that was. X body, and some chromosomes did not have the X. Later on, after many years, we got to know the X thing which he was referring to was nothing but the X chromosome. That's all, its extermination. Okay. So Henking, his main work was around in the year 1891. Around 1891, his main work was on. He observed that after spermatogenesis, he observed that 50% sperm, it received X nuclear material. He also observed that 50% sperm did not receive it. Did not receive it. The one which did not receive, right, that he did not know what it was, something was missing. One which was receiving the X nuclear material, he called it as X body. X body. Obviously, if we don't know something, we call it as X body itself. X body itself, right? X body. Later on, we got to know this X body is nothing but X chromosome. X chromosome. Later, we got to know it is X chromosome. Okay. So, what is the difference between polygenetic inheritance and pleiotropy? Polygenetic inheritance and pleiotropy are two complete opposite things. Right? Here, in polygenetic inheritance, many genes, one trait. Many genes, one trait. Here, one genes, many traits. It's complete opposite, two opposite things. Two complete opposite things. Okay. So this is called as the X chromosome. Later on, we call it as X chromosome. Okay. X body is called as the X chromosome. Okay. Now, based on that, in sex determination, there is something called as male heterogamety and female heterogamety. Very simple. Very simple. Understand. First, male heterogamety. Understand, students. Male heterogamety. Okay. Male heterogamety. In the case of male heterogamety, the organism organism will have two different type of gametes. The male will have two different type of gametes. Very simple. See this. See this. Let's take in the case of humans, we'll take first example, humans. Even in the case of Drosophila also, students. Okay. Other example is grasshopper. Example is grasshopper. Okay. So what exactly happens here? Understand. In the case of humans, the chromosomes are what? XX and XY. In the case of grasshopper, it is XX and X0. Now here, the interesting thing is, this right here is the male. This right here is the male. This right here is the male here. This right here is the male. This is the female. This is the female. So what do we understand from male heterogamety? From male heterogamety, we understand that the males have two different type of gametes and they generally decide the sex of the offspring. Obviously, if Y and X chromosome, depending upon this, the male child is going to be male or female. Same goes in the case of grasshopper. The only difference here to here is, here the males will have one less chromosome. Okay. Now let's understand female heterogamety. Very simple, students. Very simple. Many questions have come from here. Female heterogamety. Okay. Very simple. Understand. Okay. Let's take first one birds and then we'll take butterfly. Birds and butterfly. Okay. Here in the case of birds, ZW and ZZ, birds. Okay. ZW and Z. Even the chicken and all comes here only. Okay. ZW, ZZ. In butterfly, we have Z0 and ZZ. Now the interesting part here is here. Heterogamety. Heterogamety. Here the heterogamety is shown by the female actually. This is the female and this is the male. Male have the same type of, males have the same type of chromosome ZZ or ZZ here also in the case of, the females are ZW and Z0. So in the case of female heterogamety, it's the females which decide the sex of the offspring. Okay, that's all. Done. But apart from all that, there is a different type of sex determination in the case of ants and wasp and honeybee. Okay. In the case of honeybees, very different. Honeybees, it is a little different. Why? Understand. In the case of honeybees, the sex determination is mainly based on number of chromosomes or number of set of chromosomes. Number of set of chromosomes. Understand, very simple. In the case of honeybee, the diploid 2n is equal to, one second, there is no water. Water is missing. 2n is equal to 32 here. 2n equals 32 in the case of honeybee. Okay, let's take, I have a male here and I have a female here. Okay, male and female. Male is producing the sperm. Let's take, I have the sperm here. Female is also producing the egg. Is producing the egg here. They'll fuse together. Here, female is diploid. Males are always haploid. Now students, tell me, if the gamete has to be produced, this right here is haploid. Gamete is haploid. Gamete here is also haploid. Tell me what has to happen here. Diploid to haploid. What has to happen? Tell me in the chat quickly, all of you. What has to happen here? Meiosis. In this case, mitosis. Mitosis. Haploid to haploid, mitosis. When they fuse together, the resulting is always a female. Always a female. Why? Haploid, haploid. This is going to be diploid. Haploid fusing will always be a diploid. Always, as a result of fertilization, it is always a female, and this female can eventually grow into a queen or a worker. Develop into a queen or worker. Queen or worker. Always. It can develop into a queen or worker. Now you might ask me, sir, how does the males develop then? Students, from this particular egg, sometimes what happens is, there is a process called as parthenogenesis. A process called as parthenogenesis. Because of this process called as parthenogenesis, from the egg, males will directly develop. Now this was completely haploid. The male is also going to be completely haploid here. Male is going to be completely haploid here. Male is going to be completely haploid. So that is the beauty of sex determination in the case of honeybee or wasp. Females are developing as a result of fertilization. Males are developing directly as a result of parthenogenesis. Now the interesting part here, students, interesting part which I always tell, the most interesting part here is understand. Okay, understand all of you. I'll write in this corner, I'll write here. Okay. Understand all of you. Now students, the interesting part here is, let's take this male. Okay, let's take this male here. Okay, does he have a father? Tell me all of you in the chat. Does he have a father by any chance? This male right here, developing from a female, so no father. So no father at all. No father. Now this male will produce sperm, always result in female, so no son. No son. No son. Okay, no son. Now students, tell me, this female will eventually undergo parthenogenesis and produce a male. Will produce a male. This female will eventually undergo parthenogenesis and produce the male. Produce the male eventually. Will produce a male. Okay. So this particular male, even though it doesn't have a father, does it have a grandfather here? So this male doesn't have a father, but does it have a grandfather? Yes, there is a grandfather. Grandfather is there. Grandfather is there. Grandfather. Okay. Now, all of you students understand. This male doesn't have a son, but this daughter can have a son. So this male has a grandson. Grandson can be there. So grandson can be there, grandfather can be there, but there is no father, there is no son. That is the beauty of sex determination in the case of honeybee. In the case of honeybee. Okay. Crazy. So question can arise like this. See, NEET 2024. In honeybee population, sons do not have a father, but they have grandfather. All of you agree with me. All of you agree. Haplo-diploidy. This is called as haplo-diploidy. Haplo-diploidy. Okay. Haploidy in drones of honeybee is due to parthenogenesis, while fertilization results in the female. Yes. That is why they have grandfather and grandson, because of haplo-diploidy. Right? Haplo-diploidy. So the answer is given, students. What to do? Answer is given for this one. So enjoy the answer. Okay. Select the correct statement with respect to the mechanism of determination grasshopper. In grasshopper, right? Correct statement. It is an example of female heterogamety, students. Tell me. No. Grasshopper is male heterogamety. Male produces two different types of gametes. Yes. Total number of chromosomes, autosomes, 6 chromosomes is same in both a male and female. No. Males will have one chromosome less. XO, right? XO. Chromosome less. All X bear an additional X chromosome besides autosomes. Yes. This question. Exotype, NEET 2022. Exotype of sex combination can be found in grasshopper. Exotype. Okay, exotype. Now, next small concept called as mutation. Can we start? Can we start on small concept called as mutation? After mutation, we have pedigree and then diseases. That is all. Can we start? I think I've left my water bottle somewhere. I don't know where I left it. I think my dog took it and went. Okay. Can we start with mutation? Now, if I have to tell mutation in one word, if I have to say mutation in one word, mutation means change. That's all. Mutation means what? Change. It is a change which can be transferred. It is a change which can be transferred from parent to the offspring. So what is mutation? Mutation is basically a change where in DNA that is transferable. So sudden mutation, sudden inheritable change. What is mutation, students? Sudden inheritable change. We call that mutation. We call that mutation. It is an inheritable change. Now mutation can be basically, right, genetic variation can be of two types. All of you know recombination, there is change. All of you agree? Mutation can also lead to change. Mutation can also lead to change. Now there are different types of mutation. There are different types of mutation. Understand? There is something called as genomatic changes. Something called as genomatic changes. What is the genomatic change, students? Genomatic change is basically change in chromosome number. Change in chromosome number. Now students, here two types. All of you, this concept I have to teach you after also, but learn it right now only. Okay. Now there is two type of chromosome change number. One is called as aneuploidy. One is called as aneuploidy. Other one is called as euploidy. Now what is the difference? In the case of aneuploidy, a single chromosome number is there. Like instead of 46 there will be 45. Instead of 40 there will be 47 number of chromosomes. That is aneuploidy. What is euploidy? In the case of euploidy, there will be entire change in set. What is the meaning of that? 2n can become 4n. Okay, that is change in set. 2n can become 3n. That is called as euploidy. Okay. Now in aneuploidy there is again two types. Understand? In aneuploidy there are two types. One is called as hypoploidy. Hypoploidy as well as hyperploidy. Understand? In the case of hypoploidy, what happens? In case of hypoploidy, there is a decrease in chromosome. Turner syndrome. In Turner syndrome, one chromosome is missing. 45 chromosomes. That is a hypoploidy. In the case of hyperploidy, there is increase in number of chromosome. All of you remember that example of Down syndrome, Klinefelter syndrome, additional X chromosome or addition of 21st chromosome, trisomy 21 comes here, hyperploidy. So that is called as genomatic changes. Okay. Other thing is chromosomal aberration. Chromosomal aberrations. In the case of chromosomal aberrations, there is change in gene structure. Change in chromosome or gene sequence. There can be a change in that. Next thing is also very simple. That is called as gene mutation. Gene mutation. In the case of gene mutation, there is a change in DNA. Change in DNA itself. Change in DNA itself. That is what is happening here. Okay. Now you might have learned today itself. I don't think I have to teach you again. Gopika ma'am. I think Gopika ma'am would have taught today itself. What is point mutation? What is frame shift mutation? In the case of point mutation, you remove one entire nucleotide is removed. Right? One nitrogen base is removed. That is called as point mutation. Single base pair is removed. In the case of frame shift mutation, because one entire nucleotide is removed, entire frame shift moves forward. That is frame shift mutation. I think you studied today this one. Entire thing you studied today itself. And what is the main cause of this mutation? The main cause of this mutation is basically something called as mutagens. There is UV rays, right? Gamma rays. All of these called as mutagens. They're called as mutagens, right? There are certain chemicals, physical factors, UV rays, all these called as mutagens which can cause mutation. Okay. Now students, we come to something called as pedigree now. Now is something called as pedigree analysis. What is pedigree analysis? It is not the dog biscuit. Let me tell you now only. Not the dog biscuit. Okay. What is this pedigree analysis? Pedigree analysis is basically a small concept where we look at a family tree. We look at a family tree and we tell this person in this family has some disease. Right? This person in this family has some disease and we can identify the disease with the help of pedigree analysis. Okay. Right. Inheritance of particular trait is represented in the family tree over generations. In human genetics, it is provides a strong tool for help in tracing inheritance or diseases. Now students, what they can ask you, right? What they can ask you here particularly, they can straight up ask you this particular table. Directly they'll ask you what is mating between relatives, just mating, symbol for it. What is the symbol for male, symbol for female, or they can ask you the symbol for affected. What is the symbol for sex unspecified here? So that straight up they can ask you the entire table here. Apart from that, students, okay, apart from that, they can also ask you like what is autosomal dominant? What is autosomal dominant? Or they can ask you to identify what is autosomal recessive, what is X-linked dominant, what is X-linked recessive. Such questions can be asked. Now students, I'll make you write something very special today. I've always made it right. I always make you write this. If you learn this particular formula, you can solve almost any pedigree question. Can we start? One small thing I'll make you write. All you need to do is write down this entire thing and you will never forget, students. You will never forget. Okay. First thing is autosomal. Okay. First thing is autosomal. Autosomals are your normal chromosomes which are present in the body, not the sex chromosomes. Okay. Autosomal. Okay. Autosomal can be of two types. Autosomal can be of two types. Autosomal dominant and autosomal recessive. Okay. Simple thing to need to, if you want to identify autosomal disease or recessive disease, autosomal dominant students, they do not skip generation. They don't skip generation. While recessive, they skip generation. They'll skip generations. They'll skip generations. If one generation has some disease, next generation will not have disease only. They can skip generation because it's recessive. It can be recessive and not express itself. Okay. Now, here, affected parents can have unaffected children. Unaffected children. Complete opposite here, right? Unaffected parents. Unaffected parents can have affected children. affected children, because can be recessive in nature, so it can be present in next generation. So that is autosomal dominant, autosomal recessive. Now sex linked, understand. X linked. Okay, same again, we have dominant and recessive. Understand, very simple it is. In the case of dominant, students, disease never transfer from father to son. The disease will never go from father to son. First of all, the disease will never grow from father to son. It can never go. Okay. And also, all affected all daughters of an affected father will be affected. So all daughters of affected father will be affected at any cost, because it is dominant. And if it is dominant, one chromosome is coming from the father, one X chromosome, so it is going to be expressing itself. Okay, all daughters of an affected father will be affected. Okay, here in this case, daughters are more affected, women are more affected. Recessive, I'll write one more page. X-linked recessive. Okay. In the case of X-linked recessive, in this case, more males are affected. When it is recessive in nature, it is going to be there. Okay, it is going to be recessive in nature. Okay. Now understand one thing here: the disease will always go from mother to son, like how the favorites they are, and father to daughter. Always father to daughter. Okay, and disease will never grow from, never transfer from father to son. It will never go from father to son. So students, all you need to do is learn these small points and you're good to go. These are called as hack points, which I've learned throughout the years. Okay, there's something called as, if they'll never ask you generally, if they ask you Y linked, in Y linked only males are affected. Only males are affected here. Only males are affected in the case of Y linked. Very simple. Okay, very simple it is. Okay, now see here students, in your NCERT there are only 2 examples given, and these 2 examples are the places where they work around. Like for example, if you see this one, right, if you see this one students, it is not skipping generation, first generation, second generation, third generation. This is third generation, this and this is third generation. It is not skipping generation. And here affected parents can have unaffected children. So this right here is autosomal dominant. And this is an example of myotonic dystrophy. And let me tell you, the only example given in your NCERT, for autosomal dominant is myotonic dystrophy. No other example is given whatsoever. Only a single example, students. Okay. The other example is, students, unaffected parents can have affected children. It is skipping generation. It is skipping generation. This generation does not have it at all. So this is autosomal recessive. Autosomal recessive in nature. This is an example of sickle cell anemia. SCA, sickle cell anemia example. Okay. Now sometimes you get a question like this. Sometimes, ma'am, go because ma'am you sleep off. It's already too late. Okay. They give you a question like this. Same. They'll ask you to find the symbol. Okay. Which one of the following symbol represents mating between relatives in human pedigree analysis? Consanguineous mating. Right here. One more question. Which of the following occurs due to presence of autosomal linked dominant trait? See, dominant trait. Mitochondrial dystrophy. I told you. The only disease in your NCRT. Okay. Mitochondrial dystrophy. Or they can give you the same thing. I told you. Mitochondrial dystrophy, autosomal dominant, affected parents, unaffected children. Does not skip generation. Mitochondrial dystrophy. So they have given you 2 examples in your NCRT of pedigree. Learn those 2 examples to the maximum. You need to identify the 2 examples based on the pedigree and also what they represent. Mitochondrial dystrophy, autosomal dominant. Sickle cell anemia, autosomal recessive. Okay, you learn this much. Okay. Now students, now we come to the last topic for today. That is genetic disorders. Now, maximum 20 minutes, I'll finish the entire thing. Are you ready, students? I know it's been a long time, right? I think we've been live for more than 7 hours. Right? Right. So, these, yes, they are caused because of mutation. And other things. Okay. Can we start the last topic for today's class? Last topic. I'll make it so easy for you. You can solve any question after that. Can we start? Can we start, students? My hair is also giving up on me now. Can we start? Yes? Yes. Now, students, first thing which you need to know about genetic disorders is, before you understand any disorder, I want you to identify which type of disorder it is. Now, how do you do that? For that, you need to make a small chart, which I'm going to make you right now. Okay, small chart. Start. Can we start? Small chart. See here. In general, there are 2 types of disorders. Students. 2 types of disorders. One is called as Mendelian disorder. Different color use. One is called as Mendelian disorder. Other one is called as chromosomal disorder. Okay, Mendelian and chromosomal. Okay, Pedigree does not have a lot of weightage, don't worry. Okay, Mendelian chromosomal disorder. What is Mendelian disorder? Mendelian disorder is mainly because of alternation in single gene. In a single gene. Okay. This is because of chromosomal aberration. I told you already, chromosomal aberration, aneuploidy, polyploidy, right? And changing chromosome number, chromosomal aberration. Okay, chromosomal aberration. The entire Mendelian disorders can again be divided into 2 categories. One is called as autosomal. Other one is called as X-linked or sex-linked. Okay. Now again, autosomal can be divided into 2 categories. Autosomal dominant, autosomal recessive. Okay. Now here the examples you need to write down. Write down a few. Autosomal dominant, I told you there is a single example called as MD. What is MD, students? Myotonic dystrophy. Myotonic dystrophy. Single example. Autosomal recessive, there are 4 different examples actually. One is PKO disease, phenylketonuria. Other example is sickle cell anemia. Other example is thalassemia. And last example is going to be albinism, which is it's not given in NCERT, don't worry. Albinism is also an example here, but it's not given in NCERT, don't worry. 3 different examples for the recessive. Okay. Now what about X-linked? Okay, sex-linked, basically X-linked only. Okay. X-linked. In X-linked, we have 2 different diseases. One is called as color blindness. Other one is called as hemophilia. Okay. So students, you need to remember this much if you want to really get the good marks. Okay. Now, under chromosomal aberration, again we have 2 categories. Two different categories. One is called as autosomal, other is called as sex-linked. Okay. Autosomal, the single example is Down syndrome. Down syndrome. The example in sex-linked is Klinefelter syndrome as well as Turner syndrome. So I'll move aside, write down this entire thing, take a screenshot, write in your book on your own, and trust me, this one table here, very simple table, is going to fetch you a lot of marks. Let me tell you now only. It's going to fetch you lots of marks if you know this one entire table. This one entire table is going to fetch you a lot of marks. Because now we start with color blindness. Very simple. What is color blindness? Color blindness is a simple disease where a particular person cannot tell which is red, which is green. Like imagine they're driving a car, they're going, they see red light, but they think it's green because they can't tell which is green, which is green and which is red. Accident. Okay. And this particular disease is mainly because of the defect in the rods and cones in the eyes. Defect in that rods and cones. Okay. Now what exactly who does it? Right? Who does it? Okay. Yeah. Like who does this and who gets this disease mainly? This disease is mainly seen in the case of males. Females very less. Females it is very less. Okay. Females it is very less. Okay. See here. First of all, this is very important. Sex-linked recessive disease. Here if you go, sex-linked recessive, color blindness. Defect in either red or green color of the cone of the eye. Unable to discriminate between red and green color. Okay. Defect is due to mutation in the gene present on the X chromosome. That's why I call it X-linked. That's why I wrote it here, X-linked. Okay. Now, males suffer more when compared to the females. Females are usually carriers here. Females are generally carriers, around 0.3% I believe. Males 8%. Okay. Males generally affected. Now students, here I generally make you solve some numericals. Do you want to solve today? Generally I make you solve some numerical here. I have a book here. Let me pick it up. Okay. The numerical is very simple. Say you can write different numerical. I've so made you solve many times the same numerical, but this is the most easiest way if you want to start solving numericals in this part. The numerical is if a colorblind man marries a girl with normal vision. with normal vision, what percentage of daughters are carrier? Now let me tell you again one more time, students. You will usually get numericals here. Let me be honest. You will usually get numericals here. You will never, rarely get direct question. Now such question can come, right? How do you solve this? How do you solve this? Listen to me. Right. First we'll find colorblind man. Okay, man is colorblind. So it's a X-linked disease. So C and Y. Man is colorblind here. Man is colorblind. Okay. Marries a girl with normal vision. Girl is normal vision. So XX with normal vision. So in every single cross you do, when every single question you solve, first thing which you need to do is write what is given to you. All of this is given to you. Okay. Now do the Punnett square. Do the Punnett square. XCY XX. What do we get here? XCX X Y X C X and XY. Now tell me, students, what percentage of daughter is carrier now? These two daughters are 100%. This is carrier, this is carrier. So can I tell 100% of daughter is carrier now? 100% of daughter is carrier. Read the question very carefully. They have asked what is the percentage of daughters are carrier. The question is daughters being carrier. If the question is modified, what percentage of offsprings are carrier, then what is the answer? Offsprings are carrier, then 50%. Then 50% will be the answer. Then it will be 50%. Also understood? This entire block is 25, 25, 25, 25. When it comes to offsprings, entire offsprings. But when I talk about only daughter, this becomes 50%, this becomes 50%. So 100% daughter is carrier. Okay, 100% daughter is carrier. Okay, so that is the answer. Okay, do you want to solve one more question? Okay, see this question now. Students, you can get many questions like this and you need to solve many questions like this. Carrier girl. Okay. Carrier girl is marrying colorblind man. Marries colorblind man. Okay. Colorblind man. What percentage of offspring? What percentage of offsprings are completely normal? Completely normal. See this question. Look at this question now. All of you. Look at this question. Carrier girl marries a colorblind man. What percentage of offsprings are completely normal? Students, quickly smash that like button, all of you. There are 600 votes and only 300 likes, sir. Half and half. Scam. Let's solve the question now. Carrier girl, XCH, XCY, X. Carrier girl marries a colorblind man. Let's do a Punnett square. XCX female carrier and colorblind man, right? So this female is going to be colorblind, this female is going to be carrier, this male is going to be affected, this male is going to be normal, this male is going to be normal. The question says, what percentage of offspring are completely normal? Here's the answer. This is completely colorblind female, carrier female, colorblind man, colorblind boy, and completely normal offspring boy. So what is the answer? 25%, not 50%, not 75, 25%. Okay. Okay. But what if I change the question now? I change the question to what percentage of offsprings are what percentage of offspring or percentage of son are colorblind? Tell me the answer now. Sons are colorblind. Sons are colorblind. See here. Son. 100%, 50% is the answer that time. 50% is the answer that time. Okay. 50% is the answer that time. Understood all of you? XCY, XY. 100%, 50% is going to be colorblind. We're going to colorblind. What if they can ask you one more question? What percentage? Not 100. See, this is normal boy, 25%. 50 in this boys, this is boys, sons, right? This sons. Entire thing is going to be 100. Or out of this 100%, 50% is going to be colorblind. 50% is going to be normal. Right? 50% that time. 50%. What if I ask you a question? Out of all the progenies, okay, out of all the progenies, change the question again. Out of all the offspring, okay, what percentage of offspring are colorblind? What is the answer now? Offspring are colorblind. Tell me. Tell me. Colorblind. 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 50% is colorblind. 50% is not colorblind. 50% that time. Because this is carrier. Carrier normal. 50% is going to be colorblind. Not 75, 50%. This is carrier. Do not get confused. This is carrier, not colorblind. XC, it's a female carrier. Colorblind. I hope all of you get it now. 50%. Okay. So there is a NEET PYQ also on the board if you want to solve it. Okay. This is a homework question. Do it in the homework. All of you can solve it right now. Should I do it, students? So let's do it. Night is young. Let's do it. Okay. If a colorblind female marries a man whose mother was also colorblind, if a colorblind female, XC, female is colorblind female marries a man, this is the man, okay, whose mother was also colorblind. His mother was colorblind. Now, this Y chromosome he gets from the father. This X chromosome he'll get from mother. So technically he is also colorblind. All of you agree? Okay. Now carrier and normal are two different words. Okay. Now, whose mother was also colorblind. So female is colorblind. Man she's marrying whose mother was colorblind. So this X chromosome will come from here. So he's also colorblind. It's colorblind. Friends. What are the chances of our progenies having color blindness, students? Tell me. Every single one of them is colorblind. Okay. 100%. 100% color blindness. 100%. 100% color blindness. Okay. So with that, I believe all of you are feeling super confident now in color blindness. Now you have an idea which type of question can be asked in color blindness. All of you agree. Yes or no? All of you agree? I think if you do enough of more practice, you'll get the hang of it. Okay. Next thing, hemophilia. Hemophilia is again sex-linked recessive disease. Now, what happens in hemophilia is, in the case of hemophilia, because of one small cut, it will keep on bleeding. Why? Because there's a protein called as AHG protein, anti-hemoglobin protein. This AHG protein is responsible for the blood clotting. If this AHG protein is missing, there is no more blood clotting. And this disease is seen in the Queen Victoria family. Okay, Queen Victoria family, and mostly males are more affected again here. Males are more affected. Okay. Simple cut result in onset bleeding. Hemophilia may transmit from disease to son. Transmit disease to sons mainly. Queen Victoria family. Okay. Female becoming hemophilic is extremely rare. Mother of such females have to be carrier, and the father has to be hemophilic. So for a female it is very rare. Male it is very normal. This is very important line, students understand. This line is very important. Now here also, students, here also you will get questions like this. You will get questions based on numericals again. One question will solve, only one question will solve, and we'll go forward. Okay. One question is very simple. Okay. If again carrier woman is marrying a normal man, what are the chances of percentage of hemophilic boy? Percentage of hemophilic boy. So you can get such questions. You can form the entire statement and everything, but such questions can come. So carrier woman, right? She is hemophilic. Okay, she is hemophilic. Man is normal. Do the Punnett square. XH, X, XY, XH, X. So we have one carrier girl, normal girl, okay, normal girl, hemophilic boy. Told you boys are very easy to get here, and normal boy. So this is normal, carrier, hemophilic, normal. So percentage of hemophilic boy is again what? Boys is this 100%, boys is 100%, 50% of boys is going to be hemophilic. 50% of boys is going to be hemophilic. Okay. Or if the question, if I change the question to percentage of hemophilic offspring, that time it becomes 25%. Okay, because 3 are, 1 is carrier, 2 are normal, only 1 becomes hemophilic, 25% that time. But right now it is 50% because the question is boys. Okay. One question is here, students, one more question is here. This again is very, very important question. Okay, solve this question, try solving the question. If you cannot solve it, ask me in the telegram if you're not able to solve this question. Okay, answer is also given. Answer is given. See the answer and try solving this question. Okay. Next is sickle cell anemia, students. What exactly happened in sickle cell anemia? Sickle cell anemia is result of point mutation, first point. Second point is that it is also again an autosomal linked recessive disease. It is an autosomal recessive disease, not sex linked, autosomal disease. Now here what happens is because of lack of oxygen, the hemoglobin, right? Because there is a change in hemoglobin chain and the entire RBC will become sickle shaped because of lack in oxygen. Okay. Right. Mutation is there in the HB gene. Mutation in the HB gene happens, causes the abnormal hemoglobin synthesis, resulting in a formation of sickle cell shaped cells. Okay. See this. This is called as sickle shaped cells. Okay. Now what exactly happens here? Okay. What exactly happens here? Understand. Okay. What exactly happens here? Understand, students. Here what is happening here is, can you see in normal hemoglobin there is GAG here, but in the case of, because of point mutation, here we have GTC. Adenine is replaced by thymine here. Because of that there is a change in mRNA, GAG to GUG, and GAG codes for glutamic acid, right? Glutamic acid, and GUG codes for valine. So in the beta globin chain, there is a, in the sixth position there is a glutamic acid. In affected ones there is valine at the sixth position. Now because this point mutation, there is a complete change in the amino acid, entire chain is affected and entire disease is caused. Okay, so it is again a disease, students. Okay, now here quality of your hemoglobin chain is affected. So it is a qualitative disease, not quantitative. It is a quality disease. Quality is maintained, quality is gone. Okay, quality is maintained. Don't worry, if you're not able to solve, I will know. Okay, now here I told you normal person will have HbA if it is normal, carrier HbA when there is a sickle cell person will have HbS, that is very deadly. So if both genes are there, both alleles are HbS, then the sickle cell anemia. One is HbS, that is the sickle cell carrier, normal HbA. Okay, such questions can come. Such questions come in examination. Okay, in a cross between a male and a female, both are heterozygous. See, both are heterozygous. So both are what? HbA, HbS, crossing with HbA and HbS. Okay, sickle cell. What percentage of progeny will be disease? Students, do you realize? In this chapter, questions are always asked based on numerical type. Okay, do the cross. HbA, HbS, HbA and HbS. Here we have HbA, hemoglobin, HbA, HbS. This person is going to be normal, this person is going to be carrier. HbA, HbS. This person also is going to be a carrier. HbS and HbS. This person right here is going to be affected. So what is the answer? Total thing is 100%, 25%. That's all. So this is a NEET PYQ which has been asked, and if you do not know the concept of sickle cell anemia, only 50%, around 50% students can answer this question. If you are in my class, you can answer very easily because right now you know the concept. Good, no? Crazy, no? If you're in my class, right? Yes, X-linked, yes. No. In which disease order single change in students, sickle cell anemia, single base pair change, mutation. Phenylketonuria, I already told you. Phenylketonuria, what happens? In normal condition, phenylalanine gets converted to tyrosine when the enzyme is present. That is, phenylalanine hydrolase enzyme is present. If the enzyme is present, it gets transferred. If the enzyme is absent, what happens? If the hydrolase enzyme is absent, phenylalanine gets converted to phenylpyruvic acid. If this pyruvic acid gets accumulated in joints, arthritis, accumulated in brain, mental retardedness. Okay, remember that. And also you can call mental retardedness, poor absorption by kidney. Through urine it goes away. Okay. What is thalassemia? Thalassemia, it sounds like thalaiva, no? Thalassemia, right? Thalassemia is again a blood disease, but this is quantity disease, quantity. It means that there is a entire chain is missing. Like if you all of you know the quaternary structure of protein. If you remember biomolecules chapter, in biomolecules chapter you learn that a quality in the beta globin, the hemoglobin has 4 chains, 2 alpha, 2 beta chains, right? 2 alpha and 2 beta chains. Now if the chains are only missing, that is called as thalassemia. Okay, again it is an autosomal linked recessive disorder. This is very important. This part is very important, students, every single place. Okay, basically there is a change in alpha chain or beta chain. There is an effect here. Okay, students, all of you know biomolecules, no? Biomolecules is my favorite chapter. And I love teaching a chapter. So please learn the chapter very, very detailedly, because such chapters is a foundation for these chapters. Okay, so learn the chapters very well. Now, what is alpha thalassemia? What is beta thalassemia? Very easy. Okay, in alpha thalassemia, first there are two genes involved. That is gene HB2A and HBA1, and both the genes are present where? On chromosome number 16. On chromosome number 16. Okay, chromosome number 16. Now, if there is alpha thalassemia, in alpha thalassemia, alpha chain is not produced. Beta chain is produced more and more. Okay. Beta thalassemia, single gene. Okay. HBB gene present on chromosome number 11. If this gene gets affected, what happens? Beta chain will reduce and beta thalassemia. So in alpha thalassemia, alpha chain is reduced. In beta thalassemia, beta chain is reduced. Now, student, there is something called delta thalassemia also, delta. Not given your syllabus, so don't worry. Okay. So in alpha, remember the genes, chromosome number, single gene, chromosome number. Beta thalassemia, beta chain is reduced. Alpha thalassemia, alpha is reduced. Okay. You can go check out the series. You can go on the channel and write in principles of inheritance line by line. It is already done. Okay. Yeah. Now see, sickle cell anemia and hemophilia are autosomal, students. Tell me, are they autosomal, both of them? So when you get such questions, best thing is you need to remember the entire table. See, where is the table? See here. Sickle cell anemia, hemophilia. Sickle cell anemia is autosomal recessive. Hemophilia is X-linked. So both are not autosomal. Both are not autosomal. Are they autosomal, students? They are not. Sickle cell anemia and hemophilia are disorders of blood. That is true. Qualitative, quantitative. 2 is correct. Very easy, you can solve them. Very easy. You can go. Which of the following is not a autosomal recessive? Again, just now we learned hemophilia is sex-linked. Hemophilia is sex-linked, right? So thalassemia is sex-linked. Hemophilia also. Hemophilia is sex-linked. Hemophilia is sex-linked. So easy, right? Again, if you have any doubt, you can go to the table and check. Okay. Now, chromosomal disorder, students. Very simple. First is Down syndrome. In Down syndrome, what happens? In the case of Down syndrome, there is something called as, if you look at the karyotype of this Down syndrome person, 21st chromosome, one extra chromosome is there. That is called as trisomy of 21. That is called as trisomy of 21, extra chromosome on the 21st. Okay. Occurs in both male and female. It was first discovered by Langdon Don. Yes. Right. So in this person, there is 47 chromosomes. There are 47 chromosomes. One extra chromosome is there. Okay. Trisomy of 21. It is called as trisomy of 21. Okay. Now here you need to remember the different type of symptoms. Okay. Individually short-featured with small round head. First one. Furrowed tongue. A tongue is outside. Partially open mouth. Like this. Open mouth. Palm has broad characteristic creases. That is, on the palm there are lot of lines on the palm. Physical, psychomotor and mental development is retarded. So all these points you need to remember. Apart from these points, students, remember this also, because sometimes they can ask you. So many loops on the fingertips, very important. Flat back at the head, very important. Okay. Congenital heart diseases can be there. So these three are missing here, but diagram they have given. So they can ask you. Next is called as Klinefelter syndrome. Klinefelter syndrome only happens in the case of male, and there is an additional X chromosome. So XXY condition. And now again, 47 chromosomes in total, because one extra X chromosome. Right? XXY. Okay. Again, what is the symptoms? Symptoms are development of breast called as gynecomastia. Express individuals are also sterile in nature, sterile individuals. Last is called as Turner syndrome. Turner syndrome mainly happens in the case of females, and one X chromosome is missing completely. It is exo completely, exo. It happens in the case of female, and the female develops rudimentary ovary, and the females are sterile completely. Okay, sterile ovary, rudimentary. With that, chapter is done. Chapter is done with that. Here are small questions for you students. The manifestation of elongated area are given below. Choose the incorrect answer from the given below. Mental retardedness, yes sir, correct. This is for Down syndrome. Incorrect statement, Down syndrome. NEET 2024 question. Down syndrome. Look at this question. Broad palm with characteristic creases is visible if person suffering from Down syndrome. Again, Down syndrome. 2023 question. And with that, students, we come to the end of long marathon. I hope this 2 chapters, the 2 huge chapters in the syllabus, which is a nightmare for any student who are preparing for NEET examination, that is molecular basis as well as principles of inheritance. From my side and Gopika ma'am's side, we have done the justice right now. It is up to you right now to do what is necessary. That is, if you go to the description of this today's video, you will find a link where you can attempt the quiz. And students, let me tell you again, right? If you have not joined a test series, please join right now. The offer for test series is still available on the NEET English channel, NEET Vedantu, right? So join the test series right now. I'm telling you, it's very important. The earlier you join, it's better. I think it's available for 799 something, right? So join test series and buy a PYQ book. See, I made you solve some PYQs. You need to buy the books of PYQ and solve more PYQs. That is more important. And if you want to be a part of NEET Express, which is already going on, where we teach even more faster, right? So join the NEET Express, offer is still standing. And with that, we come to the end of today's lecture. Let us know in the comment section of today's video how was the session, and I will see you in the next lecture Monday with biotech. So are you ready? So if you go to the channel right now, right? If you go to the channel right now, Monday we will do one-shot biotech. Ready? Yes. So thank you so much, students. Bye bye. Take care and have a great time ahead. Bye bye, all of you. Bye bye.